Okay, good morning once more. Students, are you happy? Are you ready to face the day again? Yes, but I do hear somebody saying, oh, it's frightening. That's what I heard. Okay. All right, so this is our day 2, and we are happy with how things went yesterday, and we just hope that everyone else had or have some renewed energy this morning to face the rest of the day. So I hope we're all looking forward to the day. So we're not going to have any other thing rather than to proceed with the program. As you can see there, we have a keynote address. We've got our guest, Dr. Anwar Vahed. So he is going to share the knowledge he has in his field with us. And without any waste of time, let me read his short biography. Okay, Dr. Anwar Vahed is the director of the Data Intensive Research Initiative of South Africa, Teresa, abbreviated that way. He manages the South African National Research Data Infrastructure in support of data-intensive research and coordinates the development of relevant expertise, strategies, and policies for research data management. His career spans leadership, managerial, and capacity development roles in diverse academic, research, and operational ICT environments. Dr. Vahed is a computer scientist with more than 20 years of experience in academia, research, and development in the fields of data science, artificial intelligence, machine learning, artificial neural networks, and research data management. He was the principal researcher and research group leader of the ICT for Earth Observation Research Group at the CSIR. Prior to joining the CSIR, he managed the data and information delivery systems of Statistics South Africa and chaired the Department of Computer Science at the University of the Western Cape. He has developed the National Strategy for Research Big Data. He is a review editor of data science journals and serves on several local and international committees in the data sciences, notably the National Advisory Council on Innovation, the US National Institute of Standards and Technology, and the National Institute for Theoretical and Computational Sciences. And then he is going to present to us under the title Open Data, Open Science, Open Minds, Global Trends. Can we please give him a round of applause as he comes to the stage?
Yeah. Good morning, everyone. Firstly, thank you to the University of Limpopo for inviting me, Professor Masoko, Professor Siwia. I'm thankful for this opportunity to address you on a topic that is very close to my heart, and a topic that I've been working on and participating in various forums and committees in establishing what we call open science and open data. and my presentation will also refer to what I've invented, a term called open minds. That should become clearer in my presentation. So thank you once again, and all protocols observed. Before I start on open science, I thought I'd give a little bit of background about what Teresa is all about. And so I have a few slides that clarifies that a little bit. And hopefully the intent with this presentation, or this part of the presentation, is to invite all of you as researchers and research students to join Teresa and to make use of the services that we provide. So just by way of background, as you can see on the slide there, Teresa forms part of a bigger organization called NICIS. That's the National Integrated Cyber Infrastructure System. And together with the CHPC and SANREN, you might know about the CHPC, and SANREN as well, we constitute, that's with Teresa, constitutes the technology component of NICIS. And our mission is, as you can see there on the slide, is for all researchers in South Africa to be able to manage their research data in a proper way, to share data with others and collaborators especially, and to reuse it, combine it with other data sources, data collections, and find new ways to solve some of the problems that we have in South Africa, and to innovate new solutions. So Teresa can be thought of as a structure that is similar to other structures in other countries, such as Europe. In Europe, they've got UDAT, an organization that is responsible for managing the research data of all European countries. Similarly, you have in Australia what is called the ARDC, and in UK, JISC performs a similar function. So Teresa operates at that level. It is a national infrastructure. It's not managed, or it's not, it doesn't belong to any single institution in South Africa. It's managed by the CSIR, but it doesn't belong to the CSIR. It's an institution that was established by the Department of Science and Innovation some years ago. So this structure explains where Teresa fits into the global picture, and you will notice there are several tiers, and we consider, for example, institutional repositories of data as tier 3. Teresa itself is considered to be tier 1, and you can have combinations of tier 1 and tier 2 and tier 3 data repositories. So just to explain what Teresa is and where Teresa fits in. What we do, well, our main task is to provide a reliable cyber infrastructure for everyone in the country who does research to be able to store, upload, manage the data. And I'll explain later on why this is important for us. We have other objectives, such as the development of expertise and also advocacy and outreach, and I'll explain all of that in a little bit more detail further on. So here are the services that we provide for data management, or some of them. Data discovery is a search and browse facility that allows you to find data that might be useful for your research, to combine them with other sources of data, and to conduct research in a more comprehensive way, in a more collaborative way. The entry to the service is through what we call a user subscription process. Basically, you register as a user of Teresa, and that particular icon that you see there allows you to do that. Once you register, you get 100 gigabytes of data storage for free. And if you need more, for example, if you do research that entails the management of bigger volumes of data, come see us and let's talk. We can certainly accommodate, or we'll try our best to accommodate you. So there's a data deposit service, which allows you to upload data. There's what we call a data management planning tool. And there's also a way to allocate digital object identifiers, a very important component, as some of you might know, to identify any digital object, be it data, publications, reports, documents, and so on. Supporting these services, we have two kinds of storage, what we call a hot storage, 8 petabytes, and then what we call cold storage for long-term preservation as well. This is a look under the bonnet. If you open up the hood and you want to see what's inside, Teresa, in terms of technologies, you access the storage facilities and the services through what we call a Teresa Cloud portal, and you can access it at that particular URL, www.teresa.ac.za. That will give you access to all the services that I've just shown in the previous slide. And through that, a storage virtualization service called IROTS, you will be able to access the 8 petabyte storage facility and the 20 petabyte long-term archive, as we call it. Then you'll also be able to access the CHPC, and as some of you might know, the CHPC hosts a supercomputer, massive and advanced computing capabilities, and you can utilize those capabilities and then also interact with the data that you've stored at Teresa. Examples of some of our users. Importantly, these data sets are all open, meaning that anyone, anywhere, can access these data sets anytime. You might know about the South African Medical Research Council. ILIFU is a project run by a consortium of universities, mostly in the Western Cape region, focusing on astronomy. There's a lot more than 40 terabytes that we have there, but the 40 terabytes is openly accessible, and so on. Diplomics, you might know of that, it concerns bioinformatics research. And then also there are a number of universities also subscribing to Teresa. Now, let's turn to the major topic. Open science, open access, open data. I'm sure you are familiar with these terms. What exactly do they mean? Well, this is one perspective of what open science means, what open data means, and what open access means. And as you can see on the slide, open science is actually composed of a number of other concepts. Open access and open data being one of the two most important concepts or sub-concepts of open science. Importantly is the phrase democratization of knowledge. That's what open science is all about. It's about sharing knowledge, it's about sharing resources, sharing access, collaborating, participating collaboratively, and so on. And open data and open access are two of the major components of open science. This is a very simplistic view. If one goes into a little bit more detail, and sorry about the small print there, then open science can be structured in this way. And as you can see from the slide, there's a lot more to it than just open data and open access and so on. And it's important to understand that open science should rather be seen as an umbrella term, a term that captures quite a lot of other aspects. We'll focus on the two issues of open access and open data. So, why should we bother with open science? I've heard from some research communities, some researchers, they say, well, why do we want to share? Why do we want to share the goose that lays the golden egg? You know, all our publications means income for the university. Our research and our projects provides income for me and for the university. Well, the problem here is the data that you collect, that you ask your students to do research on. Examples of this. In the United States, you're familiar with NASA, I'm sure. There are also organizations that store and look after what is called remote sensing data. One of these data sets is called, or data collections, is called Landsat 5. It's data from a satellite covering basically the entire globe in various ways. And the Landsat 5 data was, until 2017, private property, confidential assets. And in 2017, the overarching organization called NOAA, National Observatory Association of America, decided to open that data and to make that data freely accessible and available to the public. And in the following year, 152 SMMEs were established, making use of that data, utilizing that data. Isn't that what we want in South Africa? Isn't that one of the most important factors impacting our economy? So there are many examples of how open science can advance our strategies in South Africa. And what you see here on the screen are just some of them, some of the important ones. I'd like to draw your attention particularly from a scholarly perspective to quality and integrity. Now, those of you who are publishing, you would probably know that there is an increasing trend of what they call publication retractions. People publish papers, and then afterwards somebody else discovers, oh, but there's something wrong here. This is not right, or that has been falsified, and that has been plagiarized, and so on. And in the days of ChatGPT, that trend is exploding. Now, open science, open publications, is a barrier against that. It's effectively open peer review. So many people will look at your publications and ensure that the quality and the integrity of your publications are sound. And that's an important aspect. So let's turn to global trends. What is open science ultimately all about? It's about equality, about equity. And we would know that the global south always features last on the list when it comes to equity, global equity specifically. So for us, not just South Africa, but most countries in the southern hemisphere would benefit from open science. And there are a number of precedents for this. What you see on the screen here are some of the important ones, notably the UNESCO recommendation about open science ensuring equity, fairness, sharing, equal access to knowledge and to data. You may have heard the term helicopter science, the phenomenon where you publish a data set somewhere, say in Europe. There are many examples of this. There are repositories focused on specific disciplines. For example, Elixir is a repository in Europe focusing on bioinformatics information. And what does the South African researcher do? Well, he or she publishes their data at a European or an American, no offense intended, at one of their repositories. And what happens is that the people around those repositories, they get the first bite at that data. They publish the first publications based on that data. So the value effectively transfers from where we are to where someone else is. We need to think about that. We need to think about what we call data sovereignty. We need to ask the question, who's in control of my data? Is it Google, Microsoft, OneDrive, Dropbox, multinational United States or European companies, private companies? Are they in control of our data? And is that right? You see, the issue about control is also a legislative issue. If I store your data, say in Iceland or in Russia or China, then my law applies to that data, the law of that country. So issues of privacy, issues of ethics, transfers to the legal instruments of the particular country that hosts the data. Moving on, closer to home, in Africa we've established, you may know about this, what is called the African Open Science Platform. And the mission is, as you see at the top there, to stimulate interactivity and create opportunities through sharing not just data, but capacities, resources. And the idea behind the African Open Science Platform is to have this federation of nodes that constitute data repositories and so on, to advance data science, to advance AI, to establish an institute, a multinational African institute. And all of this is aimed at promoting collaboration in the various domains and disciplines that affect and impact us directly, not others. By us, I mean Africa. And then also to establish a network of education and skills, especially in the data sciences, and establish a network of open science access and dialogue, a place where we can converse with our collaborators elsewhere in Africa. And already 2 such nodes have been established, one in East Africa and in West Africa. And the picture that you see there shows the countries that are already participating in the African Open Science platform. Even closer to home, there's been developments at governmental level on developing an open science policy. Was involved in the development of the South African Open Science Cloud, which is a part of this. And the idea of this policy is to look at issues of incentives and penalties for those who are beneficiaries of funding from various entities like Department of Science, Department of Higher Education. They are beneficiaries of funding, but they don't want to share the data. We don't think it's fair. Also, there's a move afoot to establish what is called a citizen science hub, the creation of a South African Open Science Observatory, and to federate open science infrastructures in South Africa. All of this is based on a document that was produced some years ago called the South African Open Science Framework. Some information from the Open Science Framework. There are several recommendations on governance, technical implementation, data quality, and so on. There's a roadmap for establishing the South African Open Science Cloud. We are personally, that is NICIS, involved as the implementation agent in the establishment of the South African Open Science Cloud. There's also a declaration by the Department of Science and Innovation. You see part of that on the right-hand side. And NICIS is charged with the task of implementing the Open Science Cloud. Let's have a look at what kind of value is invested in data. This statement from the ex-minister of the then Department of Science and Technology, I think, is still very relevant today. And if you look on the left-hand side, you see some examples of some of the investments in the acquisition and in the collection of research data. You may know of SARIR, South African Research Infrastructures, SANSA, of course, South African National Space Agency, and of course academia, the one highlighted here, which is relevant for us today. You can well think of the kind of funds, the kind of monies that go into the collection and the acquisition and the generation of research data. Of course, the biggest game in town is the square kilometer array. And as you can see on the right-hand side there, you can look at that numbers, projected budget, 2 billion, not rands, but euros. So there's a zero missing there. So far, 650 million euros spent on phase 1 for the SKA. And so far, South Africa spent 2 billion South African rands, 2 billion. That's a lot of houses. Yeah? That's the kind of investment that goes into the acquisition of research data. And so that statement by the ex-minister is very relevant and even more relevant today because the intent is to invest more. And the research community, they are charged with the responsibility to see that we get value from that investment, that we solve our problems, our challenges, that we leverage opportunities, that we support the knowledge economy, the digital economy. It must come out of this investment. So, turning to an issue that I mentioned earlier on, we've spoken about open science, we've spoken about open data, open access, but underneath all of this is the notion of an open mindset. Traditionally, I know I've been at university for a while. You know, when I collect my data, it's my data. I don't want to share that data with anyone because I spent all that effort to collect it, you know. And the point here is that that mindset must change. We've entered a new era of research, right? And the encouraging aspect about all of this is that you can now get recognition, acknowledgement, funding for data investments as in the same way that you get those acknowledgements for scholarly publications, journal publications. Well, there are journals out there where you can publish your data set. And you can get the same kind of funding supported by DHET if you publish in one of those journals. Of course, the journal must be accredited. We all know the game. But there's an opportunity here for you who have collected the data to get recognition and to get funding for it. More importantly, it's critical for us to realize that data connects disciplines. I'm aware of this model where at all universities you have faculties, and within the faculties you might have schools, or the other way around, or you can have departments underneath faculties. Thank you for the Faculty of Agriculture for inviting me. But what we must understand is that that model builds walls between different departments, different faculties. And the issue here is that the funding model encourages that. There's one cake and it has to be sliced up in a certain way, and so everybody competes, all faculties and schools and departments compete for the same source of funding. I know, I've been there. And this model, unfortunately, also causes barriers between disciplines, and that mitigates against actual innovation. And as you can see here from this slide, there's a lot of commonalities among different disciplines. There's a lot of innovation that happens between disciplines. And my thesis here today is that innovation happens in the cracks between disciplines where they join. It's when you combine data from two different domains, that is where innovation will take place. So it's important for us as students, as researchers, as academics, to rethink our view, our perspective of research, of data, of open science. And not think of, for example, these technologies that we are all aware of, AI, machine learning, neural networks. That's not the domain of computer science or statistics anymore. These things are tools for people in the digital humanities, in education, in law, in health, to apply to their research. And it's when you do that that you actually support innovation. So, yeah, we need to think of a world where all data is linked, We need to resolve issues of ethics and privacy. We need to consider open and free. I'll say a little bit more about that. And think about all the other aspects and factors impinging on open science and open data. So in conclusion, ladies and gentlemen, I'd like to leave you with this message. There's a much greater return on investment if we conduct our research in an open way. We also need to realize that all of our data that we do collect is funded by public funds, and therefore the public has got the right of access. We should make data open and fair which is free, accessible, interoperable, and reusable. And that will cause us to increase and broaden research vibrancy in South Africa. It will improve integrity and the provenance of our publications by linking data and literature. And there are new ways in which reward and attributions can be assigned for the process of data collection and data management. So thank you very much for your attention.
Thank you so much, Dr. Vahet, for the informative talk. Colleagues, we're going to open this session for questions. And let's have the mics ready. Any questions from the floor for Dr. Vahet?
Okay, good morning. My question is actually revolving around entrepreneurial things, right? Because I think based on what you have presented, you mainly touched the research and the academia part, right? But on the side of entrepreneurship, right, let's say we want to acquire data, because for example, you mentioned certain bodies like your SANSA, which is for Earth observations and all that. How do you mitigate the issues of data security, right? And at the same time, how do you monetize the data that would be distributed to small, medium, and enterprises and all that? How do you actually mitigate such things?
We realize that not all data can be open. I'm looking for this slide specifically. We realize that not all data can be open. Some data must stay confidential, especially if that data contains what is legislatively called personal information. There's the POPI Act, and that data we don't want to share, of course. But there's also other data. We don't want to share data about where our rhinos are in South Africa, or where our tanks are in South Africa. That would be confidential data. So the suggestion is to adopt this particular data access model, which we have in Teresa. Some data can be completely open. Some data can be shared. Most data is shared, actually. And some data must be closed at all times. And there are examples of those. So if your data contains what one can call competitive intelligence, you know, private companies are very, very sensitive about this. Companies like Standard Bank and Vodacom and you name it, they don't want to share their data. Why? It contains competitive information. And so when it comes to SMMEs, there's got to be some sort of an arrangement, let's call it that, a non-disclosure arrangement, for example, a moratorium on opening data, for example, for data that one might think of contains value for the particular instance or company that has collected it. That's the case in academia as well. There are discussions about moratoria on data. So you can, for example, say, okay, I'll collect my data, I'll keep it with me for 2 years, and after that, it becomes public. Once it becomes public, everybody's got access to it. But prior to that, SMMEs, for example, can use, say, 2 years to leverage the data, leverage funding on advantage from that data. I hope I've answered your question.
All right. Thank you so much, Dr. Vahid, for your enlightening presentation. My question is, what are your thoughts around how institutions can perhaps see as the low-hanging fruits that they can leverage from two perspectives, one at a researcher's level and one at a leadership level, in order to foster that multidisciplinary approach in as much as open data sharing is considered?
Oh, thank you. That's a very, very important question, actually. It's difficult to work against the funding models that are imposed on universities. But there are ways to join hands, and we've seen this, for example, departments of health working with the health faculty as well as the information technology faculty. So at institutions, there should be a concerted and explicit effort. I think I'm talking to the DVCs now. An explicit effort towards funding opportunities, research opportunities, that brings more than one, that brings as many faculties and departments to the table as possible. And funding should be adapted that way. I know the Department of Science and Innovation have adopted that perspective and that approach. The more people from different disciplines they see around the table, the more likely your funding, your research project will be accepted. So there should be a concerted and an explicit effort at, especially by the funders, to promote collaboration. That's at the top end. At the bottom end, researchers themselves can approach each other. People in the Department of Computer Science can go to the Department of Education and say, hey, I've got this intelligent tutoring system, blah, 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 and I've got this data, can we work together on a project? There should be a bottom-up and a top-down approach. That's my view. And in my considered opinion, the top-down approach is the more important one. There should be incentives and penalties for not joining hands.
Thank you very much for a very good presentation. I mean, data is everything. You said it all. But just 2 quick points. One, research funding, we all know that it is deteriorating at a very fast pace.
Yeah.
And this is due to a number of issues, among others, including, your global economic issues. But I'm also of a view that research funding does not seem to be a priority in South Africa. This, you may look at,
Yeah.
but this is my view. For instance, if cuts are made, the first place to go will always be research. Now, having said that, I just want to hear from you, how does the future look given the critical importance of data?
Yeah.
The second question on a lighter note is, what does it take to have access to some of your data? Does it mean that you have to be part of Teresa for you to be able to get access to some data?
Okay. Well, thank you for those questions, two very important questions. First, responding to the second question first. All that needs to be done is to subscribe to Teresa. If there's any kind of difficulty, my email address should be on the presentation. Call me, contact me, and we can make arrangements. Even if you need more than the default amount of 100 gigabytes, and you say, Anwar, I've got this project. I need 10 terabytes to store my data. No problem. We'd be glad to help. On the first question, which is the more difficult one, I wouldn't like to be a doom and gloom prophet, but it looks like things will get worse before they get better. The national fiscus is under tremendous pressure. The amounts of money paid to interest only is terrible. And so we need to climb out of this hole. What's happened is there was a sequence of events that progressively worked against improvement, starting with COVID. After that, there's a lot of other events globally, wars, as you know, and so on, that very, very badly, very negatively impacted the South African economy. And then also in the country, there's a lot of political movements which mitigated against recovery right now. But what I can say, encouraging news, is that there are efforts at improving things. But it's all dependent on the global economy, as you know, and local economy. But we'll go through a few, I would say, if I were to look at the crystal ball, we'll have to tighten our belts for a few years, and then after that, hopefully things would improve. It's up to you students. Thank you.
Okay, thank you. So my question is, I wanted to ask, how is the future protected in terms of us having this free access of information everywhere, in terms of maybe deliberating further, in terms of because now you can see we go to school for information, to get educated and information. So if now we have this open science, data is everywhere and is free, how is the future protected? Are we still gonna need to go to school? And also, according to my personal view of it, it seems like some people are going to lose their jobs because now we won't need to go to school, because now we have this information everywhere and it's also free. And we can see that back then, some source of those information or source of data, it was not free, but as it goes, it looks like it's going to be free, like just gonna get information free anyway. Thank you.
Yeah, thank you for that question. There is a concept called citizen science. I didn't mention it in the presentation. But it basically entails the involvement of the general public in science. And I use the term science in the broadest of ways. I should say research. citizen research, and that means that people at home can get involved, people on the street can get involved in research. It's a brand new, novel concept that we in traditionally, doesn't sound right. But I mean, a few years ago, when I spoke to the taxi driver in Malaysia, from the Malaysian airport to the hotel, he told me about, and he was excited about this big data science hub that Malaysia has established, and how he and his son is involved in developing solutions for the country. The point is here, or the question that we should ask, perhaps one of the questions is, where does the next viral application come from? What's wrong with it coming from that viral application, that new invention after Facebook and Instagram and what you have, TikTok? What's wrong if that new one comes from Limpopo, from Alexandra, from Soweto? What's wrong with that? Isn't that what we want? So it's about changing the way we think about education. Somewhere in that definition of open science, you will also find open access to educational resources, which means that it doesn't matter where you are when you learn. You can be at home. And I think there's generally, internationally, there's a trend towards what we call a hybrid format of education. Sometimes you're at home, sometimes you're at school. There's nothing wrong with that. We need to perhaps reconsider our mindset, our perspective of the way the education and the pedagogy has been traditionally done. You know, the colonialist style of classrooms and what goes with it. Is that the right formula still?
Okay, one last question. I think it's something that I also wanted to add on the question that I was asking you. I'm actually fascinated with the economic point of view, right? And as you know that I think countries, we are trying to address the sustainable development goals, of which some of them is socio-economic factors and environmental factors. But round about that, let me leave it. Now, with data sharing, we have IP, which is in the line of intellectual property. We are even copyright, right? Data sharing, data security, right? So now, if we are going to be taking the information that might have been gathered and we want to incorporate it in a system, for example, let's say we're developing a kind of sensor or kind of system, how do we then ensure that the IP or the information from the data collectors is actually secure and the integrity of it is maintained? And yeah, something of that nature, because now if we are deviating from the fact that we have to monetize it by just buying the information, it's something else. For example, if you take into consideration the South African Weather Bureau, right, there's some sensitive information that to access, you still have to pay in. But how do you incorporate such a thing within a system and without a backlog or something of that nature?
Okay. Thank you. Very good question. We need to distinguish between IP and licensing. The intellectual property can belong to me. The university has got IP over most of its over all the publications. It's their intellectual property. But there's nothing wrong with sharing that intellectual property with others. there's nothing wrong with adopting, if the university doesn't deny this, adopting an open access license to an intellectual property document, a document that belongs. So there's a difference between, and this is actually a cause of a lot of confusion and a cause of a lot of concern, is that these two things are conflated into one. It's two separate things. Intellectual property, that's my car. It's my intellectual property, but you can drive it. So there's a perceived contradiction between, or a tension between intellectual property and copyright, I should say. But it's not, it's a mirage. There's no conflict.
Okay, thank you so much. Thank you. I don't know, is there still maybe just one question? One? Okay, that's fine. Let's Faculty of Science and Agriculture have something special for you. And we will welcome Professor Mukhanedi to come and do the handing over of the token of appreciation. Thank you. You could just come this way. Yes, thank you. Thanks. Thank you. Okay, let's come to the announcements. The first announcement is just to remind the delegates that we'll be using the same halls as yesterday. So SACE remains here in Ivory Hall, but we've got something which is slightly different today, because if you check in your electronic programs, you will see that for SACE we have parallel sessions. So all the senior postgrads and staff will be doing their presentations here in Ivory, but then all honor students, you will do your presentations in the foyer of this hall, just here. And that means that you will first attend the plenary talk, and after that, you will then proceed to the foyer. Then SMLS is still Capricorn Hall, SMCS and SPMS, Mujaji Hall. Then, to remind you again, in the foyer of Ivory, remember we do have the sponsors who are here. Then please do visit their stations during the breaks. In terms of the afternoon tea, we will have the afternoon tea served at the halls where you will be presenting. Please just remember that you don't have to come this side. This is just to try and lessen the journey, the trips that you have to make to this hall. Then we've got water bottles where we are seated. Please, when you move to the other halls, take them so that you can have something to drink. And then the students that are, or let me just say presenters, before you move to the halls, and also especially the Ivory, those who are presenting here can load their talks before they join others for tea. But those who are going to the other halls, of course you will have your tea quickly and then proceed to the halls where you will then load your talks quickly. And the time, it's now 9:40, so it's 20 to 10. Let's have 10 minutes for tea. This is a quick one indeed. And we will then have to start at 10. 10 o'clock, we should all be seated in our halls. Thank you so much.
IRC. ARC is Agricultural Research Council. He do have an extensive experience in his research, ranging from the rangeland ecology and management, with his special focus on herbivores and their interaction with vegetation and factors governing their behavior. Today is going to present ecology and management of bush encroachment, for which is a paradigm shift. So let's give hands of applause to Dr. J. Chelele. Come on, sir.
Best, second last. So, yeah. Thank you. Good morning, ladies and gentlemen. First of all, thanks, Dr. Slepe. I'm also to think it's the two elephants on the stage. I would first like to thank the invitation by the university management, as well as the organizing committee. And in particular, I would like to thank Prof. Mandiwana for all the communications leading to today. But most importantly, we often don't appreciate enough the admin people. Thanks to Mayor Josephine Madi, who arranged all the logistics. She really made my stay very easy. Thank you very much and keep up the good work. One more thing, in South Africa, in the main, and scientists know it, we often work in silos. So this kind of platform must be appreciated because you get to know who's doing what, and that creates an opportunity for collaboration. And to students, we have all been students, and this kind of moment, sometimes it's threatening, but trust me, we all have gone through this. So just make sure that during breaks and lunch, you sit with someone you don't know. Let me get on to business of the day. As they said, my name is Julius. I'm the research manager for the Range and Forage Sciences. I've done a couple of things, including being the president of the Grassland Society of Southern Africa in the past 7 years or so, among a few more other things. I just want to, before I get to the crux of why I'm here, I'm fortunate to be leading a team of rangeland scientists whom we underpin to develop knowledge opportunities offered by dynamic Southern African rangeland environment, in the main to make sure that to ensure favorable ecosystem processes or services for a wide range of sustainable usages by people and industries. There are a few key words that I have underlined here, and they will come through as I take you through the research that we have done in the past 15 to 20 years. And for us to be able to achieve what we stand for as Range and Forage Sciences, which is part of the animal production of the ARC, is that we develop research and service excellence for diverse rangeland environments. So we're working across all the biomes in South Africa, which present various challenges and opportunities. And having done that, we also facilitate technology transfer to end users because research is meant to solve challenges that we faced with. But most importantly, as universities do, including UL, is that we also develop human capacity. So the project or program that I'll present here today has produced over the years over 20, almost 30 MScs and PhDs with various institutions. But equally, we are about promoting equity and wide information sharing. It's pleasing to see quite a number of women presenting here. But most importantly is that we integrate our scientific research with indigenous knowledge system where possible. And the reason why we're here is network sharing. So at the end of it all, you will have my contact details, so please take them for future engagement. Coming to the business of the day, I would like to take you through the research that we have done in the main on ecology and management of bush encroachment. And I've added a paradigm shift because bush encroachment has been there for many years, and it is increasing at a very fast rate. And so the approach that we are taking is that we need to have a paradigm shift in terms of our approach to science, given how it has increased over time. And we also know that climate change exacerbates land degradation, and bush encroachment is another form of land degradation through drought, desertification, and other extreme weather events that are increasing in frequency and in intensity. I've just demonstrated or showed some few photos. The one on my left, which will be your right, it's a Senegalia mellifera. I was in Botswana 2 weeks back, where I was invited to share the work that I've done on bush encroachment, given that Botswana and Namibia, as well as South Africa, are hard hit by bush encroachment. So these were some of the sites that we visited, and you can see that under the canopies there's literally no grass, and you wonder what animals therefore feed on. The picture on the middle, it's some of the situations that we faced with where there's literally nothing, and the reasons that underlie this kind of situation varies quite a lot from one site to the next, or one country to the other. And I'm going to expand or explore on that as we go. On the far right, some of you may be familiar with the plant. It's called bankrupt bush. I mean, literally it bankrupt your rangeland, or its scientific name, it's Seraphium plumosum. It's encroaching quite extensively in the grassland biome. If you have driven on the N4 from Pretoria to Emalahleni or Nelspruit, on both sides of the road you will see a grayish shrub of about 1 meter that's really taking over the rangeland. And you can imagine if you're a farmer, you have got this kind of farm, really, what's next? There has been a question that I always like to start my presentation with, particularly when talking about bush encroachment, which was posed by Prof. Dave Watt. He was my PhD mentor and my collaborator at the moment. He's with Kent State University in the US. He asked a very pertinent question to say, do we really understand the causes of bush encroachment? And this question is very critical, because if we do, then we should be able to develop management strategies to reduce it. But now you wonder whether if we really do, given the rate at which it's increasing, the rate and the extent of it, it's really alarming. And that is in the main because of quite a number of complex systems or ways that promote bush encroachment. One being the local and global factors that drive bush encroachment. And having asked the question of whether if we understand the causes of bush encroachment, The next question is, do we now really have to pursue the management of it, or we must just accept defeat and coexist with it and see how we can, coexist with bush encroachment, maybe in ways that it can benefit us instead of trying to manage it, because it seems like the more you manage it, the worse it becomes. And I will always show, pictures of some of the sites that we working on or we worked on in the past. Just touching on some of the main drivers or causes of bush encroachment. At a global level, we know that elevated atmospheric CO2, which fertilizes woody trees, is one of the key drivers that leads to, the increasing rate of bush encroachment. But at the same time, climate change is a reality. We all feel it in many ways. One being, we're not getting rain as we used to get. It's very erratic. It sometimes comes late. Sometimes we don't have rain at all, or we have, floods, as well as extreme temperature, which also plays a role in facilitating bush encroachment. But at a local level, there's quite a number of drivers as well. One being animals that are often used to manage bush encroachment, and goats being one. We know that goats are browsers, and in the main, they will feed on pods of various woody plant trees, particularly during the dry season, because they are the ones that have got, are more nutritious than grasses. But as a result of that, given that the majority or most of the woody plant species seeds are hard-seeded, cold, once they are consumed, they get, scarified through the gut and then get spread in the rangeland. So in return, animals also do contribute to the spreading or the very same problem that we're using them to, control them. But there's also a big local factor, which it's anthropogenic factors. And in simple terms, it's people who are managing and living in the rangelands. We have heard of a concept of overgrazing for as long as we were doing rangeland science or planted pasture 101 some thousands of years ago. And even today, it's still the case. So, and the frequency and the intensity of fire, of course. So you can see how complex this challenge is, and such that you cannot say the results that you got in a particular site, you can then, say this is what may be the case elsewhere. It varies from one side to another due to a number of interacting factors that some of them, I've mentioned. There's a paper written by Dave Ward as well, who has indicated that overgrazing, it's not really the driver, because in that trial or in that experiment, grazing was optimally, but the site was still encroached heavily. And now you wonder, what is really the reason here? And I'm just going to start on a very simple note with some of the work that we have done in the past, which really tried to test the point that I was referring to about animal contributing to bush encroachment. Basically, we wanted to see if when animals are fed with pods or seeds, what happens when they come out. And it was very clear that the scarified pods or seeds germinated more than the control, as you may see. But of very important is that the seeds that were mixed with diet and fed to these goats also relatively increased in their germination, which proves the fact that the passage through the gut plays a role, and it's the point that I was saying. But there's also quite a number of factors that drives just this small component. One being the quality of diet. We know the concept of good quality diet, that the seeds will pass faster through the gut versus the low quality, which will have a longer retention time, and that has got implications, for seed scarification and ultimately the viability of those seeds. But the plant species as well as the animal species and the seed characteristics, the size, the shape, all plays a role. And these are very minute factors that contributes to animal seed dispersal. But given this work, it opens doors for many. Maybe just to jump to the other aspect of the work that we do. Yes, trees are many. They're providing, source of feed during dry season, as I said. But the problem, as I've mentioned, is that animals do facilitate, the spreading of viable seeds. Then we went back and say, given that this is a resource that is there, and that starts to answer to some degree a question of coexisting with bush encroachment or managing it. Perhaps one way of managing it is to use it to our advantage, such that whilst we are managing it, but it is beneficial in some ways. And we have a project which is still running at the moment, and there is an MSc and a PhD, two MScs and 2 PhD students who are looking at various aspects of this project, where we, in the main, harvesting, shoots, pods of various tree species. But because we know and learned from the previous experiments and work that has also been published that animals do drive or facilitate seed dispersal, we then brought in the feed additives, which improve the quality and also the scarification. But at the same time, we brought in the chipper, where the seed pods are crushed such that they will not be spread viably as they used to. But here, what we're demonstrating is the value that woody plant species, in particular Vachelia nilotica, or what is previously known as Acacia nilotica. What happens in terms of when you add feed additives, to what extent does it improve the feed quality? Because there are two things that we're looking at, or that we are doing here. One is to make fodder out of this nuisance, bush encroachment, which will then start to close the feed gaps. But at the same time, we're also managing this increasing bush encroachment, such that gives room, for herbaceous layer. And some of the key points here is that you realize that when Vachelia nilotica shoots alone, they provide, or they contain insufficient, for instance, crude protein to maintain livestock, which is just below 7%. But seed pods alone contains relatively high, crude protein. But even further, when you add LS33, which is the feed additive, that significantly improves the quality of the feed. And that starts to address, the feed issue, as well as reducing the viability, spreading the viability of the seeds. And the next set of results then start to look at to what extent does these processes facilitate or reduce, seed recovery and ultimately seed germination. And one of the points that I would like to draw your attention to is that the feed additive was found to increase the ingestion of chipped seed pods, which is a good thing, as well as shoots mixed with chipped pods. And this was not seen with whole pods. And maybe the last part that I can just say to move on and to save time is that the time to first seed recovery after seed ingestion did not differ as much with chipped versus the whole seed pods. However, the time to maximum seed recovery was significantly longer for diets containing whole seed pods and cheap material. So in the main, you can see that we are starting to make some inroads in terms of using this bush encroachment meaningfully, and to some degree then start to manage it. There was another project that it's still continuing, but certain parts of it is done. And I had two PhD students from this who both completed, and one of them I was working with Prof. Ngambi here at the University of Limpopo. But the first part was because there are two aspects to it. There is a component of ecology. This is understanding the functioning of various woody plant species, but there's also the management aspect of it. So it is important to understand how certain trees or plants function in such that upon understanding you can be able to understand or develop management strategies. And there were a few key questions that we were answering here, and one being the effects of environmental factors on bankrupt bush or Seraphium plumosum encroachment, susceptibility in the semi-arid grassland community. This was such that we can zoom in to look at the differences in a community. As I said, that there is so much variation, in a landscape. So we needed to zoom in to understand further what happens within a grassland community. But we also looked at the effects of abiotic factors in the semi-arid grasslands communities of South Africa in terms of its intensity and canopy size. And thirdly, we explored the effect of this plant on the canopy cover, the fire, soil depth, and components of soil fertility in the semi-arid grassland society of Southern Africa. There was a student who presented, I think he's working with Prof. Dlamini, and as you alluded yesterday, we owe each other some discussion because there are some common areas that we can explore further given the work that your student has presented on. But we also look at how does fire season and slope position have an effect on forage quality, if at all. And the last part actually gave birth to another project, but I'm first going to show you some of the results that we got. For instance, here, when you look at the interaction of grassland community and slope position on Seraphium, density, you will realize that unfortunately I don't have a pointer, but I'm hoping that you can see. You realize that in the two grassland communities, there were completely different results in terms of the density of this plant. I mean, our hypothesis was that we used the resource allocation model that says that when resources are moving from a top to the bottom, plants that are found at the bottom tend to increase in density and cover as opposed to those that are found at the top, slope. But the opposite was true. There was no, difference at all in the Carltonville Dolomite grassland community, regardless of the slope position. But the opposite was true in the Rand-Highveld grassland, where the density was higher at the bottom than it was at the top and the mid. So this starts to show how complex, the system is, and such that you can't use a blanket approach to get to a decision. And here I just want to, never mind the noise of the graphs, there are two aspects that I want to illustrate to you in terms of the interaction effect of fire and soil depth on various components of soil fertility. And this is the work I was referring to, that it relates very well with what was presented yesterday. Here, the message is that the study has shown us that fire improves potassium and phosphorus concentration, particularly on the surface soils. And so here we were exploring in the main the interaction of the different soil depth as well as fire, whether if it has an effect or not on the soil fertility, which we know that that has implications for either your biomass or your species composition and richness and so on and so forth. The last question, objective that we were posing was to say, we know that there has been reports that animals do nibble on Seraphium at certain parts or time of the year. That starts to tell us a story to say what then happens at the time that animals browse it versus when that they not, which refers to your plant secondary metabolites and in the main your nutritional characterization of the plant. And therefore we looked at various aspects, one being season and the other being fire, to say what happens after this plant has been, because it also has a history of being prone to fire. And as you may see that in terms of the crude protein, you will realize on the first graph, which is A, you will realize, I'm sure you can see the cursor, that during the dry season and after fire, there was no difference. And the same is true with fire. And the same was true with during the dry season and no fire. But it was opposite during the wet season and after fire. So this is the regrowth after, from the dry season when we burned and then we had rain and we collected material, just to look at to what extent does it change in terms of the crude protein. And it pushed up to just over 7%. And even though it was not pretty much high here, but it was higher than in the dry season with fire and without fire. And the opposite was true, which is an inverse of your crude protein. And this then told us that it means that there is, to some degree, we can be able to manipulate this plant and make use of it. And that's where a PhD student that I said I was working with, Prof. Ngambi, who graduated last year, the PhD student, we then said that or maybe before that, I'm too ahead of myself, still on the previous part, there were a few take-home messages that I want to leave you with, but particularly on the previous aspect that I was referring to, is that although fire may be used to control Seraphium plumosum, caution needs to be exercised as it may contribute to improve the condition of the very same plant, which is one of the things that I said earlier on, through improving its soil nutrient status. And once again, I'll keep on hammering and trying to demonstrate the linkage with some of the work that were presented yesterday. And fire also reduces bush encroachment, while at the same time, to some degree, improve forage quality. Which is a combination of fire and, of course, browsers, which can be explored as ways in which that can be used to manage this problem. And the last part is what I was then starting to talk about, to say more studies needed to be explored to look at the use of Seraphium as a forage for livestock. Hence, I was then saying, we then say, what happens if we were to bring in a nutritionist? I'm not a nutritionist by training, hence I went to work with Prof. Ngambi. What happens if we formulate diets out of this, bush? And we look at few parameters, one being the animal performance, quite a number of, animal performance, but also the methane production. But we took it further and say, let's bring in the meat scientists. I did not present the results on the meat characteristics, to say, what happens when we formulate diet? Does it have any positive or negative effect on animal performance parameters? to what degree does it reduce or even increase methane production, which is again in light of climate change? And if so, does it have any improvement in terms of the meat characteristics? And the one component that still needs to be done is to look at the economic side of things. So one of our approach to our research is that we're using a multidisciplinary approach. So we don't just look at rangeland, because rangeland are used by animals, by animals, so animal breeders and animal nutritionists comes on board. Ultimately, they are meat scientists, that these plants that we are feeding has an effect, so that we look at the whole value chain. In fact, the other message that I'm saying here, apart from the results, is that if somehow you have interest in this work, see how you can fit. It may not necessarily be on this aspect, but on other various aspects that I'm still going to talk about. So we then said, because there hasn't been any work done on this work, let's start to explore this by including a 10, 20, and 30% and see what comes out of it. And I'm just going to show you some of the results. This student panicked quite a lot when the results didn't show any significance. You know how you are, students. You want things to differ. But the fact of the matter is, these are results. They're telling a story. It may actually be even a good story. As much as there were no, and I'm just showing just a few parameters just to demonstrate the value that can be derived from this. That the body condition remained the same, the feed conversion ratio as well as the daily, live weight gain. There was no difference. And that starts to say that it means that this plant can actually, or has the potential to be used as one of the ingredients given, even though the protein content is not that high, but there is some margins that you can bring in, which will mean that you will reduce on a traditional protein source that you use. And that has implication in many ways, as I said, either from the meat side of things or from the performance side of things, but most importantly to a farmer, from the rents and cents, which is what it is about. So we're also moving, which is another important in the main, that basic research is important. We do basic research, but we're moving more towards applied research. To what extent does our work is applicable, to the end users, such that we can derive value out of the work that we do? And this is also a mandate of the ARC overall. And in terms of methane emission, which is a big word as well, particularly in a changing climate, these plants, have some significant amount of plant secondary metabolites, particularly condensed tannin, which is why naturally animals don't eat it as much in the natural felt. But due to the manipulation of the feed formulation that we did, animals at the beginning struggled to eat it, but over time they actually preferred it even over our control. And you can see that the 30% inclusion reduced methane production. And this is, again, as has been said by quite a number of authors, and I've just cited one, that it's most likely due to the level of tannins that it's found in, which has an effect on methogens. So this is just to demonstrate that we were looking at various aspects, and there were really promising results, even though I have a feel that we need to relook at it such that we can even look closely at the phenological stages of this plant, so that we can have conclusive remarks. So I've spoken of animal seed dispersal. I've spoken of creating feed out of the woody plant encroachment. I've also looked at the ecology side of things in terms of the causes and consequences of bush encroachment, but in particular I was looking at Cerephium plumosum. We also know that woody plants has some value in it, and one of them is the use of or as anti-parasites. I mean, small stock farmers in the country, particularly now the rainy season is coming, the one keyword that they will complain to you about is the amount of internal parasite and the money that they use to, to buy medication for that. So we said, let's look at the number of species and to what degree does it reduce, the internal parasite load. And I'm just going to draw you attention to the last part, which is the Senegalia Kafra, of what it's usually known to be Acacia Kafra, that it resulted in nearly 100% mortality after just 2 hours of application. I'm just giving you a snapshot of some of the results because of time. This then starts to tell you that as much as bush encroachment is a problem, what if we change our mind, hence paradigm shift? Let's look at it from a perspective of it, benefiting us. So I also just want to touch on the work that we did on tree thinning. Hence I was asking a few questions when the student was presenting yesterday, because this aligns with what we do. And a PhD student from UKZN as well, completed his work, and 2 papers have been published. One is still in review in the what you call plants, MDPI, and the other has just been accepted yesterday, actually, from the African Journal of Range and Forest Sciences. So there's work that has been published in some good journals. So here we said, let's explore further. To what extent does tree thinning has an effect, if at all, on grass production once it has been thinned or cut at different levels? But then we also know that there are different systems. You will find sites that have got a monoculture, meaning that you have got one extensive tree species. In some areas you will have multi-tree species, and they function differently. So we chose those 2 sites, and the thinning was done. It was the control where nothing was removed. We removed at 10, 20, 50, 75, and 100%, which was quite a lot of work. And if one were to ask in terms of practicality, yes, it's quite a lot of work, but at least from research side of things, we start to demonstrate to what extent does tree thinning have an effect on some of the parameters. And I'll draw your attention, and we did it with over 2 seasons. And the most results that you could get is, if this work could be done over time. But I just want to draw your attention. So the first graphs are talking to the tree removal versus grass biomass in site 1. And you remember site 1 was a monoculture, and the site 2 was a multi-tree species. And you realize that there was very little difference with regards to grass production, particularly when you cut or thin at 75 and 100%, which led to a lower grass production versus where you have not thinned at all. But the opposite was true with the second season. There was no difference at all. But in site 2, where you had quite a number of various tree species, there was some difference, especially where you cut at 100%. But this is not ideal, because trees are good. We know that some of them are leguminous and they fix nitrogen. So you do not want to get rid of trees. All that you need is at what level does thinning give you optimal production. And you can see that there was quite a significant difference, particularly from 50, 75, and 100% with regards to grass production versus 0 and 10%, particularly in the second season. I think time is not on my side, but the take-home message here is that the results from site 1 after the first growing season suggest that high intensity of tree removal may further diminish, grass layer, which is what I've demonstrated. But substantial effects of tree removal on herbaceous production, was observed, particularly after 2 years, which then start to explain the results. This is what the literature says, and this corroborate with the results that we got in the second season, as I also said. But one caution is that is that high intensity tree removal of 75 and 100%, it's not ideal, as this may result in large gaps between remaining trees and or lack of woody vegetation, which may favor an increase in soil erosion. So you don't want to solve a problem and create the other, but you want that balance. So there has been extensive work that we have done, and we have decided that let's have a colloquium, a seminar, where we can bring scientists from different institutions across the country. We bring agricultural officers or extension officers, scientific managers, and the University of Limpopo will also be invited to look at how far have we gone with this work, what gaps are there, and what story does it tell when you now holistically look at the work. But most importantly, we're not doing science for ourselves. We also need to communicate this science to end users and also find ways in which that they can adopt the science that we have. And therefore, there will be farmers and the industry, being the seed industry or the red meat industry, we're also going to call upon. But equally important is the policy side of things. Policy must be informed by science. And so we will have everyone in the room where we're telling a story of how far we've come in terms of bush encroachment. And we also acknowledge that we have not covered the ground. So there will be other people, particularly in remote sensing and GIS, you see that I have not covered that space at all. So such people will be brought in so that we can paint a picture of what is really happening in South Africa with regards to bush encroachment. But most importantly, countries like Botswana, where I was 2 weeks back, are heavily suffering from bush encroachment. Namibia is one of the classic examples, and I've had meetings with the University of Namibia, and as well as Lesotho. I had meetings in Kenya last year, and African problems are the same. But the issue we have is that we're all doing our little work. And so I'm looking at a situation where I'm bringing everyone in one room, and we say, this is how far we have gone, these are the gaps, and where to from here, which will also start to look at review papers or even books, book chapters that focuses on bush encroachment in Southern Africa. So I'm calling upon some of you who may be interested. And we've worked with quite a number of institutions, almost all universities in the country. Sometimes I don't know how many students I have, which is not a good thing, because students really fight with me when I take 2-3 weeks without giving them comments. But at least we're working with quite a number of people. This is my contact numbers, including myself. Everyone really is welcome to have them, and we engage. On a lighter note, I'm married with 4 kids. So there's a lot of baggage. So the calls can only be about work. Thank you very much.
Let's thank Dr. Doe. Doc, thank you very much. I can see even you have expanded on collaboration. We are looking forward. Is there any question for Dr. Jelena?
Okay, thank you very much, Doctor. You touched on the issue of, when you talked about the issue of management of bush encroachment, you touched on the issue of the need to look at the situation holistically, so as to see the value of bush encroachments based on the benefits regarding the feed and so forth. So can you please clarify on the thresholds for bush encroachment, and also try to marry that with some of the implications, whether it's exceeded or not, on the soil ecosystem functions. And then the second question is on the issue of policy that you touched. So bush encroachment management, it's a non-marketed ecosystem service. So can you please expand on some of the ecosystem payment that can be employed? Of course, excluding the one that we had yesterday. I think it was ecotourism from Dlamini students. Thank you very much.
Sure. I forgot to.
Is there another question so that we take all the questions and then Doc can explain? Thank you.
Okay, fine. Thank you very much, Doc. That was a beautiful presentation and very informative. You touched on the issue of fire increasing KNP on the surface soil. It just brought about in me the issue of, in the sugarcane industry, what we do before you harvest sugarcane, we burn the whole field. Okay, that makes it easier for harvesting and the concentration of the sugar. So I just wanted to find out, to say, in environments like that one, would it have the very same effect that you are experiencing in those bushes? Thank you.
Any question?
I'm expecting a question about Dlamini.
Okay, let's just wait. Baba Dlamini, they expect a question from you also. Okay, just wait.
Oh, okay.
Just take them once. It will save time.
Right. Thank you, Doctor. Mine is a bit outside from what you're talking about. You talked about two management strategies, mainly bush clearing or land clearing and also fire. Are there any other management strategies that are being used to maybe to control these different types of species? And also, to follow up on that one, these types of plants, are they invasive, introduced kind of invasive plants, or are they indigenous? When I'm talking about management strategies, I know that with other plants, like with Apuntia, they also use biological control, where they apply like certain insects that will feed on the plant or certain pathogens that will kill the plants. Is this an alternative also somehow with these particular plants?
Okay. Okay.
Thanks, Chair. Doc, thank you so much for a very, very nice presentation. I'm actually very excited to have seen the kind of work that you're doing. The first question I have for you is that what is your view to the school of thought that sees bush encroachment as a form of land degradation, when, as you have shown in your presentation, there are some positive feedbacks that are associated with the increase in the density of trees in savanna ecosystems? And what's your take on small-scale farmers also looking at this bushed and encroached grasslands as a valuable natural resource, especially if they want to convert to agricultural farming? Because we have seen that they are also rich in nutrients. And we've seen quite a number of companies that are actually converting anti-stepped grasslands, and the kind of production, agricultural production they actually get from those newly converted anti-stepped savanna grasslands, especially in the first 3 to 5 years, it's immense, because they are really fertile for the most part. Thank you very much, Chair.
Okay, thanks. The platform is yours, Doc.
I think I created problems for myself by inviting too many questions. But thanks, colleagues. Let me start with you, Prof. Dlamini, with my view on the school of thought about the fact that bush encroachment is another form of land degradation. It is another form of land degradation if you look at the negative impacts that it has on various ecosystem functions, and one of them being grazing. I mean, for a livestock farmer, cattle in particular, that has a negative effect if it is beyond a threshold, and a colleague asked about the threshold. If I'm not mistaken, there's a paper, I forgot by who, that talks about a threshold being, I think, 4,500 plants per hectare, that anything at that level and beyond, then it is highly encroached. So from that perspective, it is another form of degradation. However, we're trying to look at it from a different angle to say, that as it may, this problem seems like it's here to stay. What are other ways and means that we can look at it as opposed to look at it as a problem? Because it is a problem. But sometimes when you solely just look at a problem just as a problem and without looking at other avenues, you sort of, at a practical level, limit yourself. It is for that reason that we are exploring other ways, which are also another form of managing it. And Prof. Kani mentioned other form of what are other management strategies. One of them is ecological strategy. One we're looking at, I mean, it took years for certain, for rangelands to be where they are. And definitely it's going to take years to reverse it. It's not something that can be done, overnight. And that has got financial implications. So we're looking at a basket full of options that may suit a particular farmer. One that I've spoken about, I think immensely, it's the ecological, management of bush encroachment. And I'm going to move in between the various questions just to make sure that I also create a linkage. Can farmers use some of these encroached sites as viable, agricultural resources? Definitely yes, but I will look at it from an animal production side of things, even conservation, because we know that bush encroachment has a negative impact on biodiversity, being fauna in the plants. So if you look from the animal side of things, let's say you go with tree thinning at an optimal level that gives you optimal grass production. That will mean that that conversion helps you have more grass biomass and of good quality. So that starts to address some of the ecosystem services that rangelands provide. But in terms of clearing everything, for instance, for planting, I wouldn't say that, because as you may have seen through the work that we have done, that it plays an important role in the agricultural space. Obviously, it will depend from which angle you're looking at. I mean, if you look at ecotourism, it's definitely a no, because visitors in a game park will want to see animals, and therefore, trees obviously is a problem. But again, as I've said, in terms of its negative effect on grazing capacity, it's also a problem. So if I'm not fully answering your questions, please say so. Alternatively, we'll expand during tea breaks. Prof. Kani, you mentioned other form of management strategies. I touched on the ecological component, but there are various ways, chemical being one of them. Biological control, it is one of them, but we have not really explored it. The one project that I know the ARC Sister Institute, the Plant Protection, has explored is on the management of pompong, this purple weed, which is not a bush, but it's a weed. So there are those options, and all that we're doing, and in fact, I take it also as a comment to say, you also need to explore other ways and means of managing it. But these are baby steps, and at least what we have done so far, I think we're heading somewhere, and there are some promising results. Or whether if these are invasive or indigenous, all these are indigenous, plants, because bush encroachment could be both either invasive or indigenous. I mean, there are some areas that are encroached, for instance, by black wattle and many other form of tree species. But our focus in the main was on the indigenous tree species. There was a question whether if fire and was it from it was from you, Dr. Malinga, or about a sugarcane experience where fire, it's applied before harvesting. Fire is a very important tool in managing, bush encroachment in many ways. One being that where you have got an area that is encroached, but you can still have the biomass. Where you apply fire, to some degree you kill a certain number of trees, and that starts to open up over time and allows room, for grass, to regrowth. And there is actually another project that we have done, I just didn't talk about, where we looked at fire grazing interaction, and we applied different treatments, fire being one of them. Others, we use fire and browsing some. So where we applied fire and browsing, you can, even with visual, you can see that it starts to open. But what is important, it's not just fire as a treatment, but it's the follow-up treatments that you do to maintain, because otherwise fire can make, even worse. I mean, you look at some areas that are regularly, banned accidentally. Bush encroachment, in particular Dicrostachys or sickle bush, it's thriving in that area because it's now resistant to fire because it's done unplanned. So it can work if it is planned, but it may actually lead to make situations worse. In terms of the ecosystem services, you mentioned that I looked at a holistic approach. What is the threshold? I touched on it in terms of number of trees per hectare. That then start to show you that once you cross that mark, then you're heading for disaster. And we know, and the line of questioning tells me that you're in that field or you're interested, that it impacts negatively on a number of ecosystem services, and I touched on grazing as one of them. Biodiversity, it's the other, and the issue of ecosystem payment. It's one thing that we have not explored. We have not explored it at all. But if you look at other, literature, and maybe which is the way to go, is should we also not look at, incentivizing farmers or land users who are heading towards the right direction? For instance, they are stocking at an appropriate rate. They're using fire, planned fire, such that they are compensated. And there's also an element of carbon sequestration that has to do with the management of rangeland, that land users are compensated for managing the rangeland well, which leads to a number of improving quite a number of ecosystem services. So there's quite a broad range of issues that I have not touched on, which are very critical, and that starts to say to us that we need quite a number of collaborations of people in various areas, such that we can even explore to what extent does bush encroachment cost a farmer, and that start to demonstrate that if you manage your veld or your rangeland in this way, these are the costs versus if you do this, and that way could be another form of encouraging farmers or land users to meaningfully use the rangeland. I can talk the whole day.
let's do this way. There's a lot of information. Even as I was going to come to water resource, black rain in Natal of the sugarcane is also a problem. But all in all, we just give thanks to Doc. Thank you very much for your time. Let's just take him down with the hands of applause. Then we are going to start the business of the day. As I've requested before, all those who are presenting, they must come in front so that we try to save time. The rules, you know the rules, it's 10 minutes presentation, thereafter is 5 minutes questioning. So we need to move as fast as possible. I'm just going to call the presenters to come. The first presenter and the second, followed by the second one, Juliet Manamela, come and take the stage. Then Juliet Shaibi, just prepare yourself after she finish. After she finish, and just join.
Can you do a pointer? Oh, I can use the cursor.
Just use the cursor.
Okay. Thank you.
Is it too close?
No, I thought it was too. I think it's fine.
Just wait.
Okay.
Just wait for a moment. Take your breath. Yeah. Just take your
time.
Okay. I don't think the house is in order now. Juliet, you can start.
Good morning, ladies and gentlemen. As mentioned, I'm Manamela Juliet. I'll be presenting a study on the post-harvest application of plant-based edible wax to delay sprouting development in potato tubers under the supervision of Professor Mafio and Dr. Tsateche. During post-harvest, sprouting is one of the defects that results in quality loss in potatoes. It is a defect that is characterized by the growth of new shoots or stem-like from the bud or eyes of the potatoes. This defect results in high increase in sugars due to starch degradation, which is undesirable by potato industry as it results in dark color during frying processes. And it also results in mass loss, high mass loss, where it results in tubers appearing to be wrinkled and have shriveled skin. And also it stimulates the production of toxic alkaloids such as your solanine, which is toxic if consumed. All of which negatively affect the quality, the marketability, and also consumer acceptability of potato tubers. Motivation behind this study was that currently methods used to control this, such as chemicals like CIPC, chloroprofen, and ethylene gas are being used, and also cold rooms, and also the likelihood of coating such as essential oils and paraffin wax. Methods such as CIPC and paraffin wax are considered toxic due to the residues they leave on the potato tubers and if consumed. And also they result in toxic residues that affects the environment. And methods such as ethylene gas and cold room result in undesirable sugars accumulating in tubers that are undesired by potato industries. And the likelihood of the use of essential oil. Essential oil are cost ineffective due to the fact that they are highly volatile when applied to potato tubers. Therefore, eco-friendly and cost-effective methods, such as the use of natural waxes, is a need of the hour to overcome post-harvest losses due to the defects of sprouting, such as the use of natural waxes like citrashine natural wax, which is a composite wax that contains the combination of carnauba wax and shellac. It has been used in fruits such as apples and citrus to prolong their shelf life. However, it is unknown if ever it could be used as a sprout suppressant, which is why the study is aiming at reducing post-harvest sprouting incidents on tubers using this wax, citrashine natural wax, where the objective is to determine the effect of plant-based edible wax on sprouting incidents. During the experiments, potatoes of 2 cultivars were bought from Polokwane markets, Sifra and Mondial, where they were transported to the plant lab at the University of Limpopo. They were arranged into 2 groups, the one for control and the one for wax. Firstly, all the tubers were treated in sanitation treatment, then left to air dry, followed by part of the tubers, some of the tubers being dipped in 100% citrashine, and also where the controls were dipped in distilled water. And then all of the tubers were left to dry, all of which were then stored in a dark room at ambient temperature to induce sprouting. The following data was collected: sprouting percentage, decay percentage, mass loss percentage, dry matter percentage, starch content, where all the data was analyzed using GenStat, and mean were separated at 5% level of significance. The result indicated that the wax induced decay. As you can see here, there's a high increase in terms of wax for treated tubers which are treated with wax, and the ones for control show no signs of decay symptom. This is because during the application, the wax acted as a barrier of oxygen, reducing oxygen intake by the tuber, therefore resulting in the accumulation of carbon dioxide within the tuber, therefore inducing anaerobic fermentation, which resulted in favorable environment for the microbes that lead to decay incidents. However, the wax was able to delay sprouting, as you can see by the picture here, where we see sprout development on the control, but there's no sprout development on the treated tubers. This is because even though the wax induced decay due to anaerobic fermentation. This fermentation, where it involves the degradation of starch into products such as your ethanol. It was not significantly enough, the degradation of starch was not significantly in wax treated as compared to the control. This is because the wax resulted in decay. Thus, the decay resulted in the damage of the cell wall of the tuber, where it resulted in high moisture loss, thus inducing higher percentage of mass loss. There was no significant difference between the treatments, the wax and control, in terms of dry matter and starch content. In conclusion, we can say that the plant-based wax citrushine could be used as an eco-friendly, cost-effective sprout suppressant. However, it induced an increase in decay incident. Therefore, we're suggesting future research should develop a protocol to control decay incident without compromising the sprout control efficacy of the treatment. This study will benefit potato industries and consumers, as the treatment use is non-toxic, cost-effective, which will be an alternative to synthetic chemical currently being used by potato industries. I'd like to give acknowledgement to the University of Limpopo, my supervisors. This marked the end of my presentation. Thank you.
Thank you, Ms. Manamela. I'm opening questions to the floor for Juliet. Thank you. At least 3 questions. Questions for Juliet?
Okay, thank you very much for the presentation. So I'm not really in the field, so I'll just ask a general question because no one was raising an hand. On the issue of why the problem that you're trying to solve matters, can you just at least give the figures on the production of potatoes in South Africa, at least compared to its counterparts, other tubers such as sweet potato? So I'm just trying to understand, why does it matter? Why does the problem matter? Is potato production in South Africa that significant? Maybe you can also mention some of the major production areas that we have in South Africa.
Okay, thank you for your presentation. I wanted to ask what could have caused the decay in your tubers after applying the treatment. Could it have been that you applied too much of your wax, or what could have gone wrong? Because we're expecting it to improve or to reduce the tuber. Is it the sprouting of the tubers? Thank you.
Thank you for the nice presentation. Out of interest, can you kindly let me know what causes the sprouting in potatoes, and how long does the sprouting occurs after harvesting? Thank you.
Okay, let's give you a chance to answer the 3 questions.
Thank you for the questions. I'll start with the one for decay. The wax that we used, it resulted in decay due to the fact that it created a barrier. It was supposed to, it has a hydrophobic, let's say, characteristics where it's supposed to control the moisture and all, but it reduced the oxygen intake, thus resulting in the carbon dioxide accumulation within the tuber. That resulted in anaerobic fermentation that induces decay. And the other ones that said, the third one that said on how sprouting occurs and how long it takes after harvest. As we all know, potato is a living, let's say a living thing. It goes through a respiration, right? Where the starch is degraded to maintain the tuber. As time goes on, like maybe after 10 days, the starch within the tuber gets depleted. Thus, as the starch is degraded, it releases some energies that way it provides the niche for the growth of sprouts. Yes. And then the last one would be the ones where you said, how does the study itself help in terms of potato industries and all? Am I correct on that question, if I'm asking? Can you please repeat?
Tubers, we have got, I think, sweet potato as well. So can you just, even if it's not numbers, just a picture of the production of potatoes in South Africa compared to other sources of starch or its counterparts? What is its significance looking at that perspective?
Okay, I think it's because potatoes are produced, as we know, by farmers worldwide, mainly in South Africa, and this is because they form part of our food source, right? And they are normally produced at high numbers, and during storage, these defects occurs during storage, and it reduces the quality of such tubers. That is why our treatment matters in trying to maintain the tuber quality and controlling the sprouts. Thank you.
Thank you, Juliet. I'm just stopping the questions. I do think if there are some questions to be taken, you'll take them. Let's go to the next one because time is not on our side. Thank you very much. I've got so many Juliet today. Juliet Shai? Juliet Shai, can you ascend to the podium for your presentation? Juliet Shai, okay, we can just, maybe she's still coming. Kagiso. Mr. Schali. Thanks.
Okay, thank you very much. The university executive or the presidents of the 4 faculties and the schools as well as the departments. I see Prof. Mababa was here. Students, academic support staff, ladies and gentlemen, good morning. So today I would like to share with you details of my study summarized as spatial variability of soil electrical conductivity at the University of Limpopo experimental farm. This study, I conducted it under the guidance of Dr. M.G. Zereski. So before I lay the context for the study, I'll just try and clarify on some of the concepts that are on the topic. So there are two main important concepts that you can find there. The first one is the spatial variability, which flows from geostatistics, which is a discipline on its own. And also, I'll be looking at the variability of soil salinity. So soil electrical conductivity is basically an expression of soil salinity. So we measure soil salinity based on the conductivity of the water-soil suspension. So what are other scholars saying about these two important concepts that are appearing in the topic? So issues of salinity, like they are, it's a global problem, but we also have it here in South Africa, partly. So I'll just try to clarify that. So about 35% of the decline in arable land productivity in the world can be attributed to issues of salinity. And this is expected to worsen or to exacerbate due to climate change. So moving into to South Africa, water scarcity and salinization are some of the pressing issues in irrigated croplands. So this is secondary salinity, because there's also primary salinity, which I must clarify that is not a major concern in South Africa. So I think we fall somewhere below 20%. There's a soil salinity map that has been done by guys from the University of Free State, so it's below 20%. So today I'll focus mainly on secondary salinity. So secondary salinity or salinity in general is often difficult to manage in croplands, especially in poorly drained soils, because of the costs that are involved. So it's very expensive to correct poorly drained soils, because there are instruments that are used, mostly artificial, but it's expensive to do that. And then in terms of the some of the remedies that have been proposed, so the long-term monitoring of salt levels in the soil it's one of the solutions that have been proposed. But geostatistics have added even more utopia to the situation, because it offers capability to quantify the levels of salt or the soil electrical conductivity in the soil timely. And so spatial variability of soil properties, it's important for site-specific management. So there's this fascination about precision agriculture, and one of the requirements of precision agriculture is that there must be existential variability of soil properties for you to be able to manage your croplands site-specifically. So what is the area of concern on the study area? So the study will be conducted at Seifert Cool Farm, the experimental farm. So the important things to note there is that soils are heterogeneous, so they are not uniform, and that goes to their properties. And the property of interest is the soil electrical conductivity. So at the farm, it's an experimental farm, so there's a volume of experiments that are conducted at the farm by experienced and inexperienced researchers. So this further worsens the issue of variability across space in the farm. And because we're looking at salinity, this can in part pose a risk on land degradation, because when soils are affected by salts, that is one of the causes of soil degradation. And so the data on the spatial variability of soil electrical conductivity can be a proper remedy or a proper management strategy for monitoring of soil levels in the farm, so that we can anticipate accumulation and then also intervene to prevent issues of land degradation. So this picture just shows you how salt affected soils look like. And you can see that where you see the whitish stuff, those are salts, they just accumulated. So you can see that is barren. There's no vegetation because this is a cropland. So those are some of the effects of soil salinity in croplands. So why does the problem matter? Why does this problem matter at the Saiforkul farm? The first thing is that when talking about issues of secondary salinity, climate is one of the drivers in part. So the farm is located in a semi-arid region. And because we don't receive as much rainfall, borehole water is used for irrigation. And some of the direct effects of salinity in croplands is the issue of salt stress that affects the crop water intake, as well as the stress that it imposes on soil microorganisms. And these two things may lead to either loss of crop yields or like the loss or reduction of the soil quality, which is the worst thing that can happen. So the goal of the study was to establish the soil degradation status as an attribute of soil salinity at the University of Limpopo experimental farm. And the guiding objectives to reach this goal was firstly to quantify the spatial variability of soil electrical conductivity using a semi-variogram model. And then secondly, to create a soil electrical conductivity condition map of the farm using ordinary Kriging interpolation. And the complementary hypothesis flowing from the objectives is soil electrical conductivity is spatially dependent at Saiforkul farm. And secondly, soil EC varies in space at Saiforkul farm. So what is the methodology that was followed to address those two main objectives? So firstly, I will describe the study area. So the study area is at Saiforkul farm. The coordinates are given there. And in the next slide, you see maps showing the study area. And this is located in the Mankwin region, Capricorn district, of course, in the province and in the county also. So the area, as I've already touched on the issue of the climate, is located in a region that receives hot summers, and the rainfall ranges from 350 to 500 millimeters per annum. The research block is used for mainly agronomic and plant nutrition experiments. So these are just the maps that shows the study area. So what type of analysis were done to support the two objectives? So firstly, grid sampling was employed. So at 50-meter intervals on the Saiforkul research blocks, soil samples were extracted at 15-centimeter depth using a soil auger, and a total of 55 samples were extracted. And then for analysis, soil electrical conductivity was measured on a 1 to 5 soil suspension. And for statistical analysis, the data was first tested for normality so that we can be able to subject it to the geostatistical analysis using RStudio. And then on the geostatistical analysis, the soil samples coordinates, because the soil samples were georeferenced, were first converted into UTM coordinate system, which is compatible with the model that I'm going to employ for the study. And then the interpolation of soil points was done using Kriging, ordinary Kriging model to create a semi-variogram to quantify the variability of soil EC in space. And then this was done on ArcMap 10.2 software. And then the following equation was used to quantify the spatial variability of soil electrical conductivity, or to check whether or not soil electrical conductivity was has got spatial correlation at the farm. So this is just data showing the exploratory statistical analysis to test for normality of the model that will be employed. And then based on the data from geostatistical analysis using ordinary kriging, this is the semi-variogram model that was produced. And then on the x-axis is the range, and on the y-axis is the semi-variance. So the semi-variance is the variability of the property, which is soil EC, as a function of distance. And then we've got the distance on the x-axis, but the important concept to point out is the nugget. So the nugget is important to check whether or not the soil property was spatially dependent. And it shows, if it's 0, it means there was spatial correlation because of the formula that I showed you. So this already you can see that there was spatial correlation of the soil EC across space in the farm. And then the range also shows the distance up to which there is spatial correlation, looking at the soil property of interest. So this is just text on what I already described. And then the ensuing soil condition, soil EC condition map is presented in the following slide. So this is just a map produced from interpolation. And it's a raster map, and those map patches shows the different levels of soil EC. But the important thing to note is that the maximum electrical conductivity that was measured was 236, and it's indicated by the red patches, which are marginal. They are mostly around the area where there's orchards. So if you've been to Seferkul, there's an orchard or area where orchard is planted, and that is because that area is not plowed, because drainage is also one of the things that affects issues of salinity. Let me just wrap up. So the threshold for salinity is 2,000 microsiemens per centimeter, and the pH must be greater than 8.5. And then at the farm, the pH ranges from 6.5 to 8.5. This was reference work. And then because the maximum value was below 2,000, it means the soils at the farm are not saline. And then these are some of the management practices that can be employed from the results. So in conclusion, the semi-variogram model showed us that there was partial dependence of soil electrical conductivity across the farm. And then the resulting soil map can be used to delineate management zones so that the salinity levels at the farm can be managed site-specifically. And so soil salinity monitoring is important for us to be able to mitigate any future accumulation of soils in the farm so that we can mitigate land degradation. Thank you very much.
Thank you, guys. Although you have taken all my time, I can just allow one question. I just allow one question. I know because we don't have time. Guys who have taken all my time.
Morning, Emmanuel. When you were presenting, you used ordinary kriging to interpolate. So there are various interpolation techniques. So I want to know what guided you into choosing ordinary kriging. And then another question is, you said that semi-variogram showed you that there is spatial dependence, but there are categories into the spatial dependence. It's strong, and then it's moderate, and it's also weakly. So I want to know which one are you categorizing your EC?
Just wait. They ask you another one.
Okay, very quick one, if you may give a brief answer as well. what is the historical management of the study site that may have contributed to the saline soil? You mentioned the borehole. Did you also measure the water? And you said that this is an agronomic and plant nutrition. I mean, there may have been fertilizers that were used. Did you, hence I'm asking the historical management. Just a very brief.
Okay, thank you very much. So I'll take the first one from Mr. Swafo. So of course there are many different interpolation techniques that I use, but I chose ordinary kriging firstly because I was looking at one soil property, because there's the other one, regression kriging, when you want to check the effects of other factors on the soil property that we're focusing on. So that is the first one, and also because of the relative uniform in terms of the slope at the farm. So that is also the second reason. And also because of the sampling strategy that I employed, which was grid sampling. So in literature, ordinary kriging is more compatible when you use grid sampling. So those are some of the important factors that I took into consideration when selecting the appropriate kriging interpolation technique that I used. And then the second one on the different thresholds for quantifying spatial dependence. So the first one is if the percentage nugget is less than 25%, then there's strong spatial dependence. On the percentage nugget that I calculated from the semi-variegogram model was 0. So that showed a strong spatial dependence of soil EC across space. And the question that you asked on the history of the farm, so the objective that drove the study was to establish the degradation status, to collect the data to discern whether or not there's salinity issues at the farm. Even if there's no salinity issue at the farm, the data can be used for monitoring, because of geostatistical tools, you can also quantify temporal variability. So the data can be used as primary data to, over time, quantify the temporal variability of salinity. And now, because of different modeling models that are there, you can also use the data to predict or for modeling whether or not certain management practices will result to salinity issues.
Thank you, guys. So you can step down and stop up. So without waste of time, we are going to go to the third presenter, Disley. Are you Disley? Where is he? Mr. Taba, come on stage. Okay, Mr. Taba is not around. We are having one student who will be actually presenting visually. It's Noli Tabo. Wow. She'll be doing her presentation visually. Thank you. Okay, let's just let her present visually. They will operate it. Thanks.
Okay. I'm not sure if I'm audible. Okay, thank you. Okay, so morning. Good morning to everyone present. I'm Nolutabo Wawa. I'll be presenting a study titled Factors Influencing Climate Adaptation, Evidence from Crop Smallholder Farmers in Basha Local Municipality. The study was supervised by Dr. Nesengani and reviewed by Prof. Zwane. Now, climate change, as we all know, poses severe threats to agricultural sector, possible because agriculture is inherently sensitive to climatic conditions of temperature and rainfall. Now, smallholder farmers, they are the most vulnerable as they mostly practice rain-fed agriculture. Now, adaptation to climate change has been said to be the solution or the only hope for farmers to potentially increase their agricultural production. Now, Balcha has been said to be currently faced with drought and or has already been declared a state of disaster. Now, smallholder farmers have a low adaptive capacity, according to Tarovinga 2019. Now, there was inadequate information available, or there is, about the factors that influence crop smallholder farmers' adoption to climate change. Now, most literature done in the Eastern Cape looks at district level and provincial level, and mostly some of them they look at livestock and combine sometimes livestock and crop. So this study was trying to look at local municipalities, and sometimes local municipalities find that they are facing different climate variability than maybe the rest of the district. So this study aimed to bridge the gap in the literature by evaluating current adaptation strategies employed by smallholder farmers to mitigate the effects of climate change on crop production. Now, a study on climate change adaptation is significant as climate change variability are a major challenge in agricultural production, as farming is sensitive to climate change, as I said. Now, farmers-driven adaptation strategies show more potential, as recorded by Tarovinga 2019, than external mitigation in Africa and South Africa. Therefore, the outcomes of the study may yield more information on adaptation employed by smallholder farmers to cope with climate change. Furthermore, understanding adaptation strategies through this study will assist to identify effective drought coping strategy. This also entailed more information adaptation choices. Now the study, as I said, it aims to bridge the gap in literature by evaluating current adaptation strategies employed by smallholder farmers to mitigate effects of climate variability on crop production. Sorry. Now the objectives of the study, there are two. One was to assess current adaptation strategies adopted by crop producing smallholder farmers to mitigate climate variability in the study area. Two, it was to investigate the factors influencing the choice of adopted adaptation strategies to climate variability by smallholder farmers in Basha local municipality. Methods. Now, the study was conducted in Basha local municipality in the Eastern Cape. The study adopted a quantitative research design. The study focused on primary data and adopted multi-stage research design where
sampling design where Mbashe local municipality was purposely selected because 17% of the households in Amatole district municipality are strictly involved in agriculture, and approximately 40% of that 17% are household farmers or smallholder farmers from Mbashe local municipality. Now, Ngadu, Boloto, and Sinumeni were purposely selected due to the high number of farmers involved in crop production, and then a simple random sampling was done to select the farmers to participate in the study. The sample size for the study was 358, and due to availability and willingness to participate of farmers, and the study ended up interviewing 207 farmers. The data collected was coded and recorded on a Microsoft Excel and analyzed using descriptive statistics to answer objective 1 and multivariate probability model to answer objective 2. The study followed a KM 2019 multivariate probability model, which was characterized by a set of M binary dependent variables, where, where J was 1 and 2, M denotes the type of adaptation strategies available, where X is the vector of explanatory variables, and J denotes the vector of the parameter O to be estimated, and U are random errors terms distributed as a multivariate normal distribution with a zero means of unitary variance. It is assumed that that a rational H must have a latent variable Y, which captures the unobserved preference or demand associated with the J choice of adaptation strategy. The dependent variables of the study were the adaptation strategies like conservational agriculture, drought resistance, crop irrigation, water harvest, shift in planting season, use of fertilizer, crop diversification, use of indigenous vegetables or fruit, and also not adapting, seeking of farming, employment, mixed farming, and buying of insurance. The independent variables of the study were the socio-economic factors such as farming experience, age, gender, household size, marital status, level of education, which also included access to credit, access to climate information, access to extension services, and land size the farmer owns. Now the results of the study. Now, although there were 80% of the farmers in Bashe local municipality have adopted to climate change. Now, drought resistant was the most used or adopted climate adaptation strategy, with 64% of farmers, and fertilizer use was 34%, mixed farming 21%, crop diversification 16% of the farmers adopted it, irrigation which was 13%, use of indigenous vegetables were 12%, use of indigenous fruits was 10% of the farmers, conservational agriculture was 4%, and seeking. Okay, so, sorry. And factors influencing adaptation by smallholder farmers. Now, conservational agriculture showed that education, looking at conservational agriculture, education level had a significant and a positive relationship with the conservational agriculture as an adaptive strategy at 1%. Now, this means the chances of a farmer adopting conservational agriculture rises with an increase in farmers' education level. And also the household income had a significant and a positive relationship with conventional agriculture at 1%, indicating that an increase in household income increases farmers' chances of adopting conservational agriculture as the adaptation strategy. Also, access to extension services also proved to be significant and had a positive relationship to conservational agriculture by 5%. Now, choosing to adopt a drought-resistant crop, access to agricultural extension also had a positive, it was significant, and it showed and had a positive relationship at 1% to choosing drought resistance as an adaptive strategy. Also, household income had a significance and had a negative relationship at 1%. This means that the likelihood of a farmer adopting a drought resistant can result in a decrease in household income is lower than it was on original instrument variable. Now, moving to choice of irrigation, choosing irrigation as an adaptive strategy. Now, age of a farm had a positive, it was significant and had a positive relationship by 10%. Now, the level of education of a farmer had a significant and a positive relationship at 1%. Then also access to extension services had a significant and a positive relationship at 1%.
Sorry,
Now,
so,
looking
sorry,
at
so,
shifting
sorry, Miss Wawa. You are already on my time. You have already passed 10 minutes. Now you are going to 12. Finish up.
Okay, shifting planting season as an adaptive strategy. Now, when it comes to shifting planting, had a significant influence and had a positive relationship by 5%. Mixed farming as an adaptive strategy. The farmer's level of income had a statistically significant and had a positive relationship at 5%. Now, from this, it could be concluded that smallholder farmers in Basholopo municipality have adopted several adaptation strategies to climate variability, and also that the adoption of the list of adaptation is influenced by farmers' socio-economic characteristics. However, the majority of smallholder farmers adopted more than one adaptation strategy. It is noted that farmers that were mostly affected by drought are those that are farming in the northern part of the local municipality. Now, this study recommended that educating farmers about climate change and providing them with information and anticipated climate change and early natural disaster warning to farmers through radio programs, television, social media, on information days, through extension agents, highlighting changes in temperature and rainfall patterns, drought, floods in smallholder farmers, understandable method of communication. It also recommended that extension services should be accessible to all kinds of farmers, even those that are farming individually, as it is most crucial to adaptation to climate variability. Also, the study also recommended that the government should consider the recruitment of more agricultural extension agents, as most farmers reported they had limited access to advisory service. Also, extension personnel experiences quite high ratio on farmers, which lead to ineffectiveness. These are some of the studies that were cited. Thank you.
Thank you very much. You don't go anywhere. Just let's clap the hands for her. I'm going to open this wide now because you have taken the time. I'll need 2 questions from the floor, but we are going to change it because it's visual, and I wonder if your questions will be needed to be rephrased. I was just asking here, is it possible, professors, whoever wants to ask the question, come on stage? Any question for Mrs. Wawa? I'm allowing 2 questions. Any question for Ms. Wawa? Again, any question for Ms. Wawa? You are afraid of coming to the stage? Then I'll write it down. Any question for Ms. Wawa? I'll make it easy, I'll write it down. Any questions for her? Okay. Okay. They don't have questions for you. Thank you.
Thank you, sir.
Okay, again, I'm going to request if there are 2 students who were not in, are they back? Juliet and Mr. Taba. Okay, thank you very much. I do think it's the end of my shift. Thank you very much for your time.
Good day, ladies and gentlemen. I am Keto. I will be chairing the current session. And then to the presenter, you only have 10 minutes for presentation and also 5 minutes for questions and answers. Let me call upon Mr. Kwena Mkwena to come and present.
Thanks, Chairperson. Good day, everyone. As they've mentioned, I'm Kwena Mukwena. I'm under the supervision of Professor Chasi and Professor Vijay Mbazim. I'll be taking you through some few slides on the breed influence on morphologic traits of chickens. This is the outline of the presentation. A highline silver brown, it's a commercial chicken, and then it's termed a prolific egg layer because it's one of the best egg laying chicken on the market. And then the Potchefstroom cock is just an indigenous chicken that is known to be kept for dual purpose, which is egg as well as meat. The morphometric traits are used mainly for the estimation of body weight in animal breeding to aid the farmers as they are managing their farms as well as selecting for breeding. It is difficult, however, to predict the live body weight in poultry where scales are not readily available as in most African rural farming communities. So the morphologic traits in chicken breed can assist for the comparison and characterization of breed as well as to estimate the body weight, like I mentioned on the previous slide, as well as the management. It can either be administering your medication and as well as marketing your animal. We know that we are farming so that we can sell and get profit at the end of the day. However, based to our knowledge, there is limited literature on the estimation of body weight and comparison of morphometric traits in Potchefstroom cock and Highline silver brown chicken layer breeds. There are some few papers that are there on the net. There is one that was published in 2018 on the morphometric traits, which were predicting the French broiler and the guinea fowl of the Nigerian chickens, as well as the Nigerian indigenous feather chicken breed. And the morphologic trait in chicken breed can assist for the characterization of breed. There are several studies which compare the morphometric traits of chickens as well. The aim of the study was to identify the performing breed among the Potchefstroom cock and the Highline silver brown breed, and the objective was to compare the morphometric traits of Potchefstroom cock and Highline silver brown layers. The study was done at the university farm, which is located in Mankweng. And then there were a total of 100 chickens, 50-50 from each breed of 22 weeks of age. All chicken were managed as explained by the author mentioned there. The cross-sectional design was used in this study. These are some of the morphometric traits which were measured. We had the body weight, which was measured in kilograms using a weighing scale. And then we have the morphometric traits, which were measured using a measuring tape. SAS was used to analyze the data, where Pearson correlation was used to examine the association between the measured traits, the t-test to check the effect of the chicken breed on body weight and other morphometric traits. And all these analyses were performed at the 5% significant level for statistical significant and highly statistical significant at 1%. This is the model which were used or was used to check the effect. From the results, the box plot is demonstrating that the Potchefstroom cock was heavy than the Highline silver brown. The correlation table below diagonal, let me try something here. Yes, the below diagonal, it's where now we are having the silver brown, and the above diagonal, we are having the Potchefstroom cock. If we can check, the red circle there is showing us that there was a negative relationship or correlation between wing length of silver brown and the body weight, whereas the highest, the significant correlation was found between the chest circumference and the beak length. When we are going to the Potchefstroom cock, the wing length is now positively correlated to the body weight, and the highest correlation was found between the beak length and the chest circumference. The effect was checked, and then the results are showing us that the Potchefstroom cock were heavier than the Highline silver brown. When we are looking at the keel and the shank circumference, the silver brown were performing better or recorded higher values as compared to the Potchefstroom cock. The results of the study, they are implying that by increasing the wing length of a Potchefstroom cock, we can increase or improve the body weight. Whereas in the side of silver brown, by improving the beak length, we can as well improve the chest circumference. The report that was done or was given in 2022, it is showing that the traits that are positively correlated, it means that those traits may be controlled by the same gene. The results, they are also showing that the Potchefstroom cock, it was heavier than the silver brown. And the author that is mentioned there is also supporting that statement. The body weight of Potchefstroom cock might be improved by improving wing length, while the beak length might be improved by increasing the chest circumference. The finding of the study also indicated that the Potchefstroom cock was heavier as compared to the silver brown chicken. It is recommended that more studies be done on the influence of breed on morphometric traits of chicken to improve the selection by farmers, as well as doing other studies, not only on the breed that are mentioned. I would like to acknowledge our very own university and the department for allowing me this chance to come and present. Much appreciation to my supervisors and the co-supervisor. Thank you very much.
Thank you for the wonderful presentation. Any question from the ground? Okay.
Thank you for a nice presentation. I just want to know what informed the choice between the two species. What particular characters of the species were important for you to look at them? Thank you.
Any other question? Okay, let me just add some 2 questions. On your study, did you consider or did you account for genotype by environmental interaction? And also another one, how did you manage to overcome the maternal effects on the selected traits?
Thanks for the question. The choice. To be honest, these were the chickens that we had at the time at the farm. That's why we chose these 2. But because we know that we are trying to improve the indigenous ones. So, and in overview, we did not cross, we did not do anything. We just took the chickens that were there, and then we wanted to know if we are to have these 2 breed, which one can give us the outcome that is good. So the choice, it was based on what we had at the farm. Yes, thank you. And then taking into consideration the G by E, how can I put this one? You're cornering me, Dr. Ket. I can't say we took into consideration the G by E when we were doing the study. And then because we did not look into anything that was like, let me say, the correlation. We only took the association. And then we concluded based on that statement that is saying, if they are correlated, then that means the genes are also playing a role. And then also this animal, they were kept in cages. So the environment, it was the same. I'm not sure if you answered. And then the maternal. In most cases, there are some formulas that we check for the maternal effect and paternal effect, gene additive, and whatnot. And in most cases, normally we check for those when we are crossbreeding the chickens so that we can improve the offsprings. And in this case, we did not go into that path. Thanks.
Thank you. Any other question? No, and then we still have enough time. Thank you very much for presenting a very wonderful presentation. And then I would like to call upon the next presenter. Ntabeleng Mafia. Ntabeleng Mafia? Not here. Oh, okay. And then we are going to the next presenter, who is Katlego Mutabo.
Thank you very much, Chair. Greetings to the House at large. My name is Katlego Mutabo, and I'm going to be taking you guys through my study, which is titled Assessing the Factors Limiting Water Accessibility in Some Portions of the Capricorn District Municipality of Limpopo Province, South Africa. This study is under the supervision of Dr. Akanbi, and it is co-supervised by Dr. Letswan. According to the United Nations, in 2019, 2.2 billion people lacked access to potable drinking water. Limited access to safe drinking water is the leading risk factor for waterborne diseases such as diarrhea, cholera, dysentery, hepatitis A, typhoid, and polio, and ultimately causes malnutrition in children. Therefore, it is safe to say water, sanitation, and hygiene are a critical component of every human being's health and well-being. The following key issues have been identified to be impacting Capricorn District Municipality in terms of water accessibility. The Capricorn District Municipality is in a critical state of water deficit. Water quality and quantity problems are now plaguing the municipality, and climate change is expected to further exacerbate these challenges. Amongst the concerns, the operational issues are more predominant and frequent in the district. The aging infrastructure across the district causes regular mainline pipe bursts, frequent pump failures, and to date, unprecedented events of theft, poor operations, maintenance, and ESCOM power cuts have heightened the issue of water availability to the local, urban, semi-urban, and rural surrounding areas. This study is grounded by the Malthus basic theory, which states that the population growth is exponential, while the growth of food or other resources is linear, which eventually reduces living standards to the point of triggering a population die-off, and this event is called the Malthusian catastrophe. So if we were to apply this in the context of Capricorn District and water supply issues, we would note that the population in the Capricorn District has grown over time. However, when it comes to the available water resources, they are failing to meet the heightened demand. Now the question is, why is this study important and how do we come into play? Firstly, people have the right to have access to adequate water and food as per the Constitution of the Republic of South Africa. Therefore, municipalities need to make plans to protect available water resources and ensure provision for water and sanitation service delivery, while taking into account the availability of limited water resources, prevailing water infrastructure, and human capacity requirements to meet the growing demand. This study aims to fill the gap in science by not only focusing on factors limiting water accessibility in rural areas, but to also explore challenges faced in the semi-urban and urban areas. Why is this? This is because understanding the different levels of water accessibility in the different settlement types will highlight the disparity in access to water in the province and may inform future policies, planning, and infrastructural interventions. The main aim of the study is to assess the factors limiting water accessibility in some portions of the Capricorn District Municipality of Limpopo Province in South Africa. The study has three main objectives. Objective 1 is to determine the experiences and perceptions of urban, semi-urban, and rural dwellers with regards to water accessibility in Polokwane Municipality. Objective 2 is to identify the factors limiting Polokwane Municipality's ability to ensure water accessibility to its urban, semi-urban, and rural dwellers. And lastly, the last objective is to determine the community's current adaptive responses to the water challenges in the urban, semi-urban, and rural settlements. The hypothesis of the study is that rural dwellers are more vulnerable to factors limiting water accessibility in Polokwane local municipality than the urban and semi-urban dwellers. Ladies and gentlemen, this is the study area. So the municipality of choice that we considered for this study is Polokwane local municipality. From there, we managed to get 3 settlement types, which is Flora Park, and it's urban, which has 2,891 households. We focus also on Mankweng Township Unit A, which is our semi-urban, which has 2,577 households. And lastly, the considered settlement was Ratoka Village, which has 3,068 households. And this data was sourced from Statistics South Africa. So when it comes to the methodology and analytical procedures for our study, we adopted a mixed method approach with an explanatory sequential design. So we had 2 types of data collection points. We had primary data collection, which is comprised of 2 phases. Phase 1 is quantitative data collection and its analysis, where we collected data through questionnaire administration. Phase 2, we had qualitative data collection and also its analysis, which was achieved through focus group discussions and key informant interview. So from the data and analysis coming from the 2 phases, we integrated the data as to get to understand the relationship or the experiences from the phase 1 data and phase 2. From there, we went to the secondary data, which is literature review, municipal reports, and historical climate data. This data was used to justify our primary data and to see if the tested relationships are valid. The following formula was used to calculate our sample size by Isaac and Michael. In terms of ethical clearance, it was sought with the University of Limpopo Ethics Committee, and it was granted in March 2023. So when it comes to the results of the study, objective 1 of the study was to determine the experiences and perceptions of urban, semi-urban, and rural dwellers with regards to water accessibility in Polokwane Municipality. And in answering this objective, we use household surveys and focus group discussions. And in terms of water accessibility challenges, it was seen that Ratoka Village and Mankweng Unit A experience more water accessibility challenges as compared to Flora Park. And on your, I think it will be your left, because it's my right, we also looked into the types of water accessibility challenges where residents raised that they are experiencing insufficient water, which this was mostly experienced in Mankweng Unit A. There is an issue of broken taps and hand pipes. There is also a serious challenge which was raised in Flora Park, where participants raised the issue of their water not being safe for consumption. This was noted by water having sort of like sand sediments found when they were trying to use the water for their daily functioning. So here is the study findings from focus group discussions, where it was noted that poor or inefficient channels of communication have been highlighted as the main contributing factor to water accessibility challenges. This is because respondents raised the issue of not being able to know when water will be available or when water will be cut. Just to open quote from one participant whom raised that the municipality is the main cause of water challenges. Right now, I don't have water. There is no communication from the municipality. How do I get kids ready for school in the morning? We are struggling. These findings really reveal that the impact of poor communication between the local municipality, which is Polokwane, and its residents is really severe because people do not know how to plan efficiently for their daily functioning. So these are some of the results where we're looking at water accessibility in terms of distance traveled to collect water. So the study findings indicate that across the different study types, most households traveled within 1 to 2 kilometer range, with Flora Park having 94% of households in this range. However, in Ratoka, 2% of the household traveled more than 4 kilometers and above to access water. In terms of coping mechanisms for water scarcity, the findings show that most households across the study area use reserved water as their coping mechanism to water accessibility, followed by households who purchase water. And we can also note that Ratoka also falls under this category of people purchasing water. And with our study findings, we realized that Ratoka has a very low socioeconomic status. So we can imagine people with a very low economic status having to buy water for their daily functioning. So this becomes a financial strain on the community. So these are the study findings from the key informant interviews. So the key informant interviews were conducted with water technicians from the Polokwane local municipality, and the following issues were identified. One, there is reduced water levels in the dam, which is Ebenezer Dam, due to climate change. Secondly, they highlighted that there is deteriorating water infrastructure and poor maintenance, which have been noted as a major issue. They are also experiencing financial limitations, which has resulted in insufficient investment in water infrastructure. Lastly, they noted that the ongoing population growth in Polokwane has led to the increasing pressure on available resources. So as part of data collection, we also did field observation, and the following key issues were observed. So we observed water wastage practices, especially in Flora Park, where residents were watering plants in the afternoon when the sun is at its highest, resulting in evapotranspiration. We also identified few infrastructural challenges where on my left, I think it will be your right, you see that there is no tap to draw out water. So they've connected a pipe to the main pipeline underneath just to get water. There's a lot of broken taps within the community of Ratoka. So these are some of the identified water conservation practices. There in the middle, this one, it was really problematic to witness, to be honest, because you can imagine that the way people do not have water, they use each and every resource at hand to can save as much water as they can, because the municipality in terms of water supply, they are really failing. In conclusion, ladies and gentlemen, the findings from the study confirm that residents from the different settlement typologies are facing varying levels of water accessibility challenges. Some of the factors contributing to the water accessibility constraints being experienced in the different settlement typologies assessed includes natural factors, operational concerns, aging infrastructure, poor communication, and financial limitations. The communities are therefore adopting a range of coping measures to reduce the impact of their water accessibility challenges. In terms of how is this study going to contribute towards the body of science, rather, it's going to provide empirical evidences which could inform the appropriate intervention to address the current challenges and ensure that adequate supplies of clean water are made available to the communities to protect their health, livelihoods, and improve productivity. Also, the study is going to contribute by increasing awareness on current water constraints experienced in the study areas and provide an insight into the current and future areas on infrastructural and service improvement and augmentation. Lastly, the study is going to contribute towards the development of appropriate water policy interventions and implementations within the local municipality. My sincere gratitude goes towards Dr. Aganbi and Dr. Letswalo for their profound supervision, and it also goes to the Water Research Commission for making the study possible. These are the references that I use in terms of putting the presentation together. Thank you.
Thanks for the wonderful presentation. I will want to take the questions from the floor. Only 2 minutes for the questions.
Thanks, Katla. Hopefully you will solve many issues of water in the country. When you're talking water accessibility, quality also is not very far. I mean, on the news you hear Hammanskraal and Giyani. If you can just comment on the accessibility and the quality of water that may be available. But on a lighter note, I have not heard the key word which is common in South Africa, corruption. To what extent do you think that contributes to the accessibility? Because I guess it's somewhere in the mix of things.
Any question? Any other question? No question. And then let me ask one other question. Considering there is a lot of illegal connectivity around Madimo, do you also consider that? Yes, there's a lot of illegal connectivity where they connect on the main pipeline to Polokwane. Do you also consider that?
Thank you very much for the questions. In terms of how does corruption contribute to water accessibility, I mean, we are living in times where money embezzlement is something that we are frequently experiencing, especially in Polokwane, I would say. There are a lot of interventions or infrastructure interventions that have been put into place where people are given tenders to come and maybe put together new water reticulation systems. To date, you might find that, okay, they've put new pipes, but in terms of them really putting the pipes together and making sure that there is water accessibility, it's something that has never even happened. In Ratoka, that's where I come from, you might find that people, or I would say the municipality, come and they bring new interventions and new infrastructure changes. However, in terms of really fulfilling or putting together the proper measures and giving people water, it's something that has really never happened. So corruption does really affect water accessibility in Polokwane. So coming to the second question, Ratoka has mentioned, or we have been able to identify few places where illegal connections were made. However, Mamadibo Park was not within our study area. So Mamadibo Park is just a new extension of Ratoka, but when you go to the tribal authority, they don't note it as being part of Ratoka. So we did not consider any key issues emerging from Mamadibo. Thank you.
Thank you very much. You can take it. Okay. Thank you very much for the wonderful presentation. Yes, people need water, right? Water is life. Let me call our next presenter. It's going to be Koketso Machete.
Ladies and gentlemen, distinguished guests and esteemed colleagues, my name is Koketso Machete and good afternoon. It is indeed a privilege and honor for me to stand before you to present my paper titled Vulnerability Assessment of Smallholder Maize Farmers to Climate Change Risks and Willingness to Adopt Climate-Smart Agriculture in Limpopo Province under the supervision of Professor Signolo and Dr. L.S. Gidi. This is the presentation outline and the discussion will be based on the following points. Now, climate change as a global issue, it stands as a threat to economic sectors such as agriculture, forestry, fisheries, and wetlands, threatening the productivity as well as the profitability of these segments. This global issue, it makes farmers, particularly rural farmers, to become vulnerable to extreme temperatures and pest damages, as well as low rainfall distribution. Now, maize, it is considered as the most consumed grain crop in South Africa, as it is considered a major staple feed for livestock and staple food for rural households. Now, the study assumes that smallholder maize farmers face climate change risk without an effective management or coordinated action that is put in place. Agriculture, it is a critical economic sector in the Limpopo province in terms of its contribution towards the number of employment opportunities it creates in the rural areas and poverty alleviation while addressing the sustainable development goals, which is zero hunger and also improve the standards of living. Now, this smallholder agriculture, it plays also a multifaceted role through ensuring food security, supporting livelihood through provision of income, and also fostering sustainable development in the rural areas. Now, there's a need for us researchers to assess the vulnerability of these smallholder maize farmers towards the risk of climate change and also encourage the adoption of climate-smart agriculture. The aim of the study was to analyze the vulnerability of maize farmers towards the risk of climate change and analyze the willingness to adopt these mitigations in Limpopo province. The study consists of 3 objectives, which is to profile the socio-economic characteristics of smallholder maize farmers, to assess smallholder maize farmers' vulnerability to these climate risks, and also to analyze the social factors influencing the willingness to adopt climate-smart agriculture. But however, this presentation will be based on the 2 objectives, which is the first one and the last objective, since they're interrelated. The hypotheses of the study are in line with the study objectives. The study was conducted in 3 different areas, in Ramakanye, in Gabaza, and also in Giyani. A mixed method research design was used to collect data, which it allows the study to integrate or validate the results from both quantitative descriptive statistics as well as qualitative descriptive statistics. The study used a structured questionnaire, focus groups, as well as Likert scale to collect data from the respondents. A multi-stage sampling procedure was used, whereby it consists of 3 stages, Polokwane being the first stage that was purposefully selected in terms of its number of smallholder maize farmers. And the second stage, it consists of 2 districts that were being selected, which is Mopani and Capricorn district. And lastly, the 3 areas were selected mainly because of their different agroecological climate conditions. And lastly, the study, it has a sample size of 213 that was sampled using Yamen's formula, where Ramakanye was sampled at 28, and Gabazi was sampled at 87, and lastly, Giyani was sampled at 98. Descriptive statistics, vulnerability index, double huddle model, through contingent evaluation model, it was used to address the 3 objectives respectively. This is the model specification, where the first equation, it is the linear regression model for the double huddle model to be used. And the second equation, it shows us the adoption decision, whether to adopt or not. And the third and last formulas, it shows the factors affecting the adoption decision that is mainly centered around a truncated regression model called Tobit regression model. The descriptive results for the study are presented on the slides, with the first results being the willingness to adopt of this climate-smart agriculture among the smallholder maize farmers. From the 3 figures, we can see that the willingness to adopt, it was dominating, with Ramakanye having 81% and Gabaz and Giyani constituting a total of 67% and 63% respectively. This is an indication that indeed farmers are willing to adopt these practices, and however, we need to take into consideration the factors that might affect this willingness to adopt the practices. Gender distribution of the smallholder sample farmers, it is shown on the slides. Ramakanye, it had an equal distribution of number of farmers, while Gabaza and Giyani, it had a more proportion of female farmers as compared to male farmers. One of the reasons is that the males in the areas have migrated to the neighboring cities in pursuit for better job opportunities to sustain their livelihoods and to maintain their families. The educational level of the farmers are presented in figure 1.3, 1.4, and 1.5. Where in the 3 figures, we can see that Ramakanye, which is the first figure, it shows that the farmers in the area, they constituted or they had obtained education as their formal education, while in Gabaza and Giyani, there are more illiterate farmers because the area is normally dominated by older people. The descriptive statistics of the continuous variable are demonstrated in the slides, but however, the discussion will be based on age and experience. From the 3 areas, we can see that a minimum age of a farmer it was 21 years old, while the maximum age of a farmer was 94, which is surprisingly that there are still older people out there that are still practicing agriculture. And also take into consideration the experience of the farmers. The minimum experience in agriculture it was 1 year, while the maximum experience it was 75 years. This is an indication that the area is mainly dominated by older people. The empirical results are shown on the table, where the first hurdle, we use a probit regression model to analyze the results. And looking at the log likelihood of which is negative 105.664451, it indicates that the model it is perfectly fitted, because a log likelihood that is relatively high and positive, it shows that the model it is poorly fitted. And also the likelihood ratio chi-square at 13 degrees of freedom, it was constituting to 55.71, and it was highly significant at 1%. From the table, we can see that education, crop diversification, and information about climate-smart agriculture, they were positively influencing the willingness to adopt, and agricultural experience and household size, they negatively influenced the willingness to adopt. The discussion will be based on the second hurdle, shown on the screen as well. We used a Tobit regression model to analyze the second hurdle of our double hurdle model. And also, again, in this hurdle, it shows that the same significant variables, they were also significant at 5% level of significance. Now, we can argue that based on the coefficient that is attached to education, we can argue that an additional year in farmers' education would likely increase the willingness to adopt by 28.16%, as well as crop diversification. If a farmer produces another crop, this will likely improve their willingness to adopt by 38.81%. This is indeed true, because the implication is that when a farmer produces not solely only one crop, they will be able to adopt the practice on one production of another crop, while obtaining or maintaining or observing the changes, and also looking at the cost that is associated with that particular production. In conclusion, the null hypothesis of the study was that smallholder maize farmers' social characteristics do not influence the willingness to adopt, but however, based on the findings, we reject this hypothesis, concluding that there are socioeconomic characteristics of farmers that influence the willingness to adopt, namely, which is education, information about climate-smart agriculture, and crop diversification. They positively influence the willingness to adopt, while agricultural experience and household size, they negatively influence the willingness to adopt. The study recommends that tailored educational programs and extension services should be provided, as these play a vital role in shaping the farmer's knowledge. This includes the introduction of programs like personal development programs that will allow the farmers to receive a certificate of competence, and also encourage them to engage with other farmers so that they will be able to share knowledge and equip each others with more information pertaining to this risk mitigation and adaptation strategies. And also, we need to take into consideration that there are specific needs in terms of different regions, crops, and communities. And also, the Department of Agriculture, together with various stakeholders, they should enhance the knowledge transfer of highly experienced farmers, whereby they encourage highly experienced farmers to motivate and guide young emerging farmers, so that also we could have young farmers that are passionate about farming. Encourage crop diversification to reduce the risk that is associated with monocropping, while ensuring that we maintain sustainable agriculture. I'd like to acknowledge my supervisor, Professor Signolo, my Professor KK IEC Young Researchers Club from Risk and Vulnerability Science Center, as well as NRF and the University of Limpopo for the opportunity they have provided me to present my paper. These are the references used for the presentation, and thank you.
Thank you very much for the presentation. And then any question?
Okay, thank you very much for the nice presentation. However, I know you'll be dealing with respondents, or rather participants. And at some point you find that what they respond, they look at what you have or what you bring forward. So my question is, what was your selection criteria for your participants in terms of authenticity? What was your selection criteria? Thank you.
Yeah, another question?
Thanks very much for the nice presentation. I understand you are into economics, but I want to know, when you asked these participant questions, did you present to them the kind of climate smart agriculture practices that you want them to adopt, or you just asked them general question to say, are you willing to adopt this smart agriculture, or you presented a particular what is it, model or some sort?
Another question?
Thanks, Kokezo. Well done. We know that indigenous knowledge system is very important. Did you have a sense that besides education, there is an element of indigenous knowledge system that also played a role, and if so, to what extent?
One last one.
Thank you so much for such a very great presentation. Out of curiosity, I just want to know, because I heard you said you rejected your null hypothesis. So I want to know, I mean, if you have adopted any statistical, techniques to actually, reject or accept that null hypothesis, and which one it is that you have adopted.
Okay, thank you for the questions. Let me start with the last one. In economics or in econometrics, we normally use a different approach. We reject the null hypothesis based on if we find any significant variables on our models. So since I have adopted the use of a double hurdle model, it consists of 2 stages, which is the first hurdle, where I used probability regression model, and the second hurdle, which used a Toybit regression model. So if both in the models the same variables were significant, then it shows that I have to reject the null hypothesis since I have significant variables. And to answer the first question of the selection criteria, we provided the farmers with various options using contingent evaluation method, where there were a range, a number of practices that the farmers they had to choose from, and the range it was choosing at least 3 practices. For example, if a farmer chose to use specific site nutrient management or conservation agriculture, this includes mulching or crop rotation or even crop diversification, it was showing to us that the farmer is willing to adopt. But however, the decision was based on the first decision, which is, are you willing to adopt? If they were willing to adopt, then they have to choose at least 3. The criteria did not limit them. They can choose even more than 3 practices. Then to answer the second question, yes, I provided farmers with various options to choose from, and I introduced the practices to them and explained what these practices are in their native languages, and only to find out that some of these practices, they were already practicing them as they were known as indigenous knowledge. Coming to the question again, we also need to take into account that highly experienced farmers, they may be illiterate, but they acquire more experience and practical knowledge in terms of farming. So we can also take into account the use of indigenous knowledge that they obtained from their forefathers, so that they can integrate both the indigenous knowledge as well as the formal knowledge, so that we can have an effective production in terms of their outputs. Thank you.
Thank you for a nice response. I will take this time again to call upon our next presenter, Lihlono Loshaleng. Shaleng? Seems like the presenter is not around. And then let's go to the next presenter, which is Abatakali Mafukatla.
Namaste. Good day, ladies and gentlemen. I'm Fukatla Batakari. I will be taking you through my research findings. The title of the research is Effect of Maize Density on the Productivity of Intercropped Cowpea Varieties. Background of the study: cowpea is a food grain legumes which is normally grown under soil and various intercropping system. Nutritionally, this crop is a great source of a cheap protein. We normally call it meat for the poor people, and it's also rich in terms of starch, vitamin, and minerals which are necessary for the livestock feed and human consumption. The production of cowpea can benefit farmers in the different ways. Farmer can consume it, can sell grain and fodder to generate income. And cowpea also help in terms of maximizing the land productivity through the process by increasing the nitrogen cycle through the process called biological nitrogen cycling. And this nitrogen cycling helps in terms of reducing the demand and the cost of nitrogen fertilizers to the resource-poor farmers. Furthermore, cowpea is one of the crops which can be used as a cover crops because it has the potential to conserve soil moisture and reduce soil erosion. Maize is the third most valuable cereal crops, which comes after wheat and rice. It is considered a major staple food crop for the black population of South Africa, and it has the potential to meet the global food demand. Maize is mainly produced by small-scale farmers which are situated in a region where we find that there's a marginal rainfall and the poor soil fertility. And the production of maize requires much nitrogen so that the farmer can be able to obtain the optimum yield. And this nitrogen can easily be made through the process of nitrogen fixation. And due to the cost of the nitrogen chemical fertilizers, the small-scale farmers now tend to practice the maize and legumes intercropping so that they can be able to replenish the soil mineral nitrogen through the process called biological nitrogen fixation. In South Africa, maize and cowpea intercropping is one of the dominant cropping system which is normally practiced by small-scale farmers to increase the crop production per unit area. However, the great challenge now faced by the researcher is finding a suitable combination that will increase the cowpea production when the maize population density is high under intercropping combination. So therefore, this study now seeks to investigate the impact of maize densities on the productivity of intercropped cowpea varieties. The goal of the studies was to evaluate the effect of maize density on the productivity of intercrop cowpea varieties. The study consisted of one objective, which was to determine the effect of maize density on the growth parameters and yield component of intercrop cowpea varieties. Methodology: the study was conducted at the University of Limpopo Aquaculture Research Unit area during 2022-2023 growing seasons, and the randomized complete block design was used in a 2x2 factorial arrangement with a 4 replication. The trial consisted of 8 treatments from 3 factors. The first factor was 2 maize density, whereby I had 24,700 plants per hectare maize, and the spacing was 90 by 45 centimeter. And the second density was 37,000 plants per hectare, and the spacing was 90 by 30 centimeter. The second factor was 2 cowpea varieties, whereby I had brown mix and Dr. Sanders. And the third factor was 2 cropping system, whereby I had a sole cropping and intercropping system. Data collected for cowpea, number of days to flowering, number of days to physiological maturity, the plant height, the plant biomass, the pod length and number of pods per plants, number of seeds per pods, 100 seed weight, and the grain yield were determined. Whereas for maize, only grain yield was determined, and I'm sure there will be a question, but it's fine. And the land equivalent ratio was also determined to evaluate the productivity of intercrop copy varieties. Then the data measured were subject to the analysis of variance using statistics 10.0 vision, and the least significant difference were performed for mean separation at 0.05 level of probability. Ladies and gentlemen, these are my results. So the table number 1 shows the result for the number of days to 50% flowering, days to 90% physiological maturity, plant height, plant biomass, pod length of 2 diverse copy varieties under different maize density. So the result presented in this slide, ladies and gentlemen, showed that there was a significant interaction between maize density and copy varieties in the mean number of days to 50% flowering, days to 90% physiological maturity, plant height, and also the pod length of copy varieties. And the higher mean days and the higher mean in terms of higher mean values in days to 50% flowering and days to 90% physiological maturity were observed when Dr. Sanders was intercropped with 24,700 plant per hectares and 37,000 plant per hectares. And this delay or this increase in terms of flowering and maturity can be explained by the shading effect of maize under intercropping combination. And then there was a significant interaction of maize density and cowpea varieties on the plant height of cowpea varieties. And the difference here in terms of the plant height of the cowpea varieties can be explained by the genetics of their response to the different maize densities under intercropping combination. And the post length of with brown mix was significantly reduced by the interaction of maize density and the cowpea varieties. And this reduction can be explained by the interspecific competition of the growth factors such as light, soil moisture content, and also the variation between the 2 cowpea varieties in terms of the pod length can be explained by the genetic makeup of cowpea varieties. The plant biomass was not significantly influenced by the interaction of maize density and cowpea varieties. Because the number of pods per plants, number of seeds per pods, 100 seed weight, and the grain yield of 2 diverse cowpea varieties under different maize density. The result presented in this slide shows that the number of pods per plants, 100 seed weight, and also grain yield were significantly influenced by maize density and cowpea varieties interaction. So when we look in terms of the number of pods per plants and the grain yield, you can see that there was a reduction, and this reduction can be explained by the fact that there was interspecific competition for the growth vectors and intercropping impact in terms of the citrus species like cowpea varieties. And the variation in terms of the 100 seed weight can be explained by the genetic makeup of cowpea varieties. Normally here in terms of the 100 seed weight, you may find that the cowpea varieties differs in terms of the seed size and the weight, and that determines the 100 seed weight. And the number of seeds per pod were not significantly influenced by the maize density and cowpea varieties interaction. Table number 3 shows the productivity of maize and cowpea intercropping. And the result presented here in this slide, ladies and gentlemen, shows that the partial land equivalent ratio for maize and cowpea ranged from 0.95 to 1.08 and 0.38 to 0.88 respectively. And when you look at this result, maize had a higher partial land equivalent ratio compared to cowpea, or I can say that maize was the major contributor in terms of partial land equivalent ratio compared to cowpea. And the reason previous studies explain this by saying that maize density, you may find that maize had a higher population density or the plant in an intercropping compared to cowpea. And another reason in terms of yielding higher compared to cowpea is that maize is a C4 plant. It normally has the potential to avoid photorespiration because carbon fixation, you may find that take place in 2 different cells, mesophyll cell and the bundle sheath cell, whereas cowpea is a C3 plant. It normally goes through photorespiration. And also cowpea uses a lot of energy to fix nitrogen, so that is why they yield lower than maize. The total land cover ratio ranged from 1.45 to 1.86, and this clearly showed a yield advantage for intercrop combination. Conclusion and recommendation, I can say that the mesh density and the copy varieties interaction is a component of copy, except the plant biomass and the number of seeds per pause. However, the rate of reduction differs in terms of copy varieties. And for this study, brown mix is considered a better performing crop for both soil and intercropping system because it has the potential to mature earlier and also recorded the higher grain yield as compared to Dr. Sanders. And the calculated land equivalent ratio was greater than 1. And according to the previous studies, land equivalent ratio greater than 1 is favorable for the irrigation. Ladies and gentlemen, I would like to thank my supervisor, Professor Mposi and Professor Mabapa, for their valuable guidance and support throughout this study. I'd like to vote of thanks to the plant production team, especially those who helped me when preparing the land, collecting data, and many more activities. And I also like to thank the University of Limpopo for the provision of resources, and special thanks to the Department of Agriculture, Land Reform, and Rural Development for funding my research work. Ladies and gentlemen, I thank you.
Thank you for the presentation. Let me take the questions. Any questions? Questions? There's one there. Two.
Good afternoon. I believe you use soil as your growing medium. So I want to know, how did you ensure that the soil properties match the ones that are required by your different crops?
Okay, thank you, Mr. Mafugatla. I just have one simple question. In one of your tables, table 1 especially, you have your CV percentages there, and then you have CV percentage of 48 point something for plant biomass. Please, you must explain that 48% what it means. Thank you.
Last one.
Thank you, Chair. So you concluded that you concluded, maybe if you can quickly go to that slide because I just didn't write it. It will be nice. Okay, yeah, you concluded that the interaction of your varieties influenced yield, but you mentioned that however it did not influence your biomass. So I wanted to check what was your measure, what did you measure your yield for you to be at a state to say your treatments improved or influenced your yield components?
Okay, one last one.
Thanks very much for a very good presentation. I actually, because they're saying one, I had two, but it's fine, I'll ask one. On that one that was asked by Dr. Mpai, you, based on your aim, which of course informed your design, where you have a factorial arrangement, but you did not conclude on that interaction where you had two cultivars of copies, maize, and the densities. There is no conclusion. Can you conclude on that basis, please?
You can take the stage.
All right, thank you very much for the questions. I'll start with the one for the soil properties required for the production of maize and copy varieties. Yes, of course. Before I can start conducting my research, I collected the soil sample so that I'll check in terms of the soil physical properties and also the chemical component of the soil at the location. And to be honest, I find that in terms of the soil physical properties were suitable for the production of maize and copy varieties. Because maize, the soil physical properties was sandy loamy soil. And then when I look in the cowpea thrive well under the well-drained sandy loamy soil, and also maize can also grow well under the well-drained sandy loamy soil. Then I'll go to the number 3, in terms of interaction. Why interaction influence the grain yield but did not influence the plant biomass? Okay, in terms of the yield, remember in terms of the yield, this the two tested copy varieties differs in terms of the growth habits and also in terms of the growth duration. And that it also influenced the grain yield of the copy varieties tested. And for this study, the early maturing varieties, which is brown mix, had a higher grain yield compared to the late maturing varieties, which is Dr. Sanders. So in terms of the grain yield, it all depends in terms of the adaptability of the copy varieties to the shading effect of maize. And it also depend in terms of, and also be influenced by the competition under intercropping combination. So, and also in terms of the biomass, said why there was no significant difference. So I can assume that there was no during the stage of the biomass, there was no competition, interspecific competition for the growth factors such as light, nutrients, and water. I'm not sure if you are answered. Okay, another one, the factorial arrangement. Why did I conclude? Can you please repeat your question, Dr. Sater? Matt, please repeat your question. I can't hear you.
Okay, he's saying that you will talk along the corridors. Okay, ladies and gentlemen, before you go for lunch, and then I've been advised that it's a break for lunch. Let's continue after the lunch. Thank you.
Hi, good day everyone. I hope everyone has had a nice lunch, eh? Okay, cool. So, my name is Mary Nkosi, and I'm gonna be presenting to you guys on the topic of application of mollusk in predicting and mapping land use changes, a case study of the Quaternary Encroachment X 2022J. And these are my supervisors. So this is the structure. This is the structure of my presentation. I'm going to start with the introduction, then end at the selected references. So, modules of land use change evaluation, in short, mollusk, is it's a computer application plugin which is used to analyze, model, and simulate or predict land use cover. So the plugin is equipped with some of the well-known algorithms such as the artificial neural network, the multi-criteria evaluation, and the Monte Carlo cellular automation, just to mention a few of those. So why should we predict or map land use changes? So like, why is it important? Like, we already know what's happening, so why should we do it? So, by predicting and mapping land use, we are ensuring that they were ensuring the sustainability in areas that are under development, and also this information can be used by urban planners and decision makers, so land use planners, to sort of help with coming up with strategies that will be more on the sustainability of the usage of land, and also the usage of water resources, well, natural resources, water use. Hey. This is just my study area, just a bit on the study area. The Quaternary Encroachment X 2022J is found in the Lawfield area, which is in Mpumalanga under the Crocodile River catchment, and it has a mean annual precipitation of 804.95 millimeters. And one of the major land uses in the catchment includes cultivation, urban development, and forest plantation. So just a bit on how I got my data and which data I needed for this. So I needed the DEM data, the OpenStreetMap, to sort of create the distance to road map, and also I needed satellite images for my land use classification. So the study covered a period of 33 years, which is from 1990 to 2023, and also a predicted period of 2040. Data analysis, how I classified my land uses. So the land uses were classified using the semi-automatic classification plugin on QGIS, which is an open source. And all pre-processing and post-processing were done using SCP, and the land use classes included built-up areas, cultivation, natural vegetation, water bodies, and wetlands. And for the prediction was done using Mollusk plugin in a QGIS version 2.84. You can see the difference in the version of QGIS. So the plugin, it only works on the older version of QGIS. It does not work on the new version of QGIS. And before you can start with everything, we first had to resample our images due to their different in resolution. So the resampling was done in RStudio using the nearest neighbor resampling method, and the DEM was used as an input. The image there just shows you that after resampling how the data input looked like. This is just how the whole modeling was done. I'm not going to go through this because it's going to take up a lot of time, but if you have questions, we can meet in the corridor. And model performance. So the first step after you've put the model is going to do the area change, which is also called the change detection. That's just to show you how land use have moved or shifted or how it has changed. And the other image, it just shows you the validation after the model has run. So in terms of land use, from the images that I classified, which is 1990 and 2023, you can see that there was an increase in built-up areas, while there was a decrease in cultivation and natural vegetation. And water bodies, there was also a decrease there. And wetland was like a decrease, which is also due to the image that was used to classify, because it's like the second Landsat, I think is Landsat 4 to 5. So the resolution is not as best as the Landsat 8-9. So discussion. The model scored a kappa coefficient of 0.78 and 83% of correctness, which by some studies is actually acceptable. And the study predicted an increase in built-up area of 10% from 2023, which also you can see from this result, hopefully it goes back, that it went from 14.54 to 24.49, which is like more than 50% increase in built-up areas. And while some classes, there was a decrease in them. So what does this mean? It means that these changes will most likely have a significant impact on climate change, erosion, air pollution, water quality deterioration, stream flow changes, just a change in the landscape which will have bad impacts on everything and how ecology works, because everything is connected like that. And therefore, land planners and urban planners, or any land use planners. So in conclusion, my results indicated the applicability of mollusk plugin in simulating and predicting land use in the South African landscape. And the nice thing about this model is the data that was used is all open source, like Prof was explaining earlier, like it's open source, so you don't need to worry about where you're gonna get the data. So anyone can use this, and it's very easy to use. That's what I like most about it, is its simplicity and that the data is easily obtainable. And another conclusion that I can add is that I focus mostly on built-up areas because this is part of my PhD, which is going to be looking mostly on built-up areas. But you can do the model to sort of look at other land uses, like the grassland or the savannas, just to see how it increases and decreases as it goes. And these are some of the selected references. Baie dankie.
Thank you very much for the presentation. Let me take this opportunity to take some questions. We have a question there.
The first question is, which classification algorithm did you use? Because you just said semi-automatic classification in QGIS, and justify why did you use that one? And another question is on kappa. Why did you use kappa instead of confusion matrix?
Okay, another question? Seems like there's no any other question. Then you can respond to the questions.
Okay, so thank you for that for those questions. So I love working with open sources. I use semi-automatic is because I was using QGIS, and QGIS this plugin is on QGIS for land use classification and everything. I know some people use ARC, and ARC does things differently, but because I'm working with QGIS, this is how we do it there. Yeah. And in terms of kappa, so the plugin only uses the kappa as validation. So the kappa results I've presented were only for the predicted years, but I did the matrix assessment for the classification. I just don't have the results because this presentation was more focused on the mollusk plugin. Is that okay? I answered. You good? Yeah.
Thank you very much. Thank you. And then our next presenter is going to be Matome Mkholo.
The surname is M-O. Okay. Yes, that one. Good afternoon, everyone. My name is Mkholo MJ, and I will be presenting to you about the work, the research study that we did in the past 2 growing seasons. The study is titled Sunflower Growth and Grain Yield under Different Tillage Systems and Sources of Organic Manure, supervised by Dr. Ziriski and Dr. Mzeze. Okay, we'll start with introduction. Sunflower is one of the largest grain crops in South Africa. Factors that contribute to this low yield. Infield rainwater harvesting technique is one of the soil, and it can increase the grain yield by more than 50 soil nutrient depletion. Organic manure, it can improve the crop yield and also soil properties. Therefore, the integration of this technique and animal manure will enhance the sunflower productivity under clay textured soil. And also, again, this studies also overlooked the problem of soil fertility. So it has been hypothesized that the benefits of infield rainwater harvest further could be enhanced further by its combination with organic manure. So soil nutrient depletion, water stress, and high fertilizer costs are some of the constraints that normally contribute to this low crop yield. So most studies which were conducted under these techniques have focused mainly on runoff, to minimize runoff and also evaporation by using mulches. But in most cases, these mulches are normally used as animal feed. So the use of organic manure, as they are readily available in bulk for smallholder farmers, can be used to improve the crop yield. So rainwater harvesting techniques such as pits, stone bands, which have showed to increase yield considerably when they were combined with organic manure. So therefore, infield rainwater harvesting technique and the manure application will cap the problem of water and nutrient shortages as experienced by smallholder farmers within the province. So this study will aim to investigate the synergistic interaction between water conservation practices and organic soil enrichment and their combined influence on optimizing sunflower cultivation under 2 soil types. So the objective will be to investigate the effect of this technique combined with organic manure application rate on sunflower growth and grain yield on 2 soil types. So the description of the study area. The study was conducted under 2 sites, which is University of Limpopo Experimental Farm, referred to as Sefergale, and also at the University of Venda Experimental Farm, referred to as Univen. This study was conducted for 2 consecutive seasons, which is 2021, 2022, and also 2022, 2023 cropping seasons. The soils at Sefergale and at Univen are sandy loam and clay soils, respectively. Both sites falls under semi-arid environments with low rainfalls. The treatment and design. This study used a split plot design and replicated 3 times. So the main plots were the 2 tillage systems, which was infield rainwater harvesting technique, as I described it before, and also the conventional tillage system. And the subplots were poultry manure and cattle manure applied at a rate of 0, 20, and 30 tons per hectare. And then the main plot measured 45 meters by 4.5 meters, whereas the area of subplot were 9 meters by 4.5 meters. So, data collection, sunflower seeds were planted in tram lines, row spacing of 2 by 1 meters, with 30 centimeter as an intra-row spacing. So the grain yield was determined during the maturity stage, where the 2 most inner rows were harvested, and then the grain yield was adjusted to 13% moisture content, and it was reported in kgs per hectare. Head diameter was determined using a measuring tape before drying the head of sunflower stage, flowering stage, and the maturity stage. And also the plant height and the stem girth were also determined or measured under those growing stages. Data analysis. Data was analyzed using analysis of variance using the SPSS Vision 20 software, and the treatment means were separated using LSD at a probability level of less than 0.05. In table 1 shows the pre-cropping selected surface soil properties of the two. Start looking at the pH. So the pH at the Sefergale was a little bit higher or slightly alkaline, whereas at Univen was slightly acidic. If you look at the particle size distribution, the clay content at the Sefergale was a little bit lower with 15%, whereas at Univen was a little bit higher. So table 2 depicts the chemical properties of the organic manure used during the experiment, which is cattle manure and poultry manure. So the cattle manure had higher pH values as compared to poultry manure. But if we look at the total carbon, total nitrogen and phosphorus, poultry manure recorded higher values as compared to cattle manure. So table 3 shows the monthly rainfall data during the two cropping seasons at both sides. So if we look at the first cropping season, you will see that the rainfall pattern or the distribution of the rainfall was evenly distributed in the first cropping season at Univen side and also at Sefergale. In the second cropping season, both sides experienced an uneven distribution of rainfall, as it can be seen in the month of March, April, where particularly at Univen in April, about 19.6 millimeters of rainfall were received in one day for that particular month. So table 4 depicts the means of sunflower grain yield, head diameter, and head dry matter as influenced by tillage system and manure rate application at Sefergale. So the tillage system had a significant effect on the grain yield, both in the first cropping season and also in the second cropping season. And also it had a significant effect on the other parameters, like head diameter, and also infield rainwater harvesting technique has shown to be recording high mean values, and both in the first and also in the second cropping season. These high mean values under this grain yield by this technique could have been contributed by its ability to harvest more water as compared to conventional tillage. So table 5 depicts the means of sunflower above ground diameter, plant height, stem gild, under different stages, under different growing stages as influenced by tillage system and manure rate at Sefergale. So the tillage system had a significant effect on above ground diameter, both in the first and in the second cropping season. And then have a significant effect also under this parameter. If we look at the plant height, we see that tillage system had a significant effect in the first growing season only during the flower bud stage, and also during the flower bud stage, and also in the second cropping season, the significant effect was also observed, was only observed during the flowering stage and also maturity stage. So table 6 now depicts the means of sunflower grain yield and head diameter, as influenced by tillage system at Univen site. So the tillage system in this instance again has reported high mean values under grain yield and also under high diameter, under head diameter in both seasons. But it can also be seen that the tillage system reported the high first cropping season. This can be mainly because of the tillage system as compared to the implementation in the first cropping season, whereas it was the ability of this technique, the infield rainwater harvesting, in the first growing season, but it significant effect on the grain yield, head diameter, and head dry matter in the first growing season and also in the second cropping season, with poultry manure at a rate of 35 rate. So table 7 also depicts the sunflower above ground dry matter, plant height and stem yield as influenced by the tillage system and the manure rate at Univen. So here, as you can see, the tillage system also had a significant effect on 3 parameters under all growing stage seasons. Again, here it can be seen that the infield rainwater harvesting technique also reported high mean values, or particularly under in both the manure rate application also under all growing seasons on all with okay, thank you. With okay, thank you. And also both in the first and the second cropping season. So conclusion, water harvesting technique and poultry manure at a rate of amounts and sandy loams to clay textured soils. These are the references which we used, and I would like to thank you.
Great, based on the fact that you have collected your data at 2 places. Did you do any heterogeneity test in order to check, the effects of those individual places? The postdoc test, did you do any postdoc test? And which one was it?
Thanks, good work.
To that, almost about 7. We have seed treatments that can help to reduce this kind of problem in our fields. This involves the seed treatments such as seed scarification and seed priming. For the purpose of this presentation, we're going to look at the mechanical scarification and hydropriming. Mechanical scarification involves the alteration of the seed coat, where it can be removed by knife or nickel or sandpaper to allow water intake from the soil to the seed. So for hydropriming as well, it involves the hydration and the drying of the seed to activate the pregerminative metabolic processes. So all these methods have been proved to improve germination and establishment in cowpea legumes by many from literature. So the study was aimed at investigating the mechanical scarification on cowpea cultivars as well hydropriming on Bambara groundnuts to improve their emergence and stand establishment. The following objectives were conducted to achieve this aim. Number 1, to assess the influence of mechanical scarification, to improve or on emergence of cowpea cultivars, and to assess the influence of hydropriming on the emergence and stand establishment of emergence of the cowpea cultivars, and the hydration of this hard seed coat, the hydration of Bambara groundnut seeds with emergence and the stand establishment of the Bambara groundnut cultivars. So the study were performed at the University of Limpopo Soil Laboratory Lab, where mechanically they were transplanted in they were planted in the soil, replicated three times inside plastic. Bambara groundnut seeds were soaked in water for 3 different periods, and after that they were then transferred into the shade house and transplanted in soil inside plastic pots, replicated 3 times. Was collected from this study was emergence time to check the rate and the time spread of emergence. The emergence vigor was also calculated to measure the uniformity and the ability to yield strong or vigorous seedlings. Seedling biomass was also measured to see if the seeds has ability to capture and use resources efficiently. The data subjected from the study was data generated from the study was subjected to ANOVA using just that, and the means were compared using Duncan multiple range test at 5% probability level. And table 1 here shows the mechanical scarification interaction with the 2 cowpea cultivars. And from the observation of the coloration, it shows that the seedling biomass accumulation in was significantly different in the control and the scarified seeds. And here it is the hydropriming maneuver showing the interaction of hydropriming summary in the graphs. So here we have the interaction of the two cultivars with the mechanical scarification on the percentage emergence. And then no significant difference was found on this. This is shown by the similarity of the letters on the bars. And then from the observation, you can see even control performed better on the brown mix cultivar. For hydropriming, you can see that there was significant difference among the soaking periods, in the interaction of the priming and the two groundnut cultivars. Then for emergence vigor index, no significant difference was found with the interaction of mechanical scarification and percentage, vigor of the two cowpea cultivars. For hydropriming, we can see there was a significant difference, and then for mean emergence time, similarly, we didn't find any significant difference on both seed treatments. And then for seedling biomass, we can see that there was no significant difference found on the interaction of all this with the cultivars. And this is also seen by the similarity of the letters on the bars. So we can see on these results that mechanical scarification with the interaction reduced the emergence, the stand establishment, and the seedling biomass. So according to this, what are they saying to us? So the results suggest that the two cultivars of cowpea actually have high potential to provide maximum produce and have high crop stand emerge faster and can efficiently capture and use resources without the need for mechanical scarification. So what could have led to these results? One of them might be that the seeds were already bred for high emergence, so there was no need for mechanical treatment in the first place. So that is why even control was found to be better than the treated seeds. Then for hydropriming, we've seen the improvement in the establishment and the vigor in the two Bambara groundnuts. However, no significant difference was found in the biomass accumulation and the emergence time. So in conclusion, the results revealed that mechanical scarification did not improve any emergence of the two cultivars. Therefore, for future researchers, you are advised to use the seeds that usually rural farmers buy from the street that we are 100% sure that are not bred for any emergence, so that you can see the effectiveness of application of the treatment or the mechanical scarification in this case. Therefore, hydropriming from this study is recommended as the most adequate and inexpensive treatment to improve the uniformity of the seedling emergence. I would like to acknowledge supervisors and the technical team in conducting this study in the University of Limpopo. Thank you.
Thank you. So that was a nice, quite brief presentation. So I would like to call on questions and comments that will improve or will help her study to be good as she goes to finalize her work. Questions and suggestions, comments? Yes, there is one here. Our people. One side of it.
Well done, Matlonolo. You reached the finish line, and you did well. Just on a lighter note, why was the hydro priming had a better improved emergence and establishment? You gave reasons for the mechanical that did not, but I didn't hear anything, or perhaps I missed it.
All right, thank you. As expected, usually rural farmers, we buy seeds from our local markets. So those seeds we cannot say they are modified anyhow for anything. So as expected, we expected for them to perform better because of no modification was done on them. So that could have been one of the reasons. And another reason could be that the two groundnut cultivars they differed in terms of color, as I've mentioned in the problem that these legumes, they usually have a difference in the accumulation of the thickness of the coat. So therefore, that could have led to significant difference or the treatment to work on them. Thank you.
Yeah, there is another one there.
Thank you, Ms. Rapela, for the nice presentation. I just want to find out from you, what is the reasoning behind you subjecting the treatments to 5% probability? Thank you.
Thank you. What is the reason to subjecting 5% to 5% probability? Are we supposed to subject to another percent? I think that is just the...
Okay, so, let me see. In the field and in the greenhouse, it's not the same thing.
Oh, it is not the same thing?
Yes.
Okay.
So I was going to say, hence you have a greenhouse or glasshouse experiment, and you are subjecting that to 5%. You can explain why 5%?
I'm willing to learn. You can explain for me. Thank you.
Wonderful. IT, guys. IT, I think we are having a candidate who is visual. Let's bring them in.
Hello? All right, no problem. For as long as I'm audible. So, may I start the presentation? Okay, let me start. Let me try to share my screen. Is my screen visible? All right, so may I proceed? Can you please give me a go so that I can proceed? For presentation, okay, thank you so much. I really appreciate. Greetings. My name is Mr. Mlogo William Seboni. So today I will be sharing with you one of the projects that I'm currently pursuing for my master's studies. So this is one of the projects that were presented to the International Association for Impact Assessment in the previous weeks in order to generate some of the views, from the research field. So, by the way, I'm a master's student within the Department of Geography and Environmental Sciences at the University of Limpopo, under the very same School of Agriculture and Environmental Sciences, the Faculty of Science and Agriculture. So, just a brief background behind my study. Before I can proceed, this study is to evaluate the perception of the EIA practitioners on conducting the EIA follow-up tasks for feeding station projects, specifically in Limpopo province. So to define what EIA follow-up is for those who have not came up with the concept of EIA follow-up, I would say EIA follow-up is just simply the monitoring, evaluation, and management and communication about the environmental performance of a project or a plan. So the necessity for countries to perform a environmental impact assessment as an environmental protection tool was stipulated in Principle 17 of the United Nations on the Environment of 1992. Hence, the countries adopted the EIA as a tool to protect the environment and to safeguard the environment based on their legislature, based on their local resources, to meet the major sustainable development goals. So that's why we have the evolution of the EIA law of China of 2002, which they are currently applying and they are amending, and it's working effectively for them. Similarly, in South Africa, we have the requirements for environmental impact assessment being reserved under the National Environmental Management Act 107 of 1998. And then the most important aspect is that with EIA being mandated as a tool to achieve environmental sustainability, it implies that following up on its progress is very mandatory. This pins out the fact that without adequate EIA follow-up or without any follow-up into the project performance or to assess how the project is performing, then the purpose of having an EIA in a development project is not achieved. So what made this study to be important is that it was reported in studies in most cases that the EIA is mandated. We can see we are practicing environmental impact assessment, but then following up on its progress has become a global issue. That is the weakest step in the EIA implementation progress. And then, however, this is the case that is occurring worldwide. We have countries like United Kingdom that had limitations to EIA follow-up. And the limitations were thanks to some of the barriers that included the lack of implementation costs, the lack of enforcement by the EIA practitioners, and the lack of the resources in terms of human resources and the funds. And then, however, in countries like China, as well, there were disparities in the quality and the rigor of EIA follow-up, which these ones were posed by their EIA law of 2002, which provided only 2 conditions under which EIA follow-up can be conducted. That was exception to this situation because it has been 20 years of EIA practice in South Africa, but then yet with substantial difficulties and flaws that needs improvement. So taking you to the research techniques, that this study was aimed specifically for filling station projects. This is to learn from the filling station project because we have realized an increase in the number of filling station projects that were approved over the last 10 years. And as well, considering the fact that there has been virtually little research, in fact, little research on EIA follow-up for filling station projects. So this study it is aimed to explore and comprehend the EIA practitioners. Sampling was applied to request the projects, the project documents, EIA project documents being done, or they are being incorporated into the filling station projects. And secondly, to identify the challenges that are being faced when conducting the EIA follow-up for filling station projects in Limpopo. And then thirdly, it was objective to ascertain that the efficacy of follow-up in filling station projects. Okay, and then this study employed quantitative measures and qualitative measures through the structured interviews and the survey questionnaires. Here are the research findings for the study. So the perceptions regarding monitoring tasks of EIA follow-up, we realized that the practitioners highlighted that there is, of course, EIA follow-up practice in the filling station projects, even though there are some of the limitations under the monitoring tasks of EIA follow-up, which were tracked. So we can have a look at the predicted impacts of the a project are not compared to the actual resulting impacts of a project. And as well, a situation where the project and condition sites, evaluated. And from what we can see, that there is monitoring of the impacts, the monitoring of how the mitigation measures are being carried out for filling station projects. For example, we have got a situation where the environmental data is collected for that evidence to compare the baseline, information, other resulting impacts onwards. Secondly, the perceptions on which is one of the tasks for EA. That's the evaluation with conformance standards, the evaluation of conformance with the predictions and the performance, as well the evaluation of conformance with the expectations. We have realized that the overall effectiveness of the EI process is being carried out, but then whereas we still have some areas of improvement, that's where the project is the project assessment with performance of the standards is not achieved to that well. They are falling short. And then if we can have a look at a situation whereby some of the standards, they are accounted for, of course. The evaluation of conformance with standards is well taken care of. And we're also falling short at determining the overall effectiveness of the EIA process, of which is the main purpose of having a follow-up in development projects. Thirdly, we have the perceptions from the EIA practitioners on the management of issues and impacts in filling station projects. We have came to realize that they highlighted that the conduction of regular audits as part of their monitoring process is not successfully ensured. But then if we have a look at some of the areas where they implement the environmental management program to execute the projects and to follow up on the mitigation measures, one is taken care of. So from what we can deduce on their management techniques as practitioners is that it is taken care of to some extent. And then so we still have areas of improvement to make sure that we take the appropriate actions to strengthen how we manage the issues in environmental impact assessments. So we have came to realize that the outcomes are successfully incorporated for a future learning, more concerningly for similar development projects, meaning there is no way that we can one filling station to another. In this way, there is no information that is being made available to improve other future filling stations. Adequately provided to the responsible authorities when it comes to the environmental performance of filling station projects. But then some of the activities are well taken care of, where the outcomes are reported to the project proponent, but then there are some of the exceptions to the interested and affected parties, which forms part of the community. But then overall, from what we have deduced, we can realize that the practice when it improves so that we can be in a position to our EIA follow-up system in filling station projects. Hello? Oh, so quick. Okay, can I have at least 2 more minutes? Thank you.
Hi, Mr. Moloko. I'm afraid we may not have time for you to continue because we still have a long program. So, unfortunately, I'll only take one question for you so that they can mark your presentation. Can I have one question for him?
Thank you, sir.
There is one guy there. Quick.
Thank you so much for such a very great presentation. I have one question in EIA because I assume that you conducted this study. So please educate us.
Okay, the question is noted. Is it only one question?
Yes, we will only take one for you.
Okay. To reflect on your question, based on the specific strategies that we can deploy to improve the monetary, meaning the budgets and human financial resources. So with that being said, we can, at least as we can try to make sure that the project proponents, they make reserves for such, because those are the parties for conducting a EIA follow-up as the first parties. They should make sure that their project is compliant with the environment.
Thank you.
Thank you. I don't know if
Ministers.
I managed to answer your question.
Yes, they are smiling. It means they are good. Thank you.
Thank you so much.
Log him off. So, I may call on the next speaker, who is Mahoma Ribani. They will be telling us about the aquaponic system. Guys, Mahoma Ribani. Mahoma Ribani. Hello. 10 minutes. We are strict. Okay.
We called you to come and upload your things.
I did.
Okay, you did. It's fine.
Good afternoon, ladies and gentlemen. I am Mahuma Ribani. I'm a second-year master's student at aquaculture, and aquaponic system technology has become a very popular fish production technology. In South Africa, most of the aquaponics management strategies that are required to achieve optimal production of tilapia in aquaponic systems. Aquaponics integrates hydroponics with aquaculture. Hydroponics involves the production of plants in a soilless media, while aquaculture involves the production of fish in tanks. This study chose and we chose spinach because of its capability to absorb nutrients. We also chose it because it's the most consumed for optimal production of tilapia in aquaponic systems. One of the management strategies that we're going to look at is the number of times the fish is supposed to be fed when growing in aquaponic systems. This is my integrated aquaponic system. My integrated aquaponic system consisted of 9 fiberglass tanks of a volume of 500 liter, forming part of the aquaculture subsystem, and we had PVC pipes of 10 centimeter diameter forming part of the hydroponic subsystem. We used juveniles of Orochromis mozambicus weighing 37.68 gram, and they were stocked in fiberglass tanks at a density of 30 fish per tank. The fish were fed pelleted commercial feed of 4% of their body weight at 3 different feeding frequencies. The feeding frequency treatments were once, twice, and 3 times a day, and they were designated T1, T2, and T3. The Spinacia oleracea plants were stocked in hydroponic systems at a density of 18 plants per system. We looked at different water quality parameters to see whether the different feeding frequencies affected the quality of the water. We also looked at the growth performance of the fish in terms of specific growth rate, feed conversion ratio, and other parameters. We looked at the organosomatic indices of the fish to see if whether feeding frequency can affect the health of the fish. We also looked at blood parameters to see if whether the different treatments of feeding frequency can cause any stress in the fish. Our results were analyzed using SPSS, ANOVA at 5% significance level. These are the results of my water quality parameters. Our temperature was kept at 28 degrees Celsius and the pH range of the system was between 6.99 and 7.20. The temperature and the pH remained within acceptable limits for tilapia growth in aquaponics. There was no significant difference in dissolved oxygen levels in the different treatments. With regards to total dissolved solids, it was increasing with the increasing feeding frequency and also the increasing duration of the experiment. This was due to the accumulation of fish feed in the tank. Also, the significantly highest total dissolved solids concentration was observed in fish that were fed 3 times daily. This in itself does not mean that the total dissolved solid affected the fish. We'll only be able to see whether or not it affected the fish when we look at the growth performance and the organosomatic indices. The ammonia, nitrate, and phosphorus levels in the water on day 1 were below detection limits, hence they cannot be seen on these graphs. The ammonia and nitrate levels were observed to decrease with increasing duration of the experiment. This was because the nitrifying bacteria was converting the ammonia into nitrate and the nitrate into nitrate. Also, the phosphorus and nitrate levels were also decreasing with they were decreasing with the duration of the experiment. This was because the plants in the hydroponic system were taking up the nutrients that are in the water. We looked at the growth performance. The best growth performance in terms of specific growth rate and feed conversion ratio was observed in fish that were fed 3 times daily. This is because tilapia is a herbivorous fish and plant-based feeds contain low amounts of energy in them. And also tilapia has a small stomach. It allows, the stomach allows it to feed on little feed at a time. So the fish are, as a result, the fish is required to be fed more frequently. We looked at organosomatic indices also. The condition factor, and there was no significant difference in the condition factor and other organosomatic indices. This means that the fish were in good condition in all treatments of feeding frequency. We also looked at the hematological parameters of the fish to see if whether the fish were stressed. Most importantly, we looked at the glucose levels. As we can see here, there was no significant difference in glucose levels between the different treatments of feeding frequency. This shows that the fish were not stressed by the different feeding frequencies. The same applied for red blood cell count and white blood cell count. In conclusion, you can say that one management strategy that can be used to ensure optimal production of tilapia in aquaponic system is ensuring that the fish are fed 3 times daily. This experiment demonstrated that an aquaponic system can be run at various feeding frequencies without compromising the quality of the water. I would like to acknowledge my supervisor and co-supervisor, Prof. Neji Moyo and Prof. M.M. Rapatamalaji. I'd also like to acknowledge Mr. G. Gelden Hayes for the technical support.
So I think this is a very beautiful study that we are lacking, we are much interested in. Most people are not presenting in aquaculture here. So I would like to invite questions and comments for her. There is one there. Another hand for recognition.
Hello. I would like to thank you for your presentation. It really piqued my curiosity, especially when you were talking about the spinach. So I wanted to ask, what type of nutrients does the spinach absorb on your aquaponics? And also, what was the removal efficiency of those nutrients? And were there concerns with the fishes consuming the spinach? Okay. You wanted to know the type of nutrients the spinach was consuming. It's nitrates and phosphorus in the water. What was the other question? Whether or not the fish were eating the spinach. No, I don't know if you saw the design. They are separated, so there's no way in which the fish had access to the. Can you see this? These
Yes.
pipes represent the hydroponic systems. That's where. Oh, excuse. These pipes represent the hydroponic systems. That's where the plants were planted in these little holes. Can you see them?
Yes.
And the fish were in this tank, so they were not traveling or anything.
Okay, and also I asked about the removal efficiency of the nutrients.
We were not looking at that, unfortunately.
Okay, thank you.
Thank you.
Any other curious question? Okay, there's another one here. Hello.
Thanks. I know nothing about your work, but just out of interest, I think you mentioned plants per tank. I think you said 16.
18.
8.
18.
18.
Yes.
What informed the 18?
Usually, I think they use 20 plants per system. I just followed literature, actually.
But why?
Why I followed literature?
Why not 30? Oh, 100.
Why not? Because literature said it should be
Okay.
18 years or 20 at most.
So it means 18 was the optimum. So I would like to call on the next speaker. I'm sorry. Let me do that for you. Okay. So we'll call the next speaker, who is Hope Makua. Hope Makua will be telling us about the bulb and rhizobium influences of seed germination.
Before one was in German sites, just one who receiving yo. Not seeing come fast, fast. For sure, move fast. Or my sides.
Greetings everyone, I'm Makwa Hope. I'll be presenting my study titled N6 benzyl adenine and rhizobium influences on seed germination, seedling establishment and biomass of teparabine under supervision of Dr. R.L. Muladudi and Dr. P. Mangen. Teparabine is a leguminous crop belonging to Fabaceae family. The seed consists of phenolic compounds such as phenols and phytosteroids and phenols and isoflavonoids, which are responsible for color and taste and can be used to combat different types of diseases such as cancer. BAP is a plant hormone that regulates the plant growth and development. Rhizobium inoculation with Bradyrhizobium japonicum increase the plant height and promote development of legumes. Those are teparabine seeds and there are different growing stages during germination. Teparabine production is limited by low germination, which result in uneven crop stand. It is slow to establish and it takes for about 6 to 9 days to emerge. Delayed germination affect the crop uniformity and yield. That picture shows different challenges faced in production of teparabine: low germination, no germination, and non-uniform. When intercrop with other non-leguminous crop, reduce the application of inorganic fertilizers. It promotes the rapidness and the uniform of germination, thus increase the biomass of teparabine. Studies have suggested that seed pre-treatment with BAP and microbial inoculation with Bradyrhizobium japonicum stimulate germination index, seedling establishment, and biomass. The aim of the study was to investigate the effect of BAP and Bradyrhizobium japonicum on germination, seedling establishment, and biomass. And the objective was to determine the effect of different BAP levels and Bradyrhizobium japonicum, both application and no application, on germination, seedling establishment, and biomass of teparabine. Looking on the methodology and analytical procedure, the study was conducted in the Department of Biodiversity and Plant Physiology at P Block, University of Limpopo, whereby teparabine seeds from Department of Agriculture, whereby teparabine seeds from Department of Plant Production, Soil Sciences and Agricultural Engineering were surface sterilized using liberated chlorine gas for 16 hours in a jar desiccate. 15 milligrams of benzyl amine purine was used to prepare the BA solution. And the experiment was laid out in a completely randomized design with 12 treatments and 4 replicates. Different teparabine seeds were pre-treated with different 6 levels of BAP. They were inoculated using Bradyrhizobium japonicum, and they were sown in plastic pots filled with vermiculite and organic compost at a ratio of 2:1, and distilled water was used to irrigate depending on the moisture content. Data collection, the total number of germinated seeds were collected to calculate germination parameters such as mean germination time, germination vigor index, and germination percentage. The obtained data was subjected to GenStar software, and text test was used to determine the mean separation among the treatment at 5 confidence level. Looking on the effect of BAP and BAR on germination percentage, there were no significant difference on germination percentage. However, if we're looking on the lower concentration of BAP and BAR, germination percentage was high. Looking on the seedling establishment parameters, we're going to check the epicotyle and hypocotyle. At low concentration with BAR application and BAP inoculation, with BAR inoculation and BAP application, The epicotyle was thicker at lower concentration. And when we're looking on 3.5, as the concentration increased, they decreased simultaneously. On the hypocotyle, looking on the lower concentration of BAP application and rhizobium inoculation, the hypocotyle was thicker and it reduced as the concentration increased. Looking on the effect of BAP on stem length, they were on low concentration, they were also high as compared to other parameters. Looking on the effect of BAP on fresh dry mass, the fresh dry mass was high at lower concentration, and it reduced as the concentration increased, and there was no significant difference. Even on this one, surprisingly, on this one, lower germination gradually increased as the leaves were dried. Looking on our discussion, the BAP and Bradyrhizobium japonicum had no effect difference on germination, seedling establishment, and biomass of taproot bean. This may be due to BAP, Bradyrhizobium japonicum inoculation, as it is mostly used in biological nitrogen fixation. This will be covered in my objective 3. The obtained results were in contrast with the findings by Mangena 2019, who reported that BAP stock solution improved the germination rate. Although there was no significant difference, the findings of the study suggest that a low concentration of BAP in combination with Bradyrhizobium japonicum positively affect germination, seedling establishment, and biomass. This study provided insights on the use of BAP in improving seed germination, seed establishment, and biomass. Therefore, for future research, I will conduct my study on morphology, anatomy, and biological nitrogen fixation of taproot bean and yield. I would like to acknowledge my supervisors, Dr. R.L. Maladudi and Dr. P. Mangena, also University of Limpopo, for providing resources. These are the list of references used to compile this presentation. I thank you.
Thank you, Hope. And I would like to welcome questions and comments for her. It's either you were too clear or they were lost, totally lost. So we believe that you were too clear, and thank you for okay, there is one there.
Thank you, Hope. I hope that you won't be angry. I just want to find out from you. On your conclusion, based on what you have found, on your findings, are you going to accept or reject your null hypothesis, or you'll do both? Thank you.
I'm going to reject. As there was no statistical significance.
Do we have one more? Okay. Thank you, Hope, and thank you for the questions and comments. So let's give her a round of applause. So we are moving on to the next speaker, Hendrina Manielo. Are you here? Okay, so she's telling us about the susceptibility of melodygine species in lemon balm. Thank you.
Manielo. Good afternoon, ladies and gentlemen. As introduced by the speaker, I'm going to take you through my presentation on relative susceptibility to 3 melodygine species in lemon balm, compiled together with my co-authors, Mashela, Pofu, and Malaj. Depicted is the lemon balm, scientifically known as Melissa officinalis, is an important medicinal plant which produces essential oils that have so many benefits, including antiviral and antibacterial suppression. Chemical nematode management strategies had been successfully used to manage nematode population density. However, due to their unfriendliness, products were withdrawn from the agrochemical market. Therefore, nematode resistance in terms of relative susceptibility has been proposed as a suitable measure for evaluation of nematode resistance in crops. Relative susceptibility in lemon balm to 3 melodygine species was investigated to establish if plant can be successfully cultivated without any challenges from nematodes. The aim of the study was development of empirical-based information of nematode resistance of lemon balm against thermolic melodygine species. Objective being to determine relative susceptibility descriptors for 3 melodygine species on lemon balm under greenhouse conditions. Null hypothesis was derived from the above objective. The study was conducted at Greenhouse Biotechnologies Research Center at University of Limpopo under greenhouse conditions, whereby in 3 separate trials, treatments were arranged in randomized complete block design with 6 replications. Lemon balm cuttings were set into 20 diameter plastic pots filled with pasteurized soil mixed with hygromix. Each plant was inoculated by dispensing with 300 eggs of 3 melodygine species using plastic syringe. Figure 1 shows the trial of lemon balm plants inoculated with melodygine enthrolo B. At 56 days after inoculation, nematode variables data were collected and data were further processed using relative susceptibility formula, whereby relative susceptibility is equal to reproductive potential of test nematode plant, which is lemon balm, divided by reproductive potential of susceptible crop, which is tomato floridate. Ladies and gentlemen,
Table 1 shows the reproductive potential of lemon balm and tomato plant inoculated separately with 3 melodigan species and related relative susceptibility values after 56 days of inoculation. Lemon balm inoculated with melodigan enthroloby, as highlighted with the blue color, relative susceptibility value was greater than 2.1, suggesting that melodigan enthroloby was highly susceptible to lemon balm. Hence, Collette 2020 reported that melodigan enthroloby was more aggressive than melodigan incognita and melodigan javanica, whereas lemon balm inoculated with melodigan incognita and melodigan javanica, as highlighted with the red color, relative susceptibility was ranging from 1.1 to 2.0, suggesting that they were moderately susceptible to lemon balm. This confirms with the Beda et al. Who reported that 2 melodigan species penetrate and damage the crops. In conclusion, lemon balm was confirmed as susceptible host. Therefore, lemon balm cannot be used as alternative strategy to manage nematode population density. Here are my references. I would like to thank University of Limpopo for allowing me to conduct my study and ARC for providing me with consumables. Thank you.
Okay, thank you so much, Hendrina. So I would like to welcome questions for her and comments for her project. She's working on nematodes. Okay, it looks you were too clear or people are lost, but I would like just to get one for her to get a mark that she answered questions. Okay. Okay.
For someone who's not in this field, how would you summarize your results for me to follow what your work is? Just make it simple for me in terms of something that I should remember from what you presented. It's too broad, I know. Maybe you can just say one thing that says this is what the work is about.
Okay. My research was based on the alternative management strategy of melodigan species since the chemical were withdrawn from the agrochemical markets because they were harmful to environment.
Okay, so I hope she managed to get a mark for that. She answered a question. So given that, let's give her a round of applause. So I'll be calling our next speaker, that is Velem Matsane. Velem Matsane will be telling us about Artemisia afra species. And he's talking about science data and machine learning. I think he's a mathematician. He does, yeah.
Good afternoon, and thank you for the opportunity to present my interesting research to you today. Standing before you, it's Matsane Velam. I'm going to take you through my research entitled The Use of Remote Sensing Machine Learning Algorithm in Detecting and Mapping the Spatial Distribution of Artemisia afra in the Sekhukhune district. Following is the outline of my presentation. Okay, I'll take you through the background. In history, plants have been used as the primary source for medicines to fight various diseases worldwide, with approximately 80% of the population depending on medicines as their primary source for healthcare. In Africa, especially in the developing countries, people still rely on medicinal plants to sustain their basic health care and generation of income. The large percentage is attributed to the availability of these medicinal plants, limited accessibility, and the high prices of modern medicines. In South Africa, approximately 75% of black population residing in both rural and urban regions depends on traditional medicines for their healthcare benefits. The alarming rise in diseases outbreak and the expansion in the medicine markets have led to a rapid depletion of medicinal plant resources, resulting in a rapid increase in the cultivation of medicinal plants. For instance, the outbreak of COVID-19, the coronavirus, has led to a saga in the cultivation of medicinal plants and the expansion of informal market for medicines. What motivated our study? Previous studies on medicinal plants mainly focused on the medicinal benefits and the extraction of the benefits, such as the oil that we get from medicinal plants, forgetting the most important part, which is the location. Therefore, according to our research knowledge, there is a lack of studies in investigating the spatial distribution of medicinal plants, with no exception to Artemisia afra. Artemisia afra gained popularity after it was promoted as having potential in treating the coronavirus, which is COVID-19, and it was tested in many studies. Understanding the spatial distribution of Artemisia afra plays a critical role in the conservation and management strategies. This study will provide an accurate and up-to-date information on the spatial distribution of Artemisia afra in the Sekhukhune district municipality. The aim of this study is to quantify the robustness of machine learning algorithm in mapping the spatial distribution of Artemisia afra in the Sekhukhune district, with the objective to map and quantify the spatial distribution of Artemisia afra using multispectral remote sensing data. This study was conducted in the Sekhukhune district, municipality located in the Limpopo province. This study comprises of 4 local municipalities, and it's experiencing an average annual temperature of plus or minus 23 degrees Celsius, annual precipitation of plus or minus 560 millimeters and it's located at the altitude of 494 meters above sea level. A significant portion of the land usage in the district is attributed to agriculture, where the commercial agriculture covers 7.7% of the district's land, and while the sustainable farming covers 18.1%. Figure 2 is the demonstration of the study area. It shows the study area map in terms of location in relation to South Africa and the province. Following is the material and methods. This study conducted in the Sekhukhune district, where the field survey were conducted in late summer, and we needed community members to participate, so we used the snowball and purposive sampling in selecting the participants. And the community members were, and the first site that we visited, it was the Fitahome local municipality, followed by the Great Tubatsi local municipality, and lastly it was the Makutu Tamara local municipality. This study site was chosen after the reports of unsustainable harvesting of medicinal plants in 2021. The data that collected were captured in Microsoft Excel for further processing, and the data were divided into 70% for training and 30% for validating the model, and then later on they were imported in ArcMap to create a shapefile for further analysis. In this study, we used Landsat 8 and Sentinel-2, which were atmospherically corrected images which were accessed via Google Earth Engine platform. The cloud masking techniques found in Google Earth Engine platform were used to eliminate unclear pixels that were caused by cloud interference throughout the entire collection of the images. The images were filtered based on the time interval, study area, and the percentage of cloud cover. Both images were resampled to 10 meter spatial resolution for accuracy purposes. The following images just show the kind of data that were collected, where the first one, which is A, is composite, and then B shows the Sentinel-2 composite image that were used. The image, in order to map the spatial distribution, we need to perform what's called image classification. So in this study, 2 commonly used machine learning algorithms were employed in classifying the remote sensing images that were collected, which is random forest and support vector machine. Literature have shown the potential of random forest and support vector machine in accurately classifying was performed in the Google Earth Engine, where the machine learning algorithm were applied to classify the images on the same platform. Our following is just the summary diagram of how, summarizing all the steps that we followed in answering the aim and the objective. Results and discussion. Figure 4 demonstrates the spatial distribution of Artemisia afra in adjacent to land cover types, where A, it represents random forest performed in Landsat 8, B, it support vector machine performed in Sentinel-2, where random forest performed in Sentinel-2 yielded high accuracy output with in mapping the distribution of Artemisia afra in the district, where it was found that most plants were distributed in the eastern part of the study area. The number D is the one that we selected for the final results based on the accuracy that was achieved there. So this one is just figure 5, it's just a demonstration when the other land cover types were excluded for accuracy visualization purposes. Accuracy assessment. Accuracy assessment were performed to assess how accurate the classification performed, and then the kappa coefficient was used to check or to quantify the agreement between the multi-class that were used in classifying the images. Here is just the demonstration of the user, figure 6, user and producer accuracy. They're just helping to read the legend of the figure. In conclusion, this study provided accurate information on the spatial distribution of Artemisia afra in the Sekhukhune district, with most being distributed in the eastern part of the study area. The output of this study are critical in conservation of Artemisia afra. Although Artemisia afra is not listed in the most threatened species, but this information is important for community members, because during data collection, they were complaining that there were people who were coming in numbers to come and harvest this plant, and the way that they were harvesting, it was unsustainable, and it might be extinct in the near future. I would like to acknowledge the following organizations, not forgetting my supervisors, Prof. Aidau and Ms. Mutapo, and also people who supported me as they follow. Following is the references that were used. Thank you.
Thank you, sir. I would like to call up on questions and comments for him. Yes.
Yeah, for Mark's purposes. So I have only one question with regard to your, machine learning, especially the model pruning. How many entries and entries did you really use in actually developing your models, and what really guided that?
Okay, thank you for such a mind-challenging question. The machine learning algorithms, we're using them in Google Earth Engine platform, where it used coding. So literature suggested that we can use 20, and others suggested 50. So we started using 20, and then we assessed the accuracy, and it was not that good. Then we followed the 50 because it was giving us accurate results.
Okay, it's interesting. Any other question? We have got 1 and 2, the last one.
Afternoon. Thank you for an interesting presentation. I would like to find out if you explored the spectral mixing between the Artemisia, excuse my pronunciation, and the other land covers. And then also, following up on the last question, a suggestion would be to explore how maybe changing the number of runs for your random forest as well as for your support vector machine. If you used Google Earth Engine, I'm assuming you used the basic, or not the basic, but what you have found there on the code. But if you play around with the mtry and the ntries, and in the support vector, that will be the number Cs in your runs. That might improve your classification. Thank you.
Thank you. I didn't get the first question.
The spectral mixing
Okay, thank you.
between your Artemisia and the other land covers.
Okay, to answer that one, in this presentation, I was just only presenting one objective of my thesis. So the spectral mixing, it was another objective. And then, yes, we did that. And then also on the second one, the follow-up, I didn't use the code as I downloaded it, or yes, I played around it. I did the entry, I changed, and then based on the accuracy, because we were looking to get accurate results, then we ended up deciding to use the 50.
Okay, thank you. You guys, you will engage outside. Next question. We've got a say here.
Just out of interest, what ground truthing data did you collect? I'm saying that because I've seen in one of your pictures that the plant was around, the streams, which also leads me to another question to say, what do you think underpins its spatial distribution, which that could also maybe help you look in future to model where, other areas where it could potentially be found?
Oh, okay. Firstly, to answer the one for ground truthing, we use the GPS to collect the coordinates. The presence only, yes. And then the modeling part is also part of the study, which is the third objective, and it was performed with accurate results.
Okay, thank you. I was told that we have to break now at 4 for tea. So maybe Prof. Chasi will want to say it. How are we going to do it? Okay, so he's saying we are breaking for 10 minutes, and after that we are coming back to finish up our session. Thank you. These guys should help. So those who did not upload their things, the presentation, let's finalize and upload for the last session.
Yeah, just tell him the presentation. Okay, sharp.
Oh, thank you.
sub-adults of Encroachment Mozambique subjected to handling stress under the supervision of Prof. Moyo and Dr. Mbokani. Within the introduction, in aquaculture, handling is one of the most used technique that can be used to trigger reproduction or when a fish is about to reproduce. So it shows that handling is one of the most used technique, and then it even proves to us that fish can be handled in order to be in order for it to be treated for pathogens, and then it shows that fish which are subjected to handling, fish subjected to handling, it encounters stress in such a sense that fish don't reproduce well, and others lead to high level of mortality. So we get to a point whereby we have to manage stress within the aquaculture development. So the best way to manage stress within the aquaculture development is by using the medicinal plants. Medicinal plants have been reported to contain compounds with anti-stress and antioxidant properties. So one of the plants that can be used to manage stress in tilapia culture is Artemisia afra, which is commonly known as the African woodworm or lengana. Others, they call it umhlonyane. Artemisia afra is widely distributed in Southern Africa, which is in South Africa, Namibia, Lesotho, Swaziland, and Zimbabwe. It contains several bioactive compounds such as tannins, saponins, phenolics, polyphenolics, essential oils, and alkaloids. Within the aims and objectives, the aim of the study is to reduce stress in Encroachment Mozambique subjected to handling stress when given the Artemisia afra essential oil. So I'm going to have two objectives. For the first objective, I'll be able to determine I'm determining the effect of Artemisia afra essential oil based diet on growth performance and feed utilization. The second one is to determine the effect of Artemisia afra essential oil based diet on hematology and immunological parameters in Encroachment Mozambique subjected to handling. Or for those who don't know, Encroachment Mozambique is called Mozambique tilapia. It's the commonly known species that is used in South Africa, and then it's widely known for its native immune system. Within materials and methods, the fish were acclimatized in Aquaculture Research Unit, University of Limpopo, South Africa, whereby a total of 180 Mozambique tilapia fish were stocked. And then within feed preparation, the Artemisia afra was purchased from the Zanen Market, Mopani District in Limpopo Province, South Africa. The study used 10% of Artemisia afra, which is mixed with ingredients such as maize gluten, maize meal, canola oil, canola meal, sunflower, and the likes of cellulose. So the motivation for us to use 10% within the study is because the literature showed that Mbokani and Moyo in 2018 used 3% to 12% of Artemisia afra, and it shows that 10% was the one that can be used that can be used to promote growth within tilapia species, but then it was used for disease resistance. So the fish were fed twice daily, 9 o'clock in the morning and 3 o'clock in the afternoon for 6 weeks. Within the experimental design, as you can see in the picture, on my left, I think it's on your right, on my left, this is the recirculation system whereby the fish were stocked within the tanks. Far there are tanks whereby fish were stocked inside the tanks. A completely randomized design was set up in the experiment, and the Artemisia afra based diet was randomly assigned to triplicate groups of 15 fish per tank. The water quality parameters were measured after every 2 days, and they were as follows. The water temperature was measured to be 28 degrees Celsius, the pH 7.5, the diluted oxygen 6.75, and the conductivity, it was measured as follows. Since I mentioned that the tilapia species was exposed to handling stress, they were exposed to 4 different groups, whereby I had the control, I had group B, the group B whereby the fish was confined out of the net for 10 minutes, and the group C were confined out of the net and they were handled for 15 minutes, and group D, they were confined out of the net and they were handled for 20 minutes. The statistical analysis we use, the SPSS, which is known as the Statistical Package and Service Solution, which is at a confidence interval of 95%. And ANOVA was used to analyze the data to determine the effect of handling on the parameters, whereby I was looking at growth, hematology, and immunology. On the results on growth performance. On growth performance, if you can look at the table there, looking at the specific growth rate as the written as SGR abbreviated there, it shows that as the level of handling increases, it decreases the level of specific growth rate. It shows that this fish was not feeding well on the Artemisia afra based diet. And the feed conversion ratio, it increased with increasing levels of handling. And an increasing level in feed conversion ratio, it shows that the fish growth was compromised at this point. So, and then even with the survival rate decreased with increasing levels of handling. The results that correspond with the ones that were conducted by the Gentoft et al. Reported that handling stress has shown to be associated with lower growth performance and feed utilization in rainbow trout. It raises some questions whereby we expected the level of handling to increase with increasing levels of growth performance. So at this point, the growth performance was decreasing. It is due to the fact that the Artemisia afra is palatable. It contains tannins and saponins, as I've mentioned before. So those tannins and saponins, they can compromise the growth performance of a fish. So the results which are found in this study, they correspond with the one that was found by Mbokani and Zaki et al. Within the hematological parameters, we have the white blood cells, red blood cells, and hematopoietic cells. The white blood cells, I'll explain it when I'm explaining the innate immunology, because white blood cells, they can be used to detect the stress level, and they can act as a first-line defense against pathogens or stressors. So in this point, since I'm handling the fish, they can be used to detect the level of stress within the fish. So the red blood cells decreased, the red blood cells and hematocrit count, the HCT and the RBC, they decreased with increasing levels of handling, and there was significant difference between the handled groups and the control. Looking at the hemoglobin count increased with increasing levels of handling, although there was no significant difference. Within the mean corpuscular volume, the mean corpuscular volume, it decreased with increasing levels of handling, and there was significant difference in group C and D when you're comparing into control. So going to the platelet counts increased with increasing levels of handling, and there was significant difference in group B and group C when comparing to the control group. And thus it leads to a point whereby the decrease in the red blood cells and hematocrit cells, it shows that the Artemisia afra, it failed to assist the Orycmus mozambicus with the Artemisia afra failed to support the Artemisia afra, whereby it led to a point whereby the fish was induced to the stressed anemia. So the stressed anemia, it leads to a point whereby the fish can be exposed to increased heart rate and and blood flow, which can lead to dilation of red blood cells, which lowers the hematocrit count. So since I mentioned that the hemoglobin count were higher in the handled groups, it shows that the Artemisia afra inclusion helped with binding and carrying oxygen from the gills to the tissue of the fish. And the mean corpuscular volume was higher in the handled groups than the control. And since it was higher, it shows that the red blood cells that was lost, it was compensated by the mean corpuscular volume. It shows that Artemisia afra can be able to compensate the amount of oxygen that was lost. Within the platelet counts, it shows that Artemisia afra aided recovery from injuries and prevention from excessive bleeding. So it shows that the ability of medicinal plants to assist platelet in preventing excessive bleeding when blood vessels are damaged have been reported by Gabriel when the catfish was fed with the aloe vera. Within the immunology, the white blood cells, they increased with increasing levels of handling. It shows that the increase of handling, it shows that Artemisia afra can improve the defense in tilapia culture, which results in stress resistance. And it can stimulate the leukopoiesis. The leukopoiesis is the formation of white blood cells. The lysozyme activity, The increase in ecosystem activity was also evident in fish handled compared to the control, although there was no significant difference. But then it shows us that the Artemisia afra aided in helping to weaken the cell walls of bacteria or stressors, making it easier for phagocytes to engulf and destroy them. The plasma cortisol decreased in increased levels of the decrease in plasma cortisol levels are known to be associated with stronger innate response in fish. In conclusion, the growth performance declined with increasing levels of handling when given Artemisia afra. In fish handled culture, I would like to acknowledge my supervisor, Prof. Moyo, Dr. Mbokani, and our technician, Mr. Holden Hayes. Thank you.
Thanks very much for the nice presentation. Before I invite the questions, I would like to encourage actually questions, because I feel that we're not asking questions enough, which for me, it defeats the purpose. This is part of peer review process. Some of the students have not yet submitted their work. So it's always important to ask questions so that they can improve. Thanks very much.
Thank you so much, Chairperson and Moloto, for your nice presentation. Can you please clarify? I heard you saying that this is what? Afra is palatable, right?
Yes.
And then it also have tannin saponins, which, according to my understanding, they fall under anti-nutritional factors.
Yes.
So how does the two work?
If I'm explaining quite well, since the saponins and tannins, they form part of the anti-nutritional factors, right, as you've said.
Yes.
So since they contain such, we call it the biological activities, whereby it can help a fish to fight or a fish to have a much stronger innate immunity. So in this sense of talking about Artemisia afra, the Artemisia afra, since it has high tannins and high saponins, at some point literature shows that high saponins and tannins, they're the ones that can help a fish to develop a strong innate immunity. But then in this sense, it affects the growth of a fish.
Okay, have you considered the levels of tannins that the body of the fish can tolerate? How much?
Nah, I haven't checked that.
Okay. I think it's also important for you to check that.
Thank you very much.
Thank you.
Thank you so much for also a very good presentation. I have no idea of the work that you do, so I'll go straight into statistics. I saw that you did some means and stuff like that. So I'm just interested, like, which mean separation technique did you use and why did you use that?
I used the SP, I can go back, I can use the SPSS, whereby I was checking the relationship between, let's say for instance, since I mentioned that I have different types of handling levels, right? So within different types of handling levels, let's say for instance, we're looking at the weight gain. I want to check how does it differ, the control, how is it different from group D. So I don't know if I answered your question.
No, maybe you didn't get it well. What I wanted is the post-hoc test in your ANOVA. Like, what did you use to separate your means?
Oh, post-hoc. Take it post-hoc.
Yeah.
Thank you. Sorry?
Which one did you use, the post-hoc test, for your ANOVA? I saw that you separated your means and stuff like that. So I'm just interested to know which one did you use and why did you use it?
Oh, the reason I used the Teki post-hoc, it was the Duncan test, Teki post-hoc, Duncan test, which is able to separate the means and the standard deviations. So it's one that I use.
Thanks very much. Thanks very much. No, he has answered. He said he used Tuki. Thanks very much. The next presenter is Eva.
Good afternoon. I'm Ivane Pale, a PhD candidate at the Aquaculture Research Unit under the supervision of Professor Moyo and Professor Rapadzama Laji. I'm going to take you through a study on dietary full-fat stink bug meal effects on growth performance, blood chemistry, liver and intestinal histology of juvenile Mozambique tilapia. Fish feed is the most expensive component in any aquaculture venture. It contributes over 60% of the total aquaculture production. And fish meal is the main protein source. But there's been a decline in wild catch, an increase in demand for fish meal, and a decrease in fish meal availability. And this has made fish meal price to escalate. So in the aquaculture sector, a search for an alternative protein source is the top research priority. We need to find alternative to make aquaculture sustainable. And researchers have used plant-based protein sources, such as the soybean meal, the sunflower meal, and the cottonseed meal. But there's a lot of challenges with these plant-based protein sources, such as the imbalance in fatty acid and the amino acid profile, and also the presence of anti-nutritional factors. So currently, researchers are focusing on insects. Insects, they've been deemed as the potential protein source globally. The advantage of the insect is that they are animal-based protein sources, so they are nutritious. Some of them even contain the amino acid that is comparable to fish meal. And also, fish meal form part of the natural diet of insects in the wild. And different insects are found in different geographical areas. So it is important for us to search for insects that are within our geographical areas. There's been some insects that have been used. I won't go much into details on that, like the mopani worm. And the challenge with the mopani worm is that it is expensive, very expensive, as expensive as the fish meal. So there is an insect that has not been looked at before, and this study has identified the stink bug, which is Encroachment delegogi. The stink bug is widely distributed in the subtropical open woods bushveld of the northern provinces of South Africa. It is a delicacy in some of the tribes in Limpopo provinces and is commonly known as Tongolipa. Unlike other insects that are harvested during the rainy season, the stink bug is harvested during winter, which makes it one of the insects that is available when other insects are scarce. Its nutritional profile is known to contain a very high fat content, but there is very little that is known about its amino acid and the fatty acid profile. So the distribution of the stink bug, it coincides with the distribution of the Encroachment mozambicus, which is the Mozambique tilapia. It is one of the most widely cultured warm water freshwater species in South Africa. It is a species of economic importance because it plays a role in income generation for local fish farmers and also as a gross domestic product in South Africa. Therefore, the specific objective of this study was to determine the effect of dietary full-fat stink bug meal on the growth performance, blood serum chemistry, liver and intestinal histology of juvenile Encroachment mozambicus. Going through the materials and the methods, the stink bug was purchased from the Tendo open market in the Wembe district of Limpopo province, South Africa, and we went on to assess its proximate composition, the mineral content, and its amino acid and fatty acid profile. 5 dyes were formulated to contain the same amount of protein, energy, and fat, and they substituted fish meal at 0, 10, 30, 50, and 70% inclusion levels. This study was conducted at the Aquaculture Research Unit, and 15 fish were stocked randomly at fiberglass tanks in triple Ks that were in the recirculating aquaculture system. The fish were fed twice a day for 8 weeks to apparent satiation. And this study has been approved for ethical clearance by the University of Limpopo Animal Research Ethical Committee. For water quality parameters, the water quality was monitored to to maintain the requirements for Orochromis mozambicus. And for growth performance indices, these are the growth performances that were assessed. We also further went on to assess the organosomatic indices and the blood serum chemistry using the serum that was centrifuged from the blood samples. For liver and intestinal histology, the samples were preserved in formalin solution and prepared for H&E staining, which were then later assessed using a light microscope. They were then graded and quantified. For statistical analysis, the data of the study was tested for normality and for homogeneity, and this was all done using an SPSS. Looking at the results, this study shows that indeed the stink bug contained a very high fat content, as you can see, at 48.59%, and the protein content was only 29.53. The dominant mineral in this insect was found to be sodium. This insect, it contained all the essential amino acids and the non-essential amino acid, and the most dominating amino acid was lysine, which is one of the essential amino acids. Looking at its fatty acid profile, this insect contained an omega-6 fatty acid, which is linoleic acid, but the whole insect was mainly dominated by the saturated fatty acid, like most terrestrial insects. Looking at the growth performance parameters, we have noticed that there's been a decline in growth performance parameters with an increase in stink bug male inclusion. This was confirmed by the weight gain, the specific growth rate, the thermal growth coefficient, and other growth parameters. And the decline in growth performance has been attributed to the imbalance in the amino acid profile. Although it contained all the essential amino acids, these amino acids does not meet the requirements for Orochromis mozambicus. And also, it showed that it has imbalanced fatty acid profile. Like most terrestrial insects, it lacks the EPA and the DHA fatty acids, which are the essential fatty acids for growth. Another thing, the decline in growth performance can also be attributed to the chitin content. Chitin has been known to interfere with the digestion of nutrients, and that has been, it has also contributed to the decline in growth performance that has been observed. Also, with an increase in stink bug meal inclusion, we have noticed a decline in feed intake. This was attributed to the compounds that the stink bug releases as a defense mechanism. This is the trans-2 decenal and the trans-2 octanol compounds. These are the compounds that the stink bug releases as a defense mechanism, and it is known to repel predators. And also, it releases an unpleasant odor. So this odor was also observed in the dyes, and it is speculated that it has resulted in a decline in voluntary feed intake. But that was not the case with the organosomatic indices. They shows that the fish were not under stressful conditions, and the health status of the liver was not affected. This was further confirmed by the ALT and the AST, which are commonly known as the liver enzymes, that they were not significantly affected across all the diets. Again, we have looked at the villi thickness and the height, and we have noticed that across all the dyes, there was no significant differences. Also, looking at the goblet cells, which is a cell that releases mucus as a protective measure when the fish is under stressful conditions, there was no significant difference. Looking at the liver, when it was quantified, all the fish across all the dyes, they recorded a score of 1, which shows a normal liver histology. This was further confirmed by the clear micrographs of the intestine of the Orochromis mozambicus that shows distinguishable villi, lamina propria, epithelial lifting, serosa, and the submucosa. Also, again, looking at the liver histology, the micrographs showed normal liver histology across all the dyes, showing distinguishable nucleus. We can then conclude that inclusion of the stink bug meal in the diets of Orochromis mozambicus results in a decline in growth performance. But the case is opposite with organosomatic indices, which showed that the fish were not under stressful conditions, and the liver in the intestine showed normal micrographs. But there are limitations with this insect. It contains a very high fat content, which can prone the dyes to oxidation, and also its limited availability and seasonal variation. So we can only recommend that the stink bug can replace fish meal up to only 30% inclusion level. And we also recommend that the cost benefit analysis be determined be assessed, analyzed to determine its economic viability. This work that I've just shared with you here has been published at the Cogent Food and Agriculture Journal, which is under the Taylor and Francis Publishing House. It is one of the DHET accredited journals. You can go online for more details. I would like to acknowledge my supervisor, Professor Moyo, Professor Rapazamalaji, and our technician, Mr. Gavin Helden Hayes, for his technical assistance. The University of Limpopo and the National Research Foundations are acknowledged for financial support. Thank you.
Thanks very much. That was a good presentation. Indeed, she is the CIC of her presentation. I would like to invite questions.
Thank you very much for the nice presentation. I just want to get something from you. With the results that you have found or you got, do you think you managed to solve the problem that was intended to be managed or solved? If yes, how?
To some extent, yes. We have noticed that although there's been a decline in growth performance with an inclusion of the stick bag meal, but it can replace fish meal up to only 30% inclusion level. In that way, we've managed to solve a problem that at least you can be able to replace fish meal to a certain percentage. That will reduce your feed production and also increase your aquaculture production at large.
Just to follow up on this, I'm interested in your response. Why would you prefer a 30% reduction in a fish meal over other parameters that were going the opposite? What made you to say, this is better than the other? Do you get what I mean?
No. I don't know which parameters you...
No, what I'm saying, the question was this a success? And you mentioned the reduction, or I think 30% replacement?
To some extent, it was a success, but only up to 30%. But there's been protein sources that have replaced the fish meal, and maybe others went up to 50 to 60. But particularly this insect could only go to 30%, because at 30%, fish species were still able to convert a food intake into the weight gain. Like the specific growth rate was close to 1 at 30% inclusion level. In that way, we can still replace 30% of the fish meal and reduce the feed cost, which is the main reason for this research.
I'm not disputing that, but I'm saying if you look at it in totality, the fact that the growth, I just remember the growth parameter was affected. They did not grow, am I right? With the different inclusion level. Am I correct?
That's correct.
But I'm saying if you look in totality, I'm saying in responding to the question, would you still say it's a success to some degree given the reduction of the fish meal?
Oh yes, it is still a success, because we have rural fish farmers that don't even have an access to fish meal itself. So if this insect is at their disposal and they can just still find it at the markets or still go and harvest them themselves, they will still be able to save a lot, increase production in their aquaculture ventures. No, it's not really at the expense of growth, because as long as the specific growth rate is close to 1, which is not significantly different from the control diet, we can still, yes. I was still saying, as long as the specific growth rate is at 1, it shows that the fish was able to convert the diet, the feed, into the weight gain. So there's a specific amount of weight gain that was included, but not up to our expectation, or not as fast as the control, which is the fish meal.
Okay, thank you so much, Eva, for your nice presentation. Your study is so interesting. Under your background, you were so firm about mobile worms being so expensive. So I just want to know what makes them to be so expensive. And then the second one, based on the limitation that you have highlighted, I think on your last slide. So can you still say that these stink bugs can still be recommended to be used,
Okay, let me
especially with small-scale farmers?
Oh, okay, let me start with the first one. With the mopane worm, there's been a study that has been done by Moya and Rapazza that shows that the mopane worm is a very nutritious insect, but it has been shown that the price of the mopane worm in the market is just as expensive as fish meal. Although it resulted in good growth performance or it yielded positive results, but it is not recommended for fish farmers. Because what is the point of replacing something that is just as expensive as something that you're trying to replace? So it is not recommended. Coming to the second one, looking at the limitations that I have raised here, it's limited availability. The thing is that it can be used for farmers that are in the geographical area of where this insect is used. So limited availability can be a challenge for farmers that are far or outside the geographical area of this insect. And again, with the high fat content, it can be defattened.
Okay, and then when you...
Thanks very much,
Oh,
Doctor.
sorry.
Thanks very much. Round of applause. Sorry for that. Next presenter, it's Ramahasha. It's Molobe. Molobe, are you here? Oh. Oh, okay. Can I have Makonyani? Makonyani. You are here. Oh, who are you? Malibala. No.
Molebala. Okay, it's fine. Not really, I think Can I answer? Good afternoon, everyone. I am Molebala Mpho, and I'll be taking you through my research project compiled together with my co-authors Pofu, Malaji, and Mashela. The title of the research project is The Relative Susceptibility of ARC cowpea cultivar to Melodygine enterolobi, Melodygine javanica, and Melodygine incognita. Cowpea is scientifically known as Vigna unguiculata, and cowpea is known for its important in-soil improvement, nitrogen fixation, and the prevention of soil erosion. In cowpea production, root knot nematodes are one of the most damaging soil-borne pests that causes a significant threat to both smallholder and commercial farmers. The genus has over 5,000 plant species and can be able to reduce the yield from as high as 50% to a complete crop failure. Over the past century, chemical synthetic nematicides were relied upon for the management of the plant parasitic nematodes. However, due to their unfriendliness to the environment, they were withdrawn from the agrochemical market. And the use of resistance in plants has become important as a management alternative, and it is suitable for integrated pest management. Specific and should be identified using lengthy experiments along with the mechanism of resistance. Certain crops, for example, potatoes and watermelon, do not have resistance to this melodigain species, and such crops rely on using sorry about that. Let's continue with the problem statement. I was saying that the resistance in crops is nematode specific and should be identified using lengthy experiments along with mechanisms of nematode resistance. Certain crops, for example, potatoes and watermelon, do not have resistance to these melodigain species, and such crops rely on using crops with nematode resistance in order to be able to suppress the nematode population densities. The Agricultural Research Council is researching and developing a wide range of leguminous crops with limited information on the host state have high population density of these three melodigain species, and currently there is no genotype among the potato cultivars with resistance to melodigain species. Cowpea cultivar, due to their status as a low input crop, they could be ideal for use in crop rotations that is intended. Potato is a main crop. Cowpea cultivars were sown in a 20 centimeter diameter plastic pots, and the inoculum was prepared by extracting X and J2 from the roots of the nematode susceptible tomato. Each cultivar was inoculated by dispensing approximately 250 X using a 20 centimilliliter plastic syringe, which was inoculated into a deep hole of about 5 centimeter, and it was covered with a growth medium. The cowpea cultivars were irrigated after every other day with 250 milliliter. The insects and pests were monitored daily. These are the trials for the three melodigain species, whereby the first one was for melodigain enterolobi, followed by javanica and incognita. At harvest, the relative susceptibility was assessed at 56 days after inoculation, whereby the roots of the cowpea cultivars were uprooted per plant and they were submerged into the water in order to free them from the adhering soil particles, and the excess water was removed. From there, the nematodes were extracted from the roots per plant by maceration and blending, which was followed by sugar flotation and the centrifugation methods. The eggs were counted under the stereo microscope. The data were subjected to relative susceptibility, whereby the relative susceptibility was calculated by the reproductive potential of the test plant, whereby in this case is the cowpea cultivar per the reproductive potential of a susceptible plant, whereby in this case is the tomato, whereby the reproductive potential was calculated by the proportion of the final population per the population of the initial population. Relative susceptibility depicts that it is resistant when it is ranging from 0,1 to 0 to 1,1, and it is moderately susceptible when it is ranging from 1,1 to 2,0, or it is highly susceptible when it is greater than 2. Table 1 shows the reproductive potential of the ARC VIMP cowpea cultivars and the tomato plants which were inoculated separately with the 3 melodygine species and the relative susceptibility values at 56 days after inoculation. Ladies and gentlemen, we are going to focus on the relative susceptibility, which is denoted as RSA. Looking into Melodygine interlobi and javanica, the relative susceptibility shows that it was found to be less than unity, meaning that it was resistant to the test plant, and suggesting that the cowpea cultivars cannot be used in crop rotation, can be used in crop rotation that is intended to manage the population density of these 2 nematodes. Whereby, with regards to M. incognita, it shows that cowpea cultivar VOP and VOP CO7 and cowpea cultivar VOP CO4, they show that they were greater than 1, which suggested that they were susceptible to the test nematode and they cannot be used in order to manage the population density of Melodygine incognita. The relative susceptibility test showed that all the cowpea cultivars were less than unity except for these 2 cultivars in M. incognita. And we recommend that the mechanism of resistance should further be established in order to determine whether this ARC cowpea cultivar could be used in introgression during the plant breeding program. In conclusion, all the cowpea cultivars except for these 2 cultivars on Melodygine incognita, they were better suited for use in crop rotations that is intended to suppress the population density of the test nematodes. These are some of the references that were used, and I would like to acknowledge the University of Limpopo for enrollment, the Agricultural Research Council and Potato South Africa for the consumables, and I would like to thank the National Research Foundation of South Africa for funding. Thank you.
Thanks very much. Good presentation. Questions, comments? Questions, comments? There's a question there. That's next to you.
Thank you, Mpo. A quick one. Just tell us the difference between, you are talking a lot about cultivar, this, that. Difference between cultivar and variety. Thank you.
The difference between a cultivar and a variety. Are you asking this because you don't understand it or what? Because
I'd like to interfere. Can you answer the question?
Okay. The difference between a variety and a cultivar. A cultivar is regarded as a plant whereby it has been tested or investigated for different things. And with regards to a variety, it is a plant or a crop whereby it has not been tested for different things, or it has not been investigated.
Thanks very much for answering, even though you are not in plant breeding, but thanks for the answer. Any other question? So it looks like we do not have any question. Thanks very much. The last but not the least, Leifa.
Okay. I think so. I don't know how we do it. Yes. So my 10 minutes starts now. All right. Good afternoon, ladies and gentlemen. As indicated by the program director, my name is Lefamo Konyani, and under the supervision and co-supervision of Dr. Nkwana and Dr. Gidi respectively, I present to you the determinants of the competitiveness of the South African citrus fruit industry and Armington approach. This is basically a presentation outline. The citrus industry consists of 4 broad categories, which are oranges, soft citrus, grapefruits, and lemons. Citrus production happens to be one of the most important agricultural activities in South Africa because citrus farming is a labor-intensive economic activity with high growth potential. In addition, the South African citrus industry is export-oriented. And because it is export-oriented, the exportation of citrus fruit the exportation of citrus fruits are one of the key determinants of economic growth. Because of that, there has been a need for South African producers to maintain export competitiveness in the South African citrus industry. In addition to that, it is also important because there has been the deregulation of the citrus industry market. Export competitiveness ensures that the countries produce better quality products with better production methods. Therefore, competitiveness is an important aspect of strategic market planning. Because of that, it is important to sustain and enhance the competitiveness. Therefore, it brings us to the aims and objectives of the study. The aim of the study was to investigate the factors that influence the competitiveness of the South African citrus industry. Therefore, the objective was drawn, and it was to analyze the short and long-run factors that influence the competitiveness of the South African citrus industry from 1989 to 2019. And because of that, the hypothesis was drawn from the objectives. The study employed secondary annual time series data, which was collected from Faustat from 1989 to 2019, and the data was analyzed using eViews and Excel. To ensure that the objective was fulfilled, the study employed the Armington model. This is because the Armington model takes into consideration the trade patterns, and also it takes into consideration various factors that could affect export competitiveness. Therefore, the model was specified as the average market share, which is equals to the constant, the logarithm of the price, exchange rate, specialization, and dummy variable that represents South Africa being a part of the World Trade Organization. And also, because we are using secondary data, the study employed the Dickey-Fuller test to test for stationarity, and also the Q-sum squares test. Based on the table projected, it illustrates the Dickey-Fuller test results, and the Dickey-Fuller test results illustrates that the average market share is significant at 5% level due to the T-stat being greater than the critical value at level. And also, all of the variables, which is exchange rate, price, and specialization, are significant at 5% level at first difference. The table projected illustrates the long-run results of the study of the Armington model, and it states that the constant is 3.65, which indicates that when all factors are kept constant, then the South African market share is equivalent to 3.65. And the specialization, which is illustrated by the RCA, which is illustrated by the RCA, indicates that when South Africa specializes, or where there's a 1% increase in the specialization of the South African citrus industry, there is a 0.18% decline in the in the competitiveness. It is also important to note that all of the factors are significant at 5% level of significance, and also it is important to note that the dummy variable, or when South Africa is part of the World Trade Organization, in the long run, it implies that there's a 2.97% decrease in the competitiveness. This table also illustrates the short-run results, and the results state that when South Africa is a part of the World Trade Organization, there is a 0.19% increase in the market share in the short run. This is attributed because of when farmers are usually price takers. So in the short run, pardon, I made a mistake. When it comes to the price index, pardon, when I spoke about the price and farmers being price takers, it illustrates that in the short run, when there's a 1% increase in the price, the average market share declines, which implies that farmers are not able to quickly adjust to price changes. And also when it comes to the dummy variable, which is contradicting the results of the long run, the South Africa being a part of WTO in the short run leads to a positive impact, and also the probability or the results are found to be significant at 5% level. Also, it's also important to note that the Durbin-Watson value is equivalent to 1.633, which implies that there is a normal correlation, and also the adjusted R-squared is above 50%, which implies that 67% of the variables that are included in the model are of good fit. The QSUM test indicates the stability of the model, and looking at the graph that is displayed, the red lines represent the critical lines, and then the blue line represents the model, which implies that the model is of good fit. And because it is of good fit, then the results are good. And based on that, we can conclude that the study is rejecting the null hypothesis, which was that there is no long or short run factors that affect the competitiveness of the citrus industry, because price, specialization, South Africa being a part of the World Trade Organization, and exchange rate have a significant influence towards competitiveness in the long run. In addition, price and South Africa being a part of the World Trade Organization had a significant influence on the competitiveness of the South African citrus industry in the short run. And based off of that, we can recommend that the government and various stakeholders should subsidize citrus farmers in order to ensure that they can get inputs like fungicides, because when it came to the issue of specialization, the problem was that the reason why the specialization gave a negative influence was because of the emergence of the citrus black spot disease. So if they subsidize farmers in order to mitigate that, then the competitiveness of the citrus industry will increase. Secondly, there is a need, or the study recommends that they invest in research in terms of prevention of citrus black spot, and also diversify export markets so that it can, or the South African market can reduce its reliance upon the World Trade Organization. And markets that they can look into include BRICS, the African Union, amongst others. And this is basically a synopsis of the references that I used in this presentation. And also, I would like to acknowledge the Faculty of Science and Agriculture. I'd like to acknowledge God as well, and I would like to acknowledge my supervisor and co-supervisor and everyone who contributed to the study. With that said, I would like to thank you all for being a great audience. Thank you.
Thanks very much for that. Questions? I have two. I hope I'm not beaten because I'm next to you. So we actually do work on citrus. So thanks for mentioning the citrus black spot as one of the factors that actually affects the competitiveness. Have you looked into the Citrus Research International? The work they do, Citrus Research International?
Oh, the question is, have I looked into the work that Citrus? No, usually, I was looking into the work that the Citrus Growers Association, because the study is primarily based on South African citrus. So the main source of information was Faustat and Citrus Growers Association.
Okay, let me ask another one. Have you also looked into the quality of the fruit? Does it affect the competitiveness? For instance, if fruits has some disorders like your rim pitting, chilling injury, does it affect the competitiveness in the especially the European Union market?
My study is based on the competitiveness of my main study. So here I was presenting an objective. So in the study, I looked at different markets, which included the European Union. And then it was found that when it comes to the European Union, they place phytosanitary as the sanctions on citrus that is not of quality or that is not of the preference that they would prefer, if I make sense.
Okay, any question? Okay.
Thank you very much for the lovely presentation. I'm glad that he actually asked in terms of, other determinants in the competitiveness of the SA in citrus industry. But I would have loved if you could also, give more of, other factors that can influence that. But my question to you is, have you looked, or rather, how do you view the long-term and the short-term competitiveness of the South African citrus industry? For example, if it's yearly as compared to a decade. Thank you.
Thank you very much. With the short run and long run results, firstly, I would like to mention that the study period was from 1989 to 2019, which it basically implies that we looked at a 30-year period. So from that, with respect to short run, it's basically more or less immediate. So if now the price changes, how would citrus farmers react to that? Would it make them more competitive, or would it make them want to not supply or export products to those countries that they export to? And then with the long run, it basically looks at time lag. So it varies maybe from a year or two. So in a year or two, if the price changes, then citrus farmers are able to say we are holding back on exporting oranges or lemons or whatnot. I hope I answered your question. Thank you.
Okay, thanks very much. Also for a comment, we recently signed an agreement with China. So it means we are good in terms of that market. I'm just talking based on your conclusion, where you talked about BRICS. Thanks very much. Can we have a round of applause? Oh, I can see that we have a presenter who's not appearing here on the program. That is Mr. Bongo. I think he will be our last presenter for the day. What's your... Matat? Oh, okay. You can come through before he comes because he's not on the program.
Greetings, everyone. I am Muratsetla Mujaji, and I'll be presenting to you the results of the study which I conducted on examining households' food purchasing behavior during COVID-19 in Mokgolobutu Village, South Africa. This study was supervised by Dr. M.H. Lefopan. So in early December of 2019, a coronavirus was discovered in Wuhan City, Hubei Province, China. World Health Organization declared COVID-19 a pandemic on the 11th of March 2020. In South Africa, the first case of COVID-19 was reported on the 5th of March 2020. And because the cases of COVID-19 continue to be reported, a nationwide lockdown was declared on the 23rd of March 2020 in order to curb the spread of the virus and to protect lives. During the initial lockdown period, consumers were not allowed to leave their homes unless if they wanted to perform essential activities such as purchasing food. Businesses were then forced to close except for businesses that were manufacturing, supplying, or providing essential goods and services. So the lockdown has resulted in consumer stockpiling when numerous consumers purchase unexpectedly large quantities of food products in order to avoid food shortages and inflation. So there are several investigations that were conducted on food purchasing behavior during COVID-19. However, more of these studies were conducted during the initial lockdown period when the vaccinations were not available. So now that the lockdown restrictions have been relaxed and the vaccination is made available, the consumer purchasing behavior has changed. Hence, there was a need for this study to collect data after the lockdown restrictions were eased and the vaccinations were administered. So the aim of this study was to examine household food purchasing behavior during COVID-19 in Mokgolobutu Village. There were three objectives of this study, which were to identify and describe the socio-economic characteristics of households in Mokgolobutu Village, to determine changes in households' food purchasing behavior during COVID-19 in Mokgolobutu Village, and to analyze socio-economic factors influencing households' food purchasing behavior during COVID-19 in Mokgolobutu Village. So this study was conducted in Mokgolobutu Village, which is in Greater Zanini Municipality within Mopani District, Limpopo Province, South Africa. So according to Statistics South Africa, Mokgolobutu Village has a population of 6,256, which consists of 1,790 households. So the primary data was collected through a structured questionnaire where 327 households were interviewed face-to-face. Moreover, simple random sampling was used to select the households in order to allow each member of the population to be chosen. So the descriptive statistics were used to to address the first and the second objective, which are to identify and describe the socio-economic characteristics of households and to determine changes in household food purchasing behavior during COVID-19 in Mogoloboto village. So the ordered probit regression model was used to address the third objective, which is to analyze the socio-economic factors influencing household food purchasing behavior during COVID-19 in Mogoloboto village. So, the households were asked about the frequency in their food purchase in order to measure the changes in food purchasing behavior. So the results indicate that before the lockdown restrictions were relaxed, households purchased food less frequently. This is because households feared to contract the coronavirus and they wanted to avoid the long hours in the queue before they can enter the grocery stores. But after the lockdown restrictions were relaxed and the vaccinations were administered, the household purchased food more frequently. This therefore indicates that there are changes in food purchasing behavior during COVID-19 in Mogoloboto village. So the ordered probit regression model, as alluded before, was used to address the third objective. And the results indicate that before the lockdown restrictions were relaxed, gender, age, social grant, and food purchasing intention had a negative influence on food purchasing behavior. However, educational level, marital status, household size, type of employment, social relief of distress grant, monthly household income, change in savings, as well as advertisement influence had a positive influence on food purchasing behavior. But after the lockdown restrictions were relaxed, gender, type of employment, social grant, social relief of distress grant, and brand loyalty had a negative influence on food purchasing behavior. But age, advertisement influence, marital status, educational level, and type of employment had a positive influence on food purchasing behavior. So, this study has found that the household purchased food less frequently when the lockdown restrictions were tight, but purchased food more frequently after the lockdown restrictions have been eased. Therefore, this study recommends that grocery stores should consider launching an online store and establish initiatives to promote online grocery shopping in case a comparable crisis occurs in the future. This marks the end of my presentation. Thank you.
That was short. Thanks very much. Questions? Questions?
Thank you very much for the talk. I'm particularly interested in two aspects here, age and gender. Do you mind elaborating further why there was a change? Because initially you said age had a negative influence and after lockdown it had a positive influence. Why was that? And also why gender was also considered? Thank you.
Gender, it was considered because it is part of the socio-economic characteristics of the households. So, analyzing whether the socio-economic characteristics influence food purchasing behavior was one of the objectives. So, with the changes, I think the changes were because, like at the beginning, before lockdown restrictions were relaxed, the age had a negative influence on food purchasing behavior. But after the lockdown restrictions were relaxed, age has a positive influence on food purchasing behavior. I can say this it means that now that the lockdown restrictions are relaxed, it means that older people are more likely to purchase food after the lockdown restrictions are relaxed. So yeah.
Yes, you can follow up. Still have some few minutes.
Okay, so in terms of gender, this is just an example, by the way. Can we assume that maybe men were actually buying more food as compared to women? I just want to understand, the disparities of gender there.
Okay, in terms of gender, it says that before and after the lockdown restrictions were eased, gender had a negative influence on food purchasing behavior. So, with the finding of my studies, I found that females were more likely to have purchased food during COVID-19, which means females are responsible for purchasing food in their households rather than males.
Okay, thank you. Last question, I promise.
Sorry for that.
Okay, not a problem. Thank you.
Sure. Thanks very much. Don't have time. We do not have time. Sorry. Can we then invite Mr. Bongo to come and wrap up?
Afternoon, ladies and gentlemen. Thank you very much for your grace and also allowing me to present. I was supposed to present yesterday, but the unexpected happened. Thank you very much. My name is Bango Happy. I will be taking you through my presentation under the title, Effect of Increasing NPK Fertilizer Rates on Growth of Jatropha zeeheri tea under Microplot Conditions, together with Professor Mposi and Dr. KG Shadu. Well, Jatropha zeeheri is commonly known as Sefapa wadia amongst Sepedi-speaking people. It was adopted mainly for its uses, more especially the rhizome, which is used to treat sexually transmitted infections, the urinary tract infection, menstrual pains, and also to ensure strong fetus development during pregnancy. However, we are focusing much on the leaves in this study, which were used or adopted to make a tea beverage in rural communities of South Africa. If you take a look at figure 1, it shows Jatropha zeeheri plant. Figure 2, it shows the plant at flowering stage. Figure 3 is the fruit of the plant. Now, commercialization of indigenous herbal teas has gained popularity in recent years. You mentioned the bush tea, the rooibos, and others. Jatropha zeeheri is amongst indigenous teas which are utilized traditionally with these multiple uses. However, successful domestication of this crop will require improved soil nutrient status through the application of NPK fertilizer. NPK fertilizer in tea production improves physical and enhances the quality when applied correctly. However, we have realized that inappropriate application of fertilizer can be detrimental not only to the test crop but also to the environment itself, causing pollution. Therefore, it is necessary to assess different NPK fertilizer rates that will maximize productivity while improving tea quality on physical properties. Therefore, influence of increasing NPK fertilizer rates requires assessment for successful production of this crop under microplot conditions. Now, the aim of this study was to determine fertilizer application rate that will promote physical properties associated with Jatropha zeeheri indigenous teas, high quality production under the said conditions. The objective of the study was to assess the influence of increasing NPK fertilizer rates on Jatropha zeheri growth under microplot conditions. The study was conducted at the Aquaculture Research Unit at the University of Limpopo under the plant spacing of 30 centimeter by 30 centimeter for both intra and inter-row spacings. The study comprised of 6 treatments as named on the slides, which were arranged in a randomized complete block design with 5 replications. The study took almost 130 days to be completed. Figure 4 shows the domesticated Jatropha zeheri plant at flowering stage. Now, seeds were collected at Kurim village in South Africa. The treatments were applied 7 days after transplanting the seedlings, and multi-feed was applied. The data that was collected includes the vine length, stem diameter, chlorophyll content, the NDVI, number of leaves, the leaf width, the leaf length, leaf area index, dry shoot mass, and also dry root mass. Then the data collected was subjected to the analysis of variance using statistics 10.0 software. The treatment means were separated using the LSD test at the probability level of 5%. Significant treatment means were further subjected to the lines of best fit at the probability level of less or equals to 0.05. Now, if you take a look at table 1, it shows the results of the study. You will realize that there is leaf length on my far right, it will be your far left. It is highlighted in red. Leaf width was highly significant. The treatment contributed 66% in total treatment variation, followed by the variables which were significant, the vine length, stem diameter, and also the leaf length and dry root mass, which were highlighted in blue. For vine length, the treatment contributed 61% to total treatment variation. The stem diameter contributed 67% to total treatment variation. The leaf length contributed 68% to the total treatment variation, and the dry root mass contributed 49% to the total treatment variation. All these plant variables which were black, they were not significantly affected. Now, figure 5 shows the response of the mentioned plant variables, the vine length and the stem diameter. It shows how they responded to the increasing fertilizer rates under microplot conditions. You can see that as you increase the fertilizer rate, the vine length was dropping, and also for the stem diameter. The same trend was followed under the leaf width, the leaf length, and also the dry root mass. Therefore, we had to come up with an optimum NPK fertilizer rate, because the pattern at which this NPK fertilizer has affected crops, we had to come up with a rate that will optimize the plant variables rather than destroying the plant variables. Now, we came up with the optimum NPK fertilizer application rate using the means that we got from the respective plant variables. The optimum application rate was developed at, or was optimized at 3.0 grams per plant. Now, in conclusion, Jatropha zeheri requires low quantities of NPK fertilizer for increased plant growth. We have observed the density-dependent growth patterns, whereby at low concentration or application rates, there was a stimulation, and at high NPK fertilizer rates, there was a toxic response. NPK fertilizer for Jatropha zeheri was optimized at 3.30 gram per plant NPK mixture, which can be translated to 33 kilograms NPK mixture per hectare. And as our results appears to be like this, we can conclude that this crop really appears to qualify as a low input tea crop. I would like to acknowledge this institution, the University of Limpopo, the NRF, and Technology Innovation Agency. Thank you very much for your time.
Thanks very much, Mr. Bongo. I'd like to invite questions. Yes, there is a question there next.
Don't say it's a question, it's a sort of comment, but thank you, Happiness. I cannot say it's a challenging question, but it needs you to say something. So, throughout your research, what did you enjoy most throughout whatever challenges you came up about until now? Thank you.
Thank you very much, sir, for the question. Well, I was very challenged, more especially with regard to the study itself, more especially the objectives, because my study is not only focusing on the plant variables, it focuses mostly on the microbial composition. After optimizing the NPK fertilizer, we are also looking at the phytochemical constituents, the antioxidants, and also the mineral elements, how they respond to the increasing levels of this crop.
There is a hand here. Yes, I saw it. Fortunately, the hand is talking. I saw it. Sir, we have had a lot of talks about this kind of tea. And I was asking myself, I think we will still have more and more. Is there anything significant which is different from the previous researchers? Thank you very much.
Thank you very much for the question. Well, with regard to the previous research that has been done, myself, I'm focusing much on domesticating the crop. Taking the crop from the wild, bringing it home, providing it with the necessary nutrient that is required for its growth. And after that, I wanted to optimize the level of NPK that will enhance the growth of the crop itself and also improving the quality of the crop. Thank you.
Any other question? Let me ask Mr. Bongo. I'm just worried that you have 0 as your treatment. For me, especially because you are from plant production, if you are not applying any fertilizer to a plant and then you compare it with where you have applied the fertilizer, automatically, for me, you know that the one with fertilizer is going to grow compared to the one that is not applied. Do you think that is fair? In terms of plant, of course, if it was a woman being, it was going to be something else. That is the first question. I just want you to finish all the questions when you're answering. The last one.
Let me answer the first question so that, well, with regard to that, more especially in this study, I have realized that at some point, the control that we are talking about, whereby I was not applying anything, it was much better compared to the highest level of NPK fertilizer application, which can be explained by the toxicity that the NPK fertilizer has on the crop. So there were certain variables that were better on control rather than the highest NPK fertilizer rate.
Yeah, thanks very much. That is expected. Probably you could have went for 10% instead of 0, and then you compare with others. That was going to tell you something. But also, like I indicated, is part of a peer review. You can also do the transplanting shock, which might have been the reason why you are having those results. But the last one, I think Mr. Mashau is here, he will assist. Can you use ANOVA to optimize?
Okay, you can use ANOVA. ANOVA will give you the means, right? After getting the means, you subject them to Excel. Excel will give you the graphs together with the quadratic equations. Now, you use the quadratic equations, right, to develop the optimum rate.
Oh, so you use quadratic, not ANOVA?
Yes, yes.
Okay, no, it was not clear. Thanks very much. No question? I think this was our last presenter. I hope there is something that you want to say something, Prof? Yeah, Prof will take over. Thanks very much.
Thank you. It's been another long day. How does this work? I'm a bit tall. Thank you, everybody. It's been a very long day, but a very productive day nevertheless. And I would like to thank you all for staying up to the end at 6 o'clock. I would like to thank the chairpersons who have been helping us chair the different sessions throughout the day. I would like to thank our invited guests who are also helping us together with the adjudicators who are doing a very tough duty of helping to assess the presentations by our students. And of course, our media team, we appreciate all that you are doing to make sure that this is seamless. The presenters, our students, all of you, this has been an amazing day. I have learned a lot myself, a lot of interesting research coming out of your work. I'm very proud of it. The supervisors, of course, should actually take a lot of the credit for guiding you to produce the work that you are producing. Of course, ultimately, one of the key outputs of this conference would be if this work is translated into publications. You know, we don't want it to all end in that faculty research day or the gala dinner, and then that's it. The next steps are to ensure that the research that you have presented here actually is published. Anyway, thank you. Tomorrow is the same. We meet here again at 7:30. The transport arrangements remain the same for those who are using university arranged transport. And then please note that tomorrow our program, the first part of the program, which is the science part, will end at lunchtime. It'll end at 1 o'clock and then lunch. And then we will then proceed at 5 o'clock with the gala dinner. So if you are using university transport, you are encouraged to please bring your, because the tradition is that people will have nice clothes to wear afterwards. So if you have some fancy clothes that you would like to wear for the gala dinner, please bring them in a separate bag. Then you will have the opportunity to put them on and powder and do all the makeup and all of that. Otherwise, do have yourself a good evening, and we will see you again tomorrow bright and early. Thank you.
Thank you.